Hello students, welcome to NAC Biology. This is Neelavati, your biology lecturer. So today I am going to start the first chapter of class 10th biology syllabus, that is nothing but the life processes. It is one short video, okay, for your board exam. I know many students are going to feel difficult as soon as they enter into the class 10th because they see that the chapters are very vast. They think the chapters are very difficult to understand, but don't worry, here I am with you to help, okay? See, if you just watch this video, it is a one short video, you will feel how the chapter is so easy and so beautifully we can understand. Because for every concept, I am going to explain by drawing diagram, okay? Because for many concepts, the diagrams are not given in your textbook. So that's why, so my opinion is, my way of teaching is to explain the concept, to make you understand the concept through diagram, so that the pictures speak 1,000 words, correct? So if you just imagine the things through picture, automatically that will retain into your mind. So that is what my concept. So definitely you are going to love that also, because after every concept, when it get finished, when you read your textbook, then you feel how easy the topic was, because each and every word, each and every line of your textbook will become so easy to understand after watching the video. But only one thing you have to do is, you have to watch the video, that is complete video you have to watch, that is from beginning to end, without skipping any topic, without skipping any part. So that is what the sincere effort you have to do, okay? Now the question, ma'am, the video is very lengthy. We want only this particular topic, but this video is too lengthy, how we need to watch the entire 5 or 6 hour video. So this is what the complaint from many students. Don't worry. So here is a solution for that problem also. What is that? Look at here. Can you see? Here, you can see a word called more, correct? If you just click on that word more, you are going to see a description box. In that description box, you can see here, the left side, the timings are given, and the right side, the relevant topics are also mentioned. Suppose if you want to watch a particular topic where you want to watch it again, then you click on the topic so that it will directly take you to that topic in the video so that you can watch it. For example, if you want to understand a topic called nutrition or a topic called transportation, then you just click on the time so that it will directly take you to that topic called transportation, okay? But directly don't do this. First, you have to watch the video completely, okay? Once you understand all the concept, all the topic, then if you want to rewatch it again, you can use that solution. Understood? So without wasting time, let's begin. First, listen the class, then you read your textbook, okay? Yes. First, before telling the overview of the chapter, what exactly we are going to study here, first let me tell you what is the difference between living and non-living. So this is very childish question because you are studying this from your childhood itself, but still let us recall it again. If I simply ask you a question, what is the difference between living and non-living? Or how can you say this organism is living or this thing is living, this one is non-living? How we are going to describe? So if a person is not aware about any science, what he is going to tell? For example, imagine if he just look at a man or a human being, he just look at its action. Oh, he is doing some action, he is talking, correct? He is walking. So based on its movement, he say that he is doing all the action. Of course, he is living. So like that, if I ask a common man, can you say this animal is living or non-living? What he says? He says, yes, it is living. Because that animal is making sound, maybe it's a barking or something else if it is a dog. Like that, the animal is walking, it is showing the movement. So the same question, if I ask to that common man, now tell me whether this plant is living or non-living, because it is not doing any action. It is not making any sound, it is not walking. But still, that person can able to say that plant is also living. How? Because that plant, earlier it was this much height. Now after few years, the plant will grow. So the growth is a property of a living thing. So based on certain qualities, that means what I said, the qualities like a movement, the qualities like growth, correct? So based on such quality, we can say, yes, this organism is living. Suppose if you look at a rock or this pen, it is not showing any movement, it is not growing. So you say, obviously, it is non-living, correct? That means, here in your textbook, in the first page itself, they are describing the moment you can consider this criteria to define an organism is living or non-living. But do you consider or do you think all organisms are going to show the movement? For example, see, this man is sleeping. Imagine the man is sleeping, or a dog is sleeping, that is, animal is sleeping. Then, do you say that it is not making sound, it is not showing movement, it is a non-living? Definitely not. Why? Because you are going to use some other criteria. You are going to think that though the person is not walking or making sound, but he is respiring, he is breathing, right? So animal is breathing. That means we are going to consider the respiration is also one of the criteria to describe an organism is living or non-living. Correct? So like such, many criteria we can list, many features, many characteristic features we can list in order to say an organism is living or non-living. But if you ask the same question to a biologist, imagine he is a biologist. Okay, so this person is a biologist like me. Now you go and ask the same question to this biologist. See, that man is not, he's not showing any movement. Or any organism, it is not showing any movement. How can you say this is living? So we won't consider the movement as a criteria. Then biologist says, he will get angry because he says, of course, I'm going to consider the movement as the main criteria. Though the movement is not visible to your naked eyes, but still within the cell, the movement is happening. Say, for example, look at here. If I take an amoeba or any cell you take, inside nucleus will be there and cytoplasm will be showing movement. Of course, the molecules which are so present inside the cell, they are going to show the movement. So, if I am saying a multicellular organism, how it is going to form? Of course, by combination of cell. So, more and more cell combine to become tissue. So, if a cell is showing a movement, then obviously tissue is also showing movement. Like that, the tissue become what? Organ. It is a basic knowledge. Everybody knows about this. And organ system. I am not explaining all this thing in detail because these are basics. And from organ system, many organ system together performing a function. For example, digestive system, excretory system, correct? And a transporting system. So all these systems which are so present in an organism that together make an organism to live. So that make an organism. That means we are going to consider movement as a criteria, man. Without having a movement, how an organism is going to live. So this is what the argument of biologist. But fine, okay. But in this chapter, we are not considering only a movement criteria. Or we are not just considering only criteria, most important criteria that must be present in an organism. But many criteria are there. But mainly we are considering few criteria to define whether an organism is living or non-living. Okay. So that means the overview of the chapter. What is the overview of this chapter? Here in this chapter, we are considering mainly 4 processes. What are those processes? Processes means what? As I said, the movement or a locomotion, it is also one of the process. But here, there are certain criteria. Criteria itself is nothing but the life process, which is responsible to maintain a life of an organism. Okay? How a movement within the cell is responsible to maintain a life of an organism. Like that, nutrition is a process. Without taking food, how we are going to live? So, that is one of the main function that an organism is going to do. So, in this chapter, we are going to study about this. So, like that. Another main topic in this chapter we are going to cover is nothing but respiration. See, imagine here is a person. So just now I told the person has to do certain action. He has to do many activities so that we can say that he is living. Suppose if you consider this property only to explain that the person is living, he must have energy, right? So he has to eat food. So that process of taking food and utilizing it in its body is nothing but nutrition. So he is going to ingest the thing. He is going to taking the food. So the food, what he is going to take, some organism can prepare their own food. For example, plants, they can prepare their own food. But some organism like human being and other animals, we are going to depend on plants. So in this topic, we are going to cover that area. How certain organisms like plants, they are going to prepare their food, and how the organisms like animals, fungus, they depend on the plants for their food, how it is going to digest in the body, how it is going to produce in a plant. So all those things we are going to cover in this topic. Okay? And after that, so imagine it is the digestive system of this person, esophagus, stomach, intestine, everything is there. So do you think once we have taken the food, immediately we are going to get energy? No, it is not like that. It has to enter into the blood. See, let's assume this is the blood vessel. So in this blood vessel, suppose the glucose, the carbohydrate, what we are going to take, like roti, chapati, that is rice, wheat, ragi, all those things, they are nothing but carbohydrate. So they will get broken down in the digestive system. So when it becomes simple molecule, then our blood is going to absorb it. So here the blood will refuse directly to absorb the complex food. So first the blood will say, first you just break into simple molecule, then I will absorb. So imagine it has broken into small pieces like glucose. Now the blood will get absorbed. So now this blood will take to different part of the body, because here only digestive system is taking the food, but our rest of the organ, for example, the leg is, hands require energy, our leg require energy, our eyes require energy, cheek require energy, every cell of our body require energy. How it is going to transmit that? It is through a special system called transportation. Through the blood it will run. So here, how this process is going to transmit to different part of the body, how the food is going to transmit. The air we are going to inhale, that is oxygen. We inhale it. I will come to respiration. Wait for a second. You are thinking, ma'am has skipped respiration. Ma'am must be not knowing about respiration. I know about respiration, but just wait for a second. So when we are just going to inhale the air, oxygen will enter. Do you think it will be written only in the lungs? Imagine the lungs are lying here. Do you think only in the lungs it will be present? No, my dears. It will be transmitted even to blood also. So this glucose, this oxygen, everything that will be taken from the blood, through the blood to different cells which are present in our body. That system we call it as a transportation. Now the question is, why all the cells require glucose and oxygen? So we need glucose is required to get energy. But why oxygen? I will tell you. So imagine here is a cell. Like that, overall body will be made up of cell. But for time being, just to make you understand, I am just drawing only one cell. Okay. So let me enlarge this cell. This is one cell, right? Let me enlarge this cell. I am writing here. So this cell is very upset. Why very upset? Because it is not having energy. Okay. So now, let's assume the blood that is through blood vessel, it is came near to the cell. This is our cell, right? So this blood vessel says, don't worry, my friend. I have brought glucose for you. So take this glucose. So now it become only half happy. Why not full happy? Because it says glucose is like a fuel, my friend. To break this fuel, what you require? If you have just put a petrol in your bike or in your two-wheeler, so is it possible to run? You need silencer pipe, right? If you just block the silencer pipe, what happens? You cannot ride the vehicle because you have to provide oxygen to burn that fuel, correct? So like that also in our body, to burn this fuel, that is glucose, G means glucose. We need to provide oxygen. So that's why this blood vessel says, don't worry, my friend, I have brought even oxygen for you. Take this oxygen. Take this, just enjoy. Now it becomes full. Okay? Because with the help of this oxygen, it will burn this glucose and finally it will provide energy. So our cell becomes so energetic. It becomes so happy. Okay, I got energy. So during this process, it releases a waste gas called carbon dioxide that we are going to exhale. Correct? So this process, what you are seeing now, within the cell, the exchange of gas is taking place. So carbon dioxide will be added to the blood vessel only. So again, it will give to the lungs. Lungs, you just excrete it out. So oxygen we are going to inhale, carbon dioxide we are going to exhale. That is nothing but breathing. But within the cell, oxygen is utilized, carbon dioxide will be removed. So that is called respiration. So in this chapter, we are going to learn about this respiration also in detail. And transportation, I said no. Just like how a cell got glucose and oxygen, it is not only a story of one cell. For all the cells which are so present in our body or any animal, they have to do the same work. So how it is possible? By a process called transportation. Correct? So in this topic, we are going to cover that. And finally, even it is the undigested waste, are so many reactions which are so happening in our body. After all this metabolic reaction, waste must be created. So that is what we are going to consider it as a nitrogenous waste. Correct? That is in the form of urine or fecal matter, anything which our body do not require. Okay. So are you going to store within our body? No, it will become poisonous. So we need to remove it. So the process of removal of waste is nothing but excretion. So in this topic, we are going to talk how this process will take place in the body of an organism. Understood? So this is what the overview of the chapter. So all these process, here only 4 processes they have mentioned, but apart from this, there are so many other processes like a reproduction, photosynthesis, many things are there, but our textbook has mentioned only this process. So all these processes will be keep going in a body of a person or an animal or any plant, whether you are going to sleep or you are awake or you are doing some work or anything or nothing. But still, all these processes are happening to maintain the life of an organism. If my body or my life, if it has to maintain, means in my body, whether I am simply sitting or standing or sleeping, anything I can do, it has to carry it. Okay. So that process together, which are doing their job now, we call it as life process. Got it? So this is what the definition given in our textbook. What is that? Life process means the process which together perform the function of maintenance of life are called as life processes. Understood? Now I am very clear about the introductory part. Now without making delay, let's get entered into this topic. That is the first topic called nutrition. Okay? Yes. So what is nutrition? If I ask you a question, what you are going to say? Ma'am, nothing ma'am. The food my mom is going to prepare now that I am going to take and I am going to have it, I will get energy. This is nothing but nutrition. Correct? So this is a simple, normal definition, what you are going to tell. But in a scientific way, in an accurate way, if you want to define nutrition, then we can define like this. What is that? The process by which an organism takes food and utilizes it is called nutrition. Organism has to take the food and it has to utilize. Why it has to take the food? Just now I told now, to get energy, to do activities. If you are cycling, if you are running, if you are reading, if you are writing, if you are watching my videos, you are sharing to your friends. So you are doing all this activity, right? I know you are doing that. For all these things, you need energy. So to get energy, first we need to put food inside. Correct? That is nothing but nutrition. Now within nutrition, how the organism is going to take the food? Because here, takes food is a point. Based on that, based on this mode of nutrition, we can classify the whole organism, which are so present in this world, into 2 categories. What are those? From here itself, your actual lesson start. Be serious, very simple, very interesting. Trust me, I will make you understand, don't worry. So first one is autotrophic nutrition, mode of nutrition. Second mode is heterotrophic nutrition. So what is autotrophic nutrition? Auto means what? A self. Auto means self. Okay. So here, trophic means organism they are going to prepare. Organism which prepare their own food. See, for example, a plant. So the best example we can take here, plants means particularly green plants. So green plants, they are going to do a magic. They say, we won't depend on anybody, man. We can prepare our own food. That's why we are called autotrophic. But there are certain organisms like animals. Human beings are also animals. We know that. and fungus, they are depend on plants. Then many students are telling, I know that, no ma'am, in our family, that means in all our family, my mom is going to prepare the food. We are also autotrophic. We are not depending on plants. So my mom is going to cook everything at home and we are going to have it. So we are preparing our own food. So then why you are considering us as a heterotrophic? My dears, your mom is preparing food. I know it. Even I also prepare the food. But how I will prepare? First, I will bring the things from the market. I am going to bring the dal. I am going to bring cereal, pulses, vegetables, everything. Then I am going to cook it. But I cannot prepare the vegetable of my own. Correct? I cannot prepare the cereal of my own. So, which is so produced in a plant, then only we can buy it from the market, right? So, the things which are going to produce, correct? That is nothing but autotroph. But we brought the things which are being prepared from the plants, we brought it from the market and we are going to cook it. That means we are depending on whom? We are depending on plants. Correct? So that's why we are considered as heterotroph. Not only we, not only us, even the animals, many animals like cow, tiger, everybody. Though the tiger, lions, all these organisms directly, they may not depend, but indirectly they are depending. That's why these autotrophs, green plants, we considered as producer because they are going to produce. And animals and fungus, directly or indirectly, they are depending. For example, cow, directly depending. But tiger, indirectly depending. First, it will eat cow. Correct? So, cow, in turn, depends on the plants. Correct? So, indirect dependent. But whatever, if you cannot prepare your food, no, you are heterotrophic. If you are preparing your own food, you are autotrophic. So, that is what the two mode of nutrition. Now, within autotrophic nutrition, first let us talk about autotrophic nutrition. How they are going to prepare that food. Let us see. Within that also, we can make a two category. So, this part is not given in your textbook, but still you have to learn this little bit information. If you had, that will be helpful. Photosynthetic autotroph, it is given in your textbook, but not about chemosynthetic. But no problem, easily you can understand. Photosynthetic autotroph means what? I will explain. So let me consider a plant, because in photosynthetic autotroph, the best example we can take green plant. So imagine here is a plant. So the plant is green. So plant says, I am very happy, man, because I can do magic. What magic? See, you just provide me the raw material. Just provide me raw material. What are the raw materials you want for food preparation? Very simple. Just provide me carbon dioxide. How we are going to get carbon dioxide? We means how plants are going to get carbon dioxide? That is in air. We are going to exhale, right? Animals are going to exhale. So this is nothing but waste gas for us. But plant says, don't worry, my friend. Whatever the waste that you are going to release, I am going to encash it. I am going to utilize, but I am going to give best for you. So that's why I am very kind. I am very great than you. So, okay, we should give a big salute to the plants because they are absorbing all the waste gas that we are going to release. So, this is a raw material for the food preparation because they are going to prepare their own food, right? So, the source is nothing but air. Now, the carbon dioxide is entering through air. How it is entering? There are small pores are present inside. We call them as stomata. We'll learn in detail, don't worry, about stomata. Through these pores, the carbon dioxide will enter. Now, one raw material is ready. So, like that, we need to provide water also. So, how we are going to provide water? It is present in the soil. So, when it rains, the water will seep down into the soil so that the roots of the plant will absorb. Okay, so when they absorb this water, that will be transported to leaf, stem, everywhere it will be transmitted. Correct? That means even in plant also transportation system is present. But water is a raw material that plants are going to utilize. Now the plants are saying, now look at me, I am going to do some magic. What is that magic? See, I need source, I need energy, because in order to cook food in your home, what you are going to do? First you are going to turn the gas light, right? So that flame has to burn so that you can cook your rice or whatever the ingredients you have to prepare. So here also, they also need some energy. So who is actually providing energy? Here is the sun. See, here is our sun So sun says, I am here, don't worry, I am going to provide energy. That means sunlight is also one of the raw material which is providing from the sun itself. And finally, who is going to capture? Who is going to capture? So that is nothing but the chlorophyll pigment, which is actually present in the leaf. Okay? Chlorophyll which is present within the leaf. That means all these things, this actually raw material is carbon dioxide and water. And this sunlight is the source of energy. And this chlorophyll is actually which is present in the leaf is going to absorb the things. Now by utilizing this energy, they are going to provide the food. Okay. So in the form of equation, if I have to show, then how I can show? By utilizing carbon dioxide as well as the water in the presence of sunlight and chlorophyll, the plants are going to prepare the food. So what is that food? I said glucose. Correct? The plants are also going to prepare this only. But this glucose, what is the symbolic form of this glucose? C6H12O6. So here, carbon, it is a carbon source. Glucose is a carbon source. So who is going to provide carbon? That is carbon dioxide. So carbon dioxide says, I am going to provide carbon. Take. I have not balanced the equation, but simply I am telling. And then oxygen is required. So carbon dioxide itself says, I am going to provide oxygen also. Take. So who is there? Who is going to provide hydrogen? Which is the hydrogen source? Here in water. Okay. So water says, I am going to provide hydrogen for you. Take. So C6H12O6, it is a balanced equation. Okay. In your textbook, the equation has been balanced, but simply I am telling how the source of food we are getting. So we got the food that is glucose. So this glucose, when it becomes joined like this, no? One glucose, another glucose, they are going to join with the help of a bond like this. I am one glucose, like that. One more glucose is standing in beside me. Another glucose standing beside me. We are all holding together each other's hand. That means we are making a big chain. So like such big chain, when it is formed between the glucose, we call it as a starch. So plants are going to store this food in the form of a starch. So starch is a stored food in plants. In the case of animals, this glucose only. But the chain of arrangement is quite different that you are going to learn in your higher classes, don't worry. But the stored food in animal is glycogen. So it is very important, these 2 points what I have written here, please make a note of all those things. Okay, so ultimately when they utilize carbon dioxide, water, here the water will first split. Water is going to split. First it will get separate, hydrogen as well as oxygen. So glucose says, I need only hydrogen. I do not require oxygen. Then where the oxygen will go? The oxygen will be removed out. It will be get escape from the plant. So that oxygen only we are going to utilize. So that's why we should say big thanks to plants. They are not only providing food to us, and along with that, they are also providing oxygen to us for respiration. Understood? So this is what the equation. And here, who is the source of energy? Sunlight. That's why we are going to give a name to this. What is that? Photosynthetic autotroph. Why photosynthetic? Because the source of energy is sunlight. Okay. Example is green plants and some cyanobacteria. And what is chemosynthetic autotroph? Here it is quite different. Chemo means chemical. They are going to get energy when the chemical reaction occurs. For example, purple sulfur bacteria. I will tell you how they are going to prepare. Imagine here is a purple sulfur bacteria. Let me draw it in a purple color. Okay. So this is our bacteria. So this bacteria is very upset. Why very upset? It is not getting energy. Okay, who is it? Purple sulfur bacteria. So by this time, one of his friend will come. For example, see, in our atmosphere, we know that nitrogen is present how much? Around 90, not 90. Nitrogen is present around 78%. We all know that. But do you think can we utilize that all nitrogen? No. Nitrogen is a very important part of protein. But still we cannot able to utilize, not even a plant can utilize. Why, man? Because the nitrogen is present in triple bond form. Between two nitrogen molecules, see, nitrogen, no? Between two nitrogen, the triple bond are there. Nobody is having guts to break the triple bond. Not even a plants, animals, human. Sorry. Nobody can break this. But one beautiful organism, that is microorganism only, bacteria, they have that special power. Okay. So, here is that bacteria. See, here is this bacteria here. This bacteria is very happy. So, this bacteria name is nitrifying bacteria. So, he is nitrifying bacteria. He is very happy. He says, I can do this process. I will break this nitrogen, that is triple bond present between the nitrogen, and I will convert it into nitrate. I will convert it into nitrite. Nitrite. I can convert like this. So when I convert this, no, I will put it into the soil. So when I put it into the soil, then obviously in the soil, the roots can absorb this nitrates or nitrites. So plant can easily absorb because nitrogen is very important for protein synthesis. Protein synthesis, okay. All this story we have understood. But why this purple sulfur bacteria is unhappy? Because it is not getting any energy to prepare the food. It says, look at my friend, I am having carbon dioxide. I am having hydrogen sulfide. Even my intention is also to prepare the food. C6, H12, O6. Carbon dioxide is providing. See here, O is provided by carbon dioxide. That is in O. And hydrogen sulfide is there. No, hydrogen is provided from this hydrogen sulfide. And finally, this sulfur gas will be excreted. That will be expelled out in the form of bubbles. Okay, this is the food. I can prepare this food. Just like plant. I am also great like plant. But how I can get energy? For the plant, sunlight was there. For me, who is there? By that time, this nitrifying bacteria will come to help. These nitrifying bacteria will say, my friend, don't worry. I am going to do certain chemical reaction. I am converting this nitrogen into nitrate, nitrite, everything. So during this process, some energy is going to produce. During this reaction, the energy is going to produce. You just grab that energy. You just utilize this energy for your process. You can carry this, man. Then this bacteria becomes so happy. Yes, anyhow, I got energy. But how it is getting energy? That is through chemical reaction. Correct? So that's why such organism, we call it as chemosynthetic. Why chemosynthetic? By using chemical energy or energy released from chemical reaction, they are going to synthesize the food. Synthetic means synthesizing food. Chemo means chemicals. Chemical energy, chemical reaction energy. So therefore, they are called chemosynthetic. Hope I am very clear. See, my suggestion is, whenever I am doing any topic on the board, please take a book and pen, write down in your copy, make a notes. It will definitely help you while reading your textbook, trust me. So though these things seems to be very funny and simple, oh, what is that ma'am is drawing some pictures and all, what is there? We can understand everything, but no, whatever the things that I'm going to draw on the board, either it is a small animal or a plant or anything, just write down as it is in the copy. It will definitely help you at the time of your exam. So need not to recall much. If you just open your textbook, open your notes, the handwritten notes that you are made by looking at the board, you can easily understand each and everything while reading your textbook. Okay? Now it seems to be easy, but after a few months, you will forget everything. Again, you need to watch the video. Again, you need to struggle for a time. So you can, You can actually save your time. Correct? You can save your time, you can encache your knowledge. Suppose if you want to watch it again, particular topic, you can use the timestamp solution what I gave, the suggestion what I gave in the beginning. Okay? That means 2 topics we have covered in autotrophic nutrition. One is photosynthetic, another one is chemosynthetic, example, purple sulfur bacteria. Okay? Now, Our textbook is giving more importance to autotrophic, that is plant. Let us go to that topic, how they are going to prepare the food. Yes, here is the equation of our textbook. 6 carbon dioxide if you provide along with the 12 molecules of H2O in the presence of chlorophyll and sunlight. So our plants are going to produce 1 glucose molecule. During this process, oxygen, when water is going to get split, no, the oxygen will be released and water is also going to form. So that's why when you just stand beneath the tree, you are going to feel a cooling effect. Why? During this process of photosynthesis, not only oxygen, even water is also going to be formed. So we are feeling cooling effect, we are going to feel shadow, everything. We feel somewhat coolness because of the water presence. Got it? Now let's enter into the process. How raw material are used for the plants for a photosynthesis. Let's understand. Okay, look at here. So this is our plant. Correct? Now let me take one leaf. I am just taking out this leaf. One leaf I am taking. So it's a big leaf. Got it? On the surface of the leaf, the small pores are present, I said. Those pores are called stomata. I'll do one thing. I will go to the next slide. So actually we are talking this, no? I will talk about this. First, let us explain the process. So one leaf we took, no? This is one leaf. A big leaf. So in this leaf, the small openings are present like this. So like this it will be present on the lower surface of the plants. That is on the leaf. We call it as stomata. Now, inside this leaf, many cells are there. Okay, let me take only one cell. This one big cell, what I am drawing, this cell we call it as a mesophyll cell. Many cells are there, but for time being, I am just making understand by taking the help of one cell. This is stomata. Now, within the cell, organelles are there. Mitochondria is present, chloroplast is present, Golgi apparatus is present. So many cell organelles are present that we all know. But here, for the photosynthesis purpose, chloroplast is the main organelle. Okay. So now, let me draw only one big chloroplast. Okay. Chloroplast is green color, right? So let me draw. This is one chloroplast. Many things are there. But I am just drawing only one chloroplast here. Many organelles are present. It's a double membranous structure. Okay, so let me mark it as a chloroplast. Now within chloroplast, this magic is happening. What is that magic? By absorbing sunlight, they are going to produce the food. Now, this one, who is it? This stomata says, I am going to give carbon dioxide to you, okay, which is present in the atmosphere. I am just going to give it, you take this. So this is what the carbon dioxide. So now this carbon dioxide has entered inside. And through xylem and phloem, water also entered, that is from the soil. This is water, correct? So you know, plants are going to absorb water. So water is also there. Now they have to convert it into food form, correct? But for that, energy is required, I said. So to absorb that energy, chlorophyll molecule must be there. Now within this chloroplast, there are certain coin-like structure present. Look at here, it's very beautiful. So these are coin-like structure present. We should call this name as thylakoid. So what is this name? Thylakoid. It's a coin-like structure. So like such, many coins are present, one below the other. It's like a stack of coin. So like this, there are so many coin-like structure which is present throughout this chloroplast. For example, look at here. Here I said this is our chloroplast, right? Mesophyll and chloroplast. Here many coin-like structure are present like this. This is one. This is another coin-like structure. This is another coin-like structure. So this structure, we call it as, one is thylakoid. So this group of thylakoid is called granum. Granum. So here they are going to be interconnected like this. Okay. So within this granum, the green color pigments are present. On the surface of the membrane, the pigments are present. Suppose if I just draw this as one granum, on the surface of this granum, the pigments are present like this. What are these pigments? Tell me. Yes, you are right. These are chlorophyll pigment. So this chlorophyll pigment, they are ready to capture the sunlight. Okay. So that is what I am going to draw there. So now I am not drawing all the thylakoids or all the granum. Only one granum I have taken. Okay. Yes. So here this entire thing we call it as a granum. Now on the surface, who is there? Chlorophyll pigment is there. Now here is our sun. So the sunlight is coming. Sunlight is coming. Now this chlorophyll pigment says, for this only I was waiting, just give me. Just give me. They are going to capture this sunlight. They are going to capture this. So that solar energy will be converted into a kind of chemical. Because directly solar energy will not go. Chlorophyll, they do a magic. They will convert it into a kind of chemical. So that chemical energy, so they will produce a chemical called ATP. They will produce a chemical called NADPH. So these are chemical energy. They are in the form of chemicals. Therefore, we call it as a chemical energy. How this chemical energy has formed? By this solar energy. Got it? Now, this chemical energy will be utilized to split this water. Understood what I was trying to tell? This ATP adding NADPH. So now by utilizing this energy will be utilized to produce C6H12O6. That is nothing but our food. So this energy will be finally utilized to split this water. Carbon dioxide is anyhow directly coming. Water, when it gets split, then only we are going to get oxygen. So again this oxygen, where it will be going? What do you think where it is going? This oxygen is again going through this stomata outside. That is what we are going to inhale. So this is what the overall process, my dear, very simple process happening in the plant. Understood? So what is there in this? Everything we have learned. Let us read the same thing what was given in our textbook that I have put in this slide. Okay. So hope you have written all these things in your copy. So don't skip anything. Please write down. Okay. Now, these are the points given in our textbook. Let's read this. What is the first point? Absorption of light energy by chlorophyll. So during the photosynthesis process, the chlorophyll is going to absorb the light energy. We have understood. The conversion of light energy to chemical energy. What is that chemical energy? ATP and NADPH. They have not mentioned. Our textbook has not mentioned this, but you should know this. So the detailed information of photosynthesis that you are going to learn, you have to learn in your class 11th. Okay. So in your class 11th, it is just a beginning. It is like a basics, just like a foundation for constructing a big building. So just think, if there is no proper foundation, how can you construct a building? Even you cannot construct a small hut also. There must be a proper foundation, correct? So here, these information, which is so going to be given in your class 10 textbook, is like a foundation. So when you have this knowledge now, then you can construct a big building like class 11th. Information you can get, class 12th, everything you can understand. So once you have a basic knowledge now, my dears, you can crack any sort of competitive exam in your future. If you are willing to become a doctor or some other, if you want to become join to become some professional courses, you have to write competitive exams like a NEET and all, CET, KCET, like that. Many state CET exams will be there, NEET exam is there, particularly to become a doctor or to go for a medical stream. So for such things, you should have a basic knowledge that is in class 10th. So the detailed information already I have done in class 11 syllabus. Here you can see this. Complete syllabus I have done of both class 11th and class 12th. You can watch in future. If you are a class 11 student, if you are watching this video to have a basic knowledge or foundation, just like a foundation course, so already you know that you can watch those things. Okay? Yes. So hope this diagram is clear. Now let's come here. Chemical energy, ATP and NADPH. Why they are using? They are used to split the water molecule. We are splitting this water molecule into what? Hydrogen. Water is H2O. So when you split this water molecule with the help of energy, chemical energy, obviously hydrogen will get separate and oxygen will get separate. Oxygen will be escaped out, hydrogen will be utilized. Why hydrogen will be utilized? For reduction of carbon dioxide. So this carbon dioxide, when you add hydrogen, it becomes CHO. That is reduction reaction into carbohydrate. That is what the process. Clear? Very much understood. Okay, that's it. Here you can see this diagram which is actually given in your textbook. Here they have taken the leaf. In this leaf, can you see the small section they have taken? And the upper part of the leaf. If you touch the leaf, no, if you just put a water drop, it won't absorb. It will be float. Why? Because there is a waxy layer. That waxy layer we call it as a cuticle. This is waxy layer. Below that, the epidermal cells will be there. This is upper epidermis, lower epidermis. And in between these epidermis, the cells are there. These are nothing but mesophyll cells. These only, mesophyll cells. Only one cell I have drawn bigger, right? That is what this cell. And within this cell, the green structure, actually I am showing with red color. Okay. These green structures are called chloroplast. Within that chloroplast, chlorophyll is present. I have shown it how it will looks. And between the cells, some air spaces will be present so that it has to breathe. That's why. And the guard cells are present. These are nothing but the stomata. So now let us talk in detail about the stomata. How this stomata, because a small pore, tiny pore I have shown by showing some 2 kidney shaped cells. Now if I just enlarge this stomata, how it looks, let's see that. Very simple structure stomata. See, it is having 2 bean shaped structure like this. One bean shaped structure. This is another bean shaped structure. So these bean shaped structures are called guard cells. These are called guard cells. So in the and this inside, can you see this now? Inside there is an opening. This opening is nothing but stomatal pore. When water enter inside this, imagine this is water. When water enter inside this, it automatically become bulges. It will be like this. It get bulges. When it get bulges, can you see a hole in between? So like that, the pore will be created. That is stomatal pore, through which carbon dioxide will get inside or oxygen will move outside. Like that it will happen. But how this guard cell will get the water? That is from surrounding cell. So these surrounding cell, what you are seeing here, like this, it will be present around this guard cell. So these are called subsidiary cell. So the first water will enter into this subsidiary cell, then it will enter into the guard cell. When you pour water to the plant, no, it will be passed to different part of the plant body and enter into water, guard subsidiary cell, then into guard cell, the stomata become open. Okay. The plants are also very smart. So when they think that this is the time to do photosynthesis, then they will bulge. Then the stomatal pore will open. But if there is no proper water, there is no proper sunlight, they think that there is no water, much water is there in the plant, automatically they will close. If there is no need of photosynthesis, why they have to open? They will close. So when they get closed, no, it means the water is not here. It will be coming out. That means they will get shrink. So when they get shrink, automatically we are going to see the structure like this. Can you see here? The stomatal pore has got closed. This will be surrounding subsidiary cell. This is shrinking of stomata. This is opening. This is opening of stomata. Shrinking means nothing but closing of stomata. Shrinking or closing. Understood? But what the process, photosynthesis process I said, no, they are not occur one after the other. They will be occur simultaneously. They will be stored carbon dioxide. Whenever it required, they will just do it. For example, desert plant. Imagine the plant which is growing in a desert. It's very harsh sunlight, scorchy sunlight. So if they just keep open the stoma, that is stomatal pore, it will just evaporate the water, right? Because the stomatal function is not only for exchange of gases. Even the water vapor is also get exchanged. Because I told no, some quantity of water is also formed during the photosynthesis reaction. That water will also escape out. So if keep evaporating the water, the plant will become shrink. So that's why the desert plant will do a beautiful mechanism. During daytime, they will just close the stomata. They will just hold the water through their roots, and in the night, they open the stomata. They start doing the work, and even if the transpiration occurs, it is not that much. Okay, so evaporation of water is nothing but transpiration process. And another one is exchange of gases. So these are the two functions of stomata. So hope stomatal structure is very clear. Look at this. This is your textbook diagram. This is opening and this is closing structure. So if the question comes, opening and closing you have understood. If the question comes, the function of stomata, what you are going to write? One is for transpiration. Transpiration means what? Evaporation of water. Another one is exchange of gases. Okay, so exchange of gases during respiration, the plants also breathe like us. They will also utilize oxygen, they give carbon dioxide. But for photosynthesis, they will take carbon dioxide but give out oxygen. So you are going to study everything in class 11th in detail about all those things. Already have done, I told. But for this year, this much is enough. Okay. Hope the autotrophic nutrition is very clear for you. Now 2 activities are given in our NCERT. Let's read. So these 2 activities are explaining why chlorophyll is important. Because photosynthesis to occur, chlorophyll is important. And photosynthesis to occur, carbon dioxide is important. So because of these 2 only, the food will be prepared. How the plants are going to store the food? That will be in the form of a starch. Okay. So if you test the presence of starch, we can see that plant has prepared the food. To prove that, they have shown 2 activities. First, let me explain that activity, then I will read this. Okay. So in the first activity, they have talking about chlorophyll is important. Very simple experiment. What they have taken, take some variegated plant. Variegated plant means different color. For example, money plant you have seen, no? Patches of green color will be there. So like that you take this money plant. Imagine this is a money plant. Just take it. Here the patches of green color you can see on this plant. Now keep this plant in a dark room. This is the first thing they are telling. Just keep this plant in a dark room. Why they are telling? Let in the dark room no photosynthesis occur. So whatever the food the plant has prepared already, let it get utilized. So when completely it has utilized, no? Now keep this apparatus. Because no photosynthesis, no food is there right now. Now when you keep this plant in a sunlight, same plant, almost around 6 hours, photosynthesis must have taken, correct? So now what you have to do, take one leaf. So imagine this is one target leaf, you just pluck it, take it and keep it in a boiled water. Okay, just keep this leaf. And before keeping, they are telling, first trace this leaf in a butter paper. I mean, wherever the green color patches that you are seeing, imagine this is a butter paper. On this butter paper, just mark wherever the green color you have seen. Here is the green color. Okay, you have just marked it by plucking this leaf. Okay, now keep this leaf, just cut one of the leaf and keep it in a boiling water. Okay, and after that, take out the leaf. Take out the leaf. Take a beaker. In this beaker, just take some alcohol. Now keep the same leaf. Okay. Now you take this apparatus and put it into this boiling water. Now, let me... Okay, or else let me draw here. This is boiling water. In this boiling water, just keep this alcoholic content where you have kept the leaf. Now you just boil it. Okay. Now the green color which was so there, it will completely get vanished so that the alcohol color itself will turn into green. Now you take out the leaf. Now you take out the leaf. You just test the presence of starch with iodine. Test this. Iodine is to test the presence of starch. When you test this leaf, only in the patches you could be able to see the blue color. Okay, let me show the blue color here. In this patch, you could be able to see blue color. Blue color is nothing but it is the indication that starch is here. So where the starch is here, wherever that you have marked the green color, no, before doing all this activity, the green color part is nothing but food prepared part or starch produced part. So how you can say that in this area starch has been produced? When you test it with iodine, you can see the blue color patches. That is what this experiment is. So from this, it is very clear that the chlorophyll pigment is very essential for photosynthesis. Let us read the same thing from our textbook, what they have mentioned. Take a potted plant with variegated leaves, money plant. Keep the plant in a dark room for 3 days. Okay. And all the starch gets used up. Now keep the plant in sunlight about 6 hours. Pluck a leaf from plant and mark green areas and trace them on a sheet paper. Okay. Dip the leaf in boiling water for a few minutes. After this, immerse the beaker containing alcohol. Carefully place the above beaker in a water bath. That means that alcoholic beaker. Keep it in the water and alcohol begins to boil. When it begins to boil, what happens to the color of the leaf? It will completely get vanished because the leaf color will enter into the solution and the entire solution become green. Now dip the leaf in a dilute solution of iodine for a few minutes. Directly you can put the iodine solution or you can dip there. Take out the leaf and rinse the iodine solution. Observe the color of the leaf, compare with the tracing of leaf done with the beginning. That means when you put iodine, there everywhere the blue color patches you can see, and before that the green color patches wherever was there only the blue color patches could be seen. This is what the experiment is, very simple experiment. Once again, this experiment is basics for your class 11th. If you take a science as your part in your future, it's a basic, all those experiments are there. But there they say that anyhow you have learned in your previous classes, in your lower classes, here you are going to learn. Next activity says that here carbon dioxide is important for photosynthesis. The same thing. Here also what they'll do, take 2 plant. Okay, same type of plant. And now here in one plant, near one plant, take a dish, petri dish, and put some KOH solution, potassium hydroxide solution. Now you close this beaker, plant with a beaker, bell jar. Just close this, both the beaker. So how we have to close means no air should enter. That's why just put some Vaseline jelly so that air should not enter inside. So it is a potted plant. Now, in beaker 1, we have kept potassium hydroxide. So this potassium hydroxide is going to absorb carbon dioxide. Carbon dioxide which is so present in this plant. And here carbon dioxide is already there. Okay. Now you take out, just keep this apparatus for a few hours so that let all the carbon dioxide of this plant should get utilized. So now take one leaf from here. So this is one leaf I have taken from beaker 1. And this is another leaf I have taken from beaker 2. Now you test the presence of iodine. I mean, with iodine, if you test for the presence of starch, this also, this leaf will turn into blue color. This one is not. Why? When blue color turning occur, it indicates that starch was there in this leaf. How the starch was there? The carbon dioxide was there. That's why starch has been prepared. Photosynthesis has occurred. But here, you have kept the apparatus in sunlight, everything was there, but photosynthesis didn't occur. Why? The carbon dioxide has been utilized by the potassium hydroxide. Therefore, it is not showing blue color. This is what the experiment. Let's read this. Take 2 healthy potted plants which nearly of same size. Keep them in dark room for 3 days. Okay, I didn't say dark room. First, keep them in dark room. Now, place each plant on separate glass plate. Place watch glass containing potassium hydroxide, KOH, by side of one plant. Potassium hydroxide used to absorb the carbon dioxide. It is important. Cover both the plants with separate bell jar. Use Vaseline to seal from the bottom so that to make the setup airtight. Keep the plants in the sunlight for 2 hours because the photosynthesis has to occur. Why earlier they have kept in dark room? Because let all the starch which are so present in the plant to get utilized completely. That's why. Keep the plants in sunlight for 2 hours, okay? Pluck the leaf from each plant and check for the presence of starch. So do both the leaves show the presence of same amount of starch? What can you conclude from this activity? One leaf shows the presence of starch, another one is not. It means carbon dioxide must be needed for photosynthesis. This is what the experiment is. Simple, done. So, okay. The plant case nutrition part is over. Why the photosynthesis has to occur? What is the significance? It is an extra information. One is the solar energy is available to different organisms, but we cannot utilize. Only plants can utilize. From that plant we are getting through food chain. And the second thing is the plants are the producers. Directly or indirectly, all organisms are depending on plants. So therefore, if they do not produce anything, what we should eat? And the third thing is oxygen and carbon dioxide, which is so maintaining in the air only because of plants. So we should say big thanks to plants. So this is what the significance. Now let's move on to next part within the nutrition. What is that? Heterotrophic nutrition. Okay, now let's talk about heterotrophic nutrition. So as I have explained earlier itself, heterotrophic means the organisms which depend on other organisms for their food. That means they cannot prepare their own food. For example, human beings, we cannot prepare our own food. We need, of course, ingredients. By taking that ingredients, we could able to prepare the food, but we cannot manufacture our own like plants. Correct? So that's why we come under the category of heterotrophic. Though we are depending on the plants, though other organisms depending on other sources for their food, but all comes under heterotrophic only, but based on their mode on which they are depending, whether they are depending directly on the plants, whether they are depending on dead organisms, whether they are depending on living organisms. So based on that, we could able to classify this heterotrophic nutrition into 3 categories. Okay, what are those? One is Holozoic nutrition. That means the human beings or animals, even the herbivorous, carnivorous, these are all nothing but animals only. These organisms comes under this category of Holozoic nutrition. I will explain one by one by taking example, don't worry. And the second mode of nutrition is saprophytic. Saprophytic means they depend on dead and decaying organic matter. For example, yeast, fungus, like a mushroom and all. So they depend on dead and decaying organic matter. Then you may ask the question, ma'am, you said just now animals, human beings, that is carnivorous, omnivorous, all comes under Holozoic nutrition. Then a lion also depend on dead organism only, it also eat dead animal. Do you think that animal like a lion, tiger also saprophytic? No. Saprophytic means it is totally different. That of Holozoic. I will give examples and I will explain that, okay, while taking example itself. Then the third mode of nutrition is parasitic, okay. So now let us discuss one by one so that all your doubts will get clear, right? Yes. The first one is Holozoic nutrition. So in Holozoic nutrition, what the statement is given here? The organism ingests solid organic matter, and digestion happens inside the body. Let me take example of a human being itself so that you can understand very clearly. See, of course, I'm a human being, no doubt in that, correct? So what I'm going to do, if I get hungry or if I feel to have anything, so I will eat as such. Suppose if I have some pani puri, if I have some pizza, burger, anything, okay? Suppose if I want to have, I will eat as it is, correct? So in that pizza or a burger or any food molecule, what I have chosen to eat, it may have carbohydrates, it will be having protein, it will be having lipids, correct? Carbohydrate means ragi, wheat, jowar, all this comes under carbohydrate, their energy source. Protein, for example, your nuts, as well as the pulses, dal, all those things comes under the protein category. And your ghee, butter, everything comes under the lipid category. But you take any food, these three ingredients will be present. Maybe their concentration will be high or less, but the ingredient will be there. So what I will do, I'm just going to have as it is. That is the first step. And when it enters, then it is going to get breakdown inside my body. Okay, so that is what they are telling here. The organism is going to ingest the solid organic matter, and the digestion means breaking process, happens inside the body. So in Holozoic nutrition, mainly which carry 5 steps. What are those? Example, herbivorous, carnivorous, omnivorous. You know the difference. What are herbivorous? Herbivorous are the organisms that directly depend on plants. Correct? For example, cow, buffalo, sheep. So all these organisms directly eat plants. So they are herbivorous. Carnivorous, for example, lion, tiger, cheetah. So many examples we can take. They eat these cow, deer, etc. But indirectly they depend on plants only. Though they're directly not eating the plant, but by eating cow, they are satisfying their hunger, but still the cow is to depend, it has to depend on the plants. Correct? That's why we say, oh, you are also depending on plants, man, but indirectly. So they comes under the category of carnivorous, that is flesh-eating animals. Then what are omnivorous? Omnivorous are nothing but human beings, comes under this category. Pigs comes under this category. Dogs comes under this category. So here what happens? Human being, we eat directly the plant source. That is vegetable, fruits, everything which is a plant source, we are going to eat it directly. Like that, we are going to eat even animal source also. For example, egg, milk, everything, even the meat. So we are going to eat both the plant source as well as the animal source. You may say, ma'am, I am purely vegetarian, ma'am. I won't eat meat, I won't eat any chicken. But my dear, I am talking in general about the human beings. So human beings are the group, they can feed on both plant source as well as the animal source. Like that even pig, like that even dog. So they comes under the category of omnivorous. So in all these organisms, the food will be taken directly, and after ingesting, after taking inside, the breaking process occur. So let us discuss about these steps in detail, so that if you have the knowledge of these steps, then in our upcoming topic, that is how the nutrition or the digestion process occur in human being, you can understand very clear. So that portion will become so easy for you once you understand these steps. Okay. What is the first step here? They are telling ingestion. So what is ingestion? Look at here. Yes, let me draw here itself. Imagine he is a person. So now, what he is having? Let's imagine he is having some pizza or a burger, anything. Any food molecule you can take. Now he is having it. Okay. So now let's consider the food, what he is having inside. It is having carbohydrate. So the food not only having carbohydrate, it is even having the protein. It is having component like lipid. Lipid are nothing but fats. Okay. So all these components are present in the food, what this fellow is having now. Even we also, whatever the things that we are going to eat, it will be having these sources in it. Correct? Now, I will means, of course, we are going to consider these molecules or these components of food as a polymer. Now it's a new word for you. Polymer. Polymer means what? Imagine I have a big wall. This is a wall. So how this wall has been constructed? Of course, with the help of bricks. Everybody do agree for this, correct? Now imagine I have started placing the bricks one after the other, one above the other, like this. So if I just place a bricks like this, do you think the wall has been constructed? No. We need to bind it. We need to gum it, right? So how we are going to do? With the help of cement as well as the sand. So like that you are going to blast it, right? Then only we can say, okay, big wall has been constructed. Now, if you consider this wall as a polymer and the bricks, what I have placed one above the other as a monomer. And this pink color structure, what I said, cement, sand, everything I have mixed in order to bind these bricks are considered as a bond. Then I could able to get a polymer like a wall, correct? So like that, even in our components of the food like carbohydrates, protein, lipids, fat, they are made up of monomer. For example, if I take a carbohydrate, either it is a ragi or a wheat or a jowar, anything you can take, it will be made up of a monomer called glucose, fructose, okay, galactose. So these are the monomers, simple sugar. Let me take glucose as an example. See, one glucose is there. Imagine this one glucose is a monomer. Like that, so many glucose are present together and they are going to be get connected with the help of this bond. So name of the bond you are going to learn in your higher classes. Don't worry. In your class 11th, there is a chapter called biomolecules. I have done video of that chapter because full syllabus I have completed. You can see here, this is a biomolecules chapter of class 11 syllabus. If you take a science, then you are going to study biomolecules chapter in detail. So to understand that chapter, it is a basic. Okay. Okay, let's come back. So now carbohydrate is a what? It's a polymer. Now in order to break this bond, we need a special chemical substance. Those special chemical substance are nothing but enzymes. So imagine he is an enzyme. He is an enzyme. Let's assume he has a scissor in it. He is holding a scissor to cut this bond. Now the question is, why he has to cut the bond, ma'am? Why he has to cut the bond? If he eat carbohydrate as it is, let it be like this. Why he has to cut all these bonds so that he has to make free these glucose? This is because the food molecule what we are going to eat, that is the first step. This is the first step. It is ingestion. And after, so where it will go? It will go into our alimentary canal, that is esophagus. We are going to discuss shortly, don't worry. It will enter into our stomach, then it will enter into our intestine, then large intestine, and ultimately undigested food will be ejected out through anus. But whatever, when the food molecule enter inside, there itself it is going to get breakdown. That is the second step called digestion. So digestion is nothing but with the help of this enzyme, it has to get break. Now imagine this scissor, that is enzyme, is going to cut this bond so that all these molecules, that is glucose, is now become free. Because no bond is there in between. Now they are simple molecule. It was a complex, now it becomes simple. When it gets simple, then only our blood, imagine this is our blood vessel, inside blood is there. It could able to absorb these simple molecule. Unless and until, if it is not get broken, the blood will not accept it. If blood do not accept this or do not absorb it, what is the use of having food? Correct. So this is the third step. So what is the third step? The third step is nothing but absorption. This is absorption by blood. Okay. So like that, it happens for every food component. If you take protein, proteins are made up of what? Amino acid, one amino acid, 2 amino acid, 3 amino acid. Like that, many amino acids are going to be get connected by a bond. Now it is a big chain of amino acid. I call it as a protein. So again, I need a special enzyme, not this one. So if I call this enzyme, he will say, my duty is only to break the carbohydrate, not your protein. So I need to call a special enzyme. Imagine he is a special enzyme. So he is also holding a scissor in order to break the bond between amino acid. So when it got break into small amino acid, no, simple amino acid, again it will be get absorbed by the blood. Again it will go back and absorbed by the blood. Okay, so like that, if I come to the lipid case or a fat case, fat says, I am also very complex man. See, glycerol plus 3 fatty acids are there. Glycerol plus 3 fatty acids. This is one molecule. Like that, if I take another molecule, again the bond is there. Again to this bond, glycerol plus 3 fatty acids are attaching. Okay, like that, there are so many molecules of glycerol fatty acid are connected together. Again, I need to call a special enzyme. Again, he has to hold in order to break this with the help of a scissor. Scissor means the enzymes are chemicals, they could able to break this. Now, when they break, then only it will absorb. How they are going to break? First, they will remove one fatty acid, another fatty acid, then glycerol. Then all these molecules get absorbed like fatty acid, glycerol, like that to the blood. Okay. So, after breaking, that is after digestion, if it has to get absorbed into the blood means it requires the enzyme function. Now the question is, why it has to get absorbed into the blood? Okay. Let it get absorbed into the blood. What is the purpose? What is the use? It is very simple. See, that you will understand in fourth step. What happened in fourth step? Imagine, here our cell is waiting. This is our cell. So this cell is energyless. Now it is not having any energy. So to provide energy to this cell, I need to give glucose to this. So after utilizing glucose by combining with oxygen, just now we have studied, it got energy like ATP. Now it says, okay, I have energy. I got energy. I am so powerful. Suppose if this cell, if it is got damaged, then to repair this damaged part also, it required amino acid. So we need to supply amino acid to this. That means whatever the molecules of food that has been absorbed into the blood should be supplied to the cell for making energy, for to repair, for the growth, for all this purpose. That is the fourth step called assimilation. So hope you are making notes of all this point. And after utilizing everything, there are so many waste product will be released. So that waste product will be accumulated here into the rectum part, that is in the large intestine, and finally that will be ejected out as a fecal matter. That is the fifth step called egestion. So these are the 5 steps that must be followed in Holozoic nutrition. Either it is a human being or a lion, a tiger, sheep, cow, buffalo, dog, pig, anybody. Okay, so such type of nutrition is Holozoic nutrition. Hope you understood this. So that's why we say whenever you have a food, it has to get digested. Then only your body will absorb it. How? This is the method. So hope you have written all these things. Now let me read the definitions or a statement what we have given for these steps. Okay. Please don't neglect anything. I said these are like a basics for you. Right. Let's go back. So here the 5 steps definitions have mentioned. Let us read it. So in the first step, what is happening? The process of taking in food. That is ingestion. And after ingestion, the digestion is happening. That is breakdown of food into simple substance. That is glucose, amino acid. These are simple substance. To get absorbed into the bloodstream, it has to get absorbed, right? And in the third step, absorption of digested food. Okay, digested food is going to get absorbed. And in the fourth step, assimilation, utilization of digested food for growth and repair. So if the cell has to repair it, the cell has to utilize it for growth process, and if it has to utilize for energy production, for all this purpose it can utilize. And finally, in the last step, the removal of undigested food from the body. Okay, our textbook has given 2 examples. One is about amoeba, another one is about Paramoecium, to explain how Holozoic nutrition is taking place in such a small organism also. Okay, let us understand that process because that is important for your board exam. But these 5 steps are clear, I think. Okay. Yes. Look at here. Holozoic nutrition in amoeba. This is amoeba, right? So amoeba, everybody knows it is not having a special particular shape. Sometimes the amoeba may look like this. And in another time, the amoeba may look like this. It is totally different. And in some other time, the amoeba may look like this. So it will look in different shape. Why it is so? Because this amoeba have the capacity to push its cytoplasm. Cytoplasm means here the liquid portion present inside the cell. It will be pushed towards the source of the food. So here imagine, let me rub out these things. So let's assume here is the food. Then what amoeba will do? It will push the cytoplasm towards the food so that it will come like this. It will block the food so that it create a vacuole inside itself by blocking it. So that's why when you just look at this amoeba, now already its shape has got changed. So this is the first step. Here, can you see the blue color part is food? It has pushed its cytoplasm so that it is coming like an arm, coming like a feet. Hence, such structures are called pseudopodia. Pseudo means false. Poda means feet, false feet. It could able to change its structure, not every time in the same position. Suppose if the food is present here, it could able to change its position in this direction. Correct? Pseudopodia will be found here, not only in this particular place. And after that, when it got ingested, if it has created like a food vacuole, the enzymes will be presented there also. It will start breaking the food. That is called digestion. And after digestion, of course, it has to be absorbed into the body. That is absorption. And after absorption, it will be utilized. So amoeba is also utilizing for making new food vacuoles, new molecules inside its body only. That's why it is called assimilation. And after assimilation, of course, the waste product which is so produced in amoeba body also will be get removed outside. That is called digestion. So even though it is a very simple one cell structure or organism, it is following all 5 steps of nutrition. Then obviously it is a holozoic nutrition. Correct? So this is not our textbook diagram. This is the image that I have put to make you understand. Here, this is your textbook diagram. The same diagram, but they have not mentioned the steps. That's why. Okay? But you have to be very clear about your identification, because sometimes if the question comes based on diagram based, you should be able to mark it. Okay? Look at here, the food particles, pseudopodia, food vacuole inside. Okay? The food particle has been taken inside. Then assimilation, digestion, everything has been happening here. Okay? Now, another example, let us talk by taking Paramoecium. In Paramoecium also, the process of ingestion, digestion, assimilation, absorption, everything is taking place. But by a particular part, it is not changing its shape like amoeba. Okay? Let me show. Imagine, this is our Paramoecium. It is something like the sole of a slipper. Correct? It will be like this. Now on the surface of this structure, the organism is having so many hair-like structures like this. So these hairs are called what? Do you know? So these hairs are called cilia. Now let's assume, imagine the food particle is here. Okay, and this portion, here is a groove, no? This is called oral groove. Oral groove. So below this only, there is a special food vacuole present. So now what happened? Here is the food. This is the food. Now, this food cannot be entered into the body of the Paramoecium inside directly because there is no passage. Suppose if you place a food here in my hand, how can I get inside my body? Without bringing near to my mouth, correct? So like that, here also, this is a oral groove. That means it is like a mouth for this Paramoecium. Of course, inside, cytoplasm will be there, nucleus will be there. There are so many contractile vacuole will be there. Let us not focus about all those things. But imagine this is the mouth. We need to bring the food near the mouth. So how it is possible? It is with the help of cilia. So cilia is going to push the food like this. Imagine my hands are like a cilia. I'm going to just push it near my mouth. So when it comes near to the mouth, after being pushed, so it will be get injected. That is the ingestion process. And when it get entered inside the food vacuole, enzyme will be act on it. It will be get digested. Then it will be get absorbed into the body. Absorption. Then it will be utilized for so many purposes, for making some other structure, assimilation, and finally waste food also, unwanted egestion will occur. So this is what about nutrition in Paramoecium. Again, the same holozoic. Okay? Yes. So holozoic nutrition we have understood. We need to study the nutrition in man also by talking in detail about all the structure. But before going to that, let us talk 2 more nutrition, that is saprophytic as well as parasitic. Okay? Yes. Now the second category of Heterotrophic nutrition is Saprophytic nutrition. Saprophytic nutrition means what? It feeds on dead and decaying organic matter. Okay. So I said, while explaining this, while talking about saprophytic nutrition, all the organisms which are directly eating dead organisms, they are not saprophytic. Lion is not a saprophytic. Tiger is not a saprophytic. Though they are killing and eating those dead organisms only. Why? Because, see, here is one major difference. Here, the digestion of the food occurs externally before the nutrients are absorbed. See, what the lion is going to do or what the tiger is going to do, even though it kill a deer, so that deer meat will be ingested as it is, correct? Then only inside the body of the lion or a tiger, the digestion occur as I have told with the help of enzyme. But here in the case, like a fungus, okay, look at here. This is bread mold. Its name is bread mold. And this is a mushroom. So mushroom can occur in any shape. You have seen the mushroom in like this. Like this mushroom you have seen. White color. Correct? This is also mushroom. These are also mushroom. They are called bracket fungus. Okay? This is bread mold and yeast. Everybody heard about this. It will be used in preparation of bread as well as the cake. So in all these organisms, what happened? First, they are going to release the enzymes outside. Imagine I am a bread mold. Okay, my name is Rhizopus. Its name is Rhizopus. It's a bread mold. Now I saw a bread. Okay, so what I will do? I won't eat it directly. If I eat the bread as such, I am going to follow Holozoic nutrition. But I am not doing. I am a saprophyte. Okay, so what I am doing? Bread is there. Okay, bread. Just wait. I'll do one thing. I am going to spit something on you. Spit means here it is going to release the enzyme outside. The enzymes are actually inside. Now they are going to spit outside. All the enzymes are there outside. Okay. Now these enzymes, they are going to break the component of the food into simple molecule outside only. I am just waiting. Okay. You just do your work, enzyme. I am just waiting. Then I will watch. Okay. All the food molecule become completely simple. That means the complex molecule becomes simple. Then I am going to ingest. Ingest means I am going to absorb it. Imagine I am going to have a glucon D. So glucon D or a glucose, it is in a simple form. If I just take it as such, I am going to get instant energy. Correct? So like that, it is going to break everything outside. Then it is going to absorb it. So such type of nutrition are nothing but saprophytic nutrition. It is only depending on dead and decaying organic matter. If dead bodies are there, then it is going to do like that. If any dead vegetables are there, there they are going to do such process. If any fruits are there, they are going to do on this. So that's why if you just touch a rotten fruit or a vegetable, we are going to see something watery on the surface. We could easily see that yes, it has been deteriorated, it has been spoiled. How? Because the smell started coming out, because digestion is occurring outside. And it start becoming softening. Why? Digestion is happening there. Correct? That is called saprophytic. And the third category of nutrition is nothing but parasitic nutrition. Here parasitic are totally different. Here they are going to obtain the nutrition from living organisms. Okay, I will make you understand by giving example only. See, within parasite there are two types, ectoparasite and endoparasite. First of all, parasite means they are totally depending on others. Of course for food, but if another organism living, from that living organism, it is going to suck the nutrition, it is going to get the shelter, everything. So that's why we call it as a parasite. For example, see, look at here. This is a leech, correct? So leech, what it is going to do? It is present on human body. Not every time. If the person has been attacked by leech, the blood will be sucked by it. Leech is going to suck the blood. So here it is present on the surface. That's why we call it as a ecto. Ecto means outside the body. Like that, this is the lice. In a hair, lice will be there. In animal body, if you have any pets in your home, you can see in their fur, we are going to see ticks. Okay. So these lice and ticks are present on the surface of the body. They are going to suck the blood. That is nothing but nourishment only. So when they are present outside the body, we call them as what? Ectoparasite. Parasite which attach to external body on the surface of the host. Leech, ticks, lice. Correct. So why it is a parasite? Because the person is living. When he is living, then only it is taking the food from him. Correct. So like that, within the parasite, we have another category called endoparasite. In endoparasite, what happens? These parasites, for example, roundworm or a tapeworm. This is tapeworm example. This is roundworm. What they are going to do? They will enter inside the body. You might have seen, especially to the children, the parents are going to warn, don't eat too much of sweets, don't eat only chocolates. So your body start getting worm. Okay, your stomach will be filled by worm. Why it is so? This is because in order to alert them, they are telling. In fact, it is not only enter into our body by sweets or chocolates. When you eat food without washing your hands properly, the eggs will be present in your fingers. Okay, the eggs will be present in your nails. Knowingly or unknowingly, it will be entered through the soil or through any other material. So when you eat the food without washing your hands, or when you eat the food without washing them properly, the eggs will enter into our body. There they are going to get hatch and multiply. So when they enter inside, in the intestine particularly, what they will do? The food which is so going to be get digested in your small intestine, they start absorbing it. Okay, suppose it is a tapeworm or a roundworm. My hand is an intestine. So all the food material will comes here in the form of a blood. Okay, so it is directly going to absorb it. That's it. So it is getting nourishment. You are not getting it. So as it is present inside your body, we call it as endoparasite. Okay, look at here. Parasite which attach to internal body surface of the host. Host means all living organism on which these organisms are depending. If in my body, if roundworm is there, roundworm is not there in my body. Just for example, I am telling. If in my body, if the roundworm is there, I am a host. Roundworm is a parasite. Okay, understood? Example, roundworm and tapeworm. So that's it. Here, these are example of parasite, animal case parasite. So here, even for plant also, we can see the parasitic plant. See, the best example is Cuscuta. It is given in your textbook. It is very important. It is a parasitic plant. Why parasitic? Can you see here wire-like structure, yellow color, greenish. So these wire are nothing but Cuscuta. It is not actually wire. They are plants. These plants, they do not have chlorophyll. That's why we say they lack chlorophyll. So without chlorophyll, how they can prepare the food? That's why it is depending on other green plants. It is growing above the green plant so that it will suck the nutrition from the green plant. Suppose if it is a green plant, through photosynthesis, if it is prepare all the food, then this Cuscuta, it is going to grow on it so that it could able to send a kind of root inside and start absorbing those nutrition which have been prepared by green plants. That's why they are called parasitic plant. Okay, clear. So hope I am very clear about three types of nutrition: Holozoic, Saprophytic as well as the parasitic. Now let us enter into the nutrition or a digestion process, how it is going to take place in a human being. It's very important. But let us understand in detail, in a simple way, without making any complication. Okay? Yes. Nutrition in human beings. Whenever we talk about digestive system of a man, it can be divided into two parts. One is alimentary canal. Alimentary canal means what? It is a tubular structure which begins from the mouth and up to the anus. Look at here. So imagine he is a person. Okay. So the food has entered into his mouth. Right? The food has entered into his mouth. So now a pipe will be continued like this. It will become a pharynx. It will become a esophagus. Then the pipe will get bulged like this. It will become a stomach. Then the pipe show many foldings like this. It will become an intestine. Then it will twist like this only. It will become a large intestine. And finally it will be ejected out through anus. So whatever the food that you have eaten, that will enter into this pipe, that will be retained in this pipe only, though in a different part. It may enter into the stomach, it may enter into the intestine, large intestine. After digestion completed, undigested food will be ejected out through anus. That is from mouth to anus, a complete tubular structure, what you are saying, that we call it as an alimentary canal. Okay, so it is a tubular structure, food remains here. But only this much is not enough for our digestion. What I said, After ingestion, the digestion has to occur with the help of enzymes, I said. That means the enzymes are the chemicals, they have hold the scissors like to break that bond. So, those enzymes will be present in a special gland. So, those glands are salivary gland, pancreas and liver. So, they are going to hold the enzymes. Okay, pancreas is, I have a lot of enzymes. If you want, I am going to pour it into your alimentary canal. Okay, it will be present side by side. Look at here. So, it is your pancreas. Here your pancreas. So, pancreas is holding that enzyme. So, you want to get your food to get digest. Okay, I am going to pour all the secretion inside. So, it start pouring the secretion. Like that. Here, the liver is present. It is having some juice. It is going to pour the things inside. Are you getting my point? So, even the saliva is also having the enzymes. It is going to pour its juice. So, all these glands are present aside, nearby the alimentary canal. Whenever it wants, it is just going to pour according to the necessity. Okay. So, such glands we call it as accessory glands. They are side actors. This is the main hero of the movie. They are the side actors, assisting actors, so that they are helping for the digestion process. Okay. Now, let me draw a complete alimentary canal so that you can understand all the structures clearly, and then we'll talk about all this process, how it is going to happen in the mouth, that is digestion, how the digestion is going to happen in alimentary canal like stomach, intestine, everything we'll talk. Okay. Yes. Let me draw a structure of alimentary canal. Let me take a separate slide. It's a nose of a person. It's a mouth. Okay. So, this is the nostril. Through this nostril, what happened? He is going to inhale the air. So, when he inhale the air, it will enter into the nasal cavity. So, this is the mouth. He is going to eat the food through his mouth. When he eat, it will enter into his buccal cavity. This is buccal cavity, this is nasal cavity. But of course, there is a separation. There is a partition in order to prevent the food to be being entered into the nasal cavity. Okay. But one thing we have to remember, when air enters here or the food enters here, it will pass down through a common passage. There is a common passage. There is a common passage for both food as well as for the air. So, this common passage is called what? Pharynx. Okay, let me mark it. Okay, so it's a common passage for food and air. Now, let's assume the food has entered inside. The food has entered inside. But now this pharynx is showing two roads. That means bifurcation. Something like this. Here is one structure. Here is another structure. Now food is in a confusion. Where I should move? If I move here into this place, the person will get collapse. He will get die because this is the windpipe. What is this? This is the windpipe. So in the windpipe only air should enter. So we should not enter here. Therefore, there is a gap. There is a closing structure that we'll talk in respiration. Don't worry. So what is the destiny of this food molecule? Of course, it has to enter into this structure. So this pipe is nothing but food pipe. So what is the other name for food pipe? Yes, esophagus. Now when the food enter into the esophagus, it will move down. It will move down, and here there is a muscular bag. J-shaped muscular bag is present, made up of muscle. Are you getting my point? This is nothing but the stomach. Now the food is in the stomach. So it starts showing the digestion inside the stomach only. Now from the stomach, this tube will be continued by showing many foldings. Okay, something like this. It started showing the foldings. It started showing foldings, many foldings. And finally, it is going to be enter into another structure. This folding, what you are seeing now here, all this structure, this is nothing but small intestine. So the small intestine itself has three parts: ileum, jejunum, duodenum. Okay, so I will mention here duodenum. Duodenum, ileum and jejunum. The majority of the digestion occurs in duodenum. If you want, you can write in your copy, because right now it is not much important, this structure, but in your upcoming classes it will be important. Okay. Now the small intestine, it is going to connect to another tube. So what is the tube? Something like this. Look at this. The tube is like this. At the end it shows a worm-like structure. So this is our large intestine. Got this? So this is large intestine. So finally, the undigested food that will be collected in this place is called rectum, and this opening is nothing but anus. Through this, the undigested food will be ejected out. So this is what the complete structure of digestive system. And here, whatever the food the person is going to eat, that will be present within this tube only. To assist the digestion of this food, the accessory gland which are so present, that will be present here and there. For example, inside the salivary gland is present. And here, on the right side, if the stomach is present on the left side, on the right side, the liver is present. Imagine this is the liver. So here the liver is present. So like that, here the structure called pancreas is present. But this pancreas and the liver and the salivary gland are accessory gland. They are not actually tubular structure. They are not alimentary canal. They are assisting the food which is present inside the alimentary canal. Understood? Now, let us take one by one, that is each and every part. We'll discuss how the food is going to get digested. If I say food, of course, you must be knowing that food is having carbohydrate, protein, lipids, everything. So, when that food enters, when ingestion occurs, first step, is the digestion occur within the mouth? Yes, it do occur. If it is occurring, how it is happening? Let us study like that. When it enters into the esophagus, I said no digestion. Why it is not? Let us see. When the food enters into the stomach, I said digestion occur. If it is occurring, what are the food component is going to get digested? Like that, we'll talk about each and every part of alimentary canal, and we'll discuss what are all the food are digesting here. Okay, very simple, easy, don't worry. So hope you have written this also in your copy. Let me move into the next part. Okay, first thing is mouth. So when you just open your mouth, what are the structure you are going to see? Of course, teeth. The first thing is teeth. I'm not drawing every teeth completely, but teeth will be there. Correct? What is the purpose of having teeth? Of course, that is helpful to talk, but along with that, when it comes to food or a digestion process, we are going to chew the food with the help of teeth only. Okay, we are going to crush it. We are going to make a paste. So that is what the purpose. The teeth helps in chewing the food. And after that, in our mouth, what is that? Tongue is there. This tongue is going to roll up the food. Because of the tongue only, we can sense the taste, we could able to talk. But when it comes to the digestion, it helps to rolls up the food. Okay. So the muscular organ rolls up the food. Why muscular organ? It is made up of muscle, that's why. And after that, within the mouth, we have a salivary gland. In fact, we have three types of salivary gland. One salivary gland is present near the cheek. Another salivary gland is present below the tongue. If you say is a person, if he is the tongue of this person, below this one salivary gland is there. Here one salivary gland is there. Below this lower jaw salivary gland is there. So they are going to pour the saliva into the mouth through this duct system. Okay, I have a diagram here. See, this is one parotid gland. It is a salivary gland. So here there is a channel through this duct. It is going to release the secretion and pour into the mouth. Like that, here also below the tongue, sublingual. Lingual means tongue. Another gland present below this mandible. So all these are having a duct system. Through this duct system, they are going to release the saliva into the mouth. Now the question is, why saliva is important? Saliva is nothing but it is a watery fluid. Correct. Why it has to present? Of course, it make our mouth to be in a moist condition. But when it comes to the food digestion purpose, what is the use? Look at here. The saliva consists water. Because of this only, the food is going to become softened. Okay, it softens the food. And it is also having electrolytes. Potassium, sodium, chlorine, bicarbonate, all these electrolytes are present. But main purpose of saliva, having saliva in our mouth is, it is having two enzymes. One is lysozyme, another one is salivary amylase. Lysozyme is an antibacterial. It is just watching like a soldier. If any enemy come, that is in the form of bacteria, it is going to kill within the mouth itself. That's why it is say it's an antibacterial. But for a digestion purpose, there is one important enzyme is there. It is very important. We call it as salivary amylase. What this enzyme will do? So this enzyme is going to act on the carbohydrate. We know we have a carbohydrate in the food when we eat. So this carbohydrate, as we all know, it is made up of many molecules of glucose like this, monomer. Then only it become a polymer. Correct? So imagine this salivary amylase is an enzyme. It is holding a scissor. Okay, it is going to have a scissor to cut this bond. So the bond which is so present, it has to cut. But now the salivary amylase is, I am very small. I can't cut all the bond which are present between them. But I can cut in between. That also, how much? If you eat 100% carbohydrate, I can cut only 30%. Remaining 70%, I am not going to touch. I will just act on 30%. In that 30% also, I am not going to break it completely. I will make like this. 2,2-glucose. I am just going to leave like this. Between 2-glucose, I am going to cut. Between 2-glucose, I am going to cut. Like that, I am going to leave it like 2,2-glucose molecule. So if you ask your blood vessel, can you absorb this? Blood vessel says, no. First, you have to break this bond also. Make it complete simple, then I will absorb. That means, if it is present in a 2-sugar molecule form, we call it as a disaccharide. So, there is a name for this disaccharide called maltose. If you do like this, I can't absorb. First, let it get breakdown further. Okay, but at least 30% it has made into simpler way, no? So that's why we should say thanks to this salivary amylase. Okay. So if any question comes, from where the digestion process begins, you have to answer that it is begin from mouth itself with the help of salivary amylase. Now, if you ask the salivary amylase, can you break protein like carbohydrate? It says no. Can you break lipid like carbohydrate? It says no. That means salivary amylase acts only on what? Carbohydrate. So the mouth part is over. Now where the food is entering? After the mouth, it has to enter into pharynx. No digestion there. It has to enter into the esophagus. Okay? Done. This one done. Yes. Now the food is in esophagus. So imagine from the mouth, we know that what happened in the mouth. The food has entered into this esophagus. So when it entered into the esophagus, again there is no digestion. Here no digestion occurs because there is no enzymes. No enzymes. It is just like a pipe, just for pushing the food. Okay. So it will just move. It will just push the food in a wavy motion. For example, look at here. Imagine I have a pipe. I have a rubber tube. So in this rubber tube, we are forcing a marble. Okay. So let's consider the marble is right now here. So by the time the rubber, this part of the rubber seems to be bulge. Correct? And after that, when the rubber comes here, this part become normal. Again, this portion will seems to be bulging. Wherever this rubber comes, that portion will be seems to be bulging, right? It means something like this. Something like this we are going to see. Somewhat wavy like motion. So this type of movement, wherever it goes, the food moves, it starts showing the movement. It shows the movement That movement we call it as peristaltic movement. So this peristaltic movement will occur throughout our alimentary canal. When the food comes here in the esophagus, the peristaltic movement occurs. It shows like this wavy movement. When the food enters into the stomach, again the stomach is going to show the churning movement, again peristaltic. When it is entered into the small intestine, again the food has to push down because here also peristaltic movement. Like that, into the large intestine, the food has to move up. So like that, throughout the alimentary canal, the pushing action of the food will be occur. Why it is so? Because our entire alimentary canal is made up of muscles. Alimentary canal made up of muscles. Inside, the muscle layer is there. Therefore, it could able to push the food. But one thing we have to remember, in esophagus, no digestion occurs. Now, where the food has to enter? Yes, you are right. Now, food is entering here into this stomach. Okay, let's see what happens in the stomach. Now, food is in the stomach. So, it is a J-shaped muscular organ, I said. So, when the food enters here, the muscle that will be show inward movement. Some of the muscle which are so present in the esophagus, between esophagus and the stomach, which show inverted movement. So, what it will do? It says, food, if you want to move down, okay, I will allow. You can move here. But the same food, if it want to move up, I won't allow. So, that do happen, the upward movement of food only during vomiting. So, by that time, the special signal will be sent from our brain towards these muscles. So, you have to open, the food has to come out. Okay, he wants to puke. Then only it will open. Unless and until, if no signal comes, no upward movement, only downward movement. Okay, that thing should be clear. Now, in the stomach, you might have observed that there is an acid present. We call it as HCl, correct? How it is going to happen, I will tell you. In the wall of the stomach, there are certain foldings present like this. So, these foldings, we call it as rugae. What are rugae? Rugae are foldings of the stomach. So, when you enlarge any one part, so how it looks? Look at it. It looks something like this. So, inside these foldings, there are many glands present. So, these glands are known as gastric glands. These are all basics. You should know about all those things. It's very important. Inside this gastric gland, 3 types of cells are present. This is one category of cell. This is another category of cells. Imagine this is another category. 3 category of cells are there. So, one category of cells is one type of cell. Okay, or else I will write like this. So, one type of cell says, I am going to produce mucus. Mucus means what? It is somewhat a gum-like substance. And another category of cell says, I am going to produce an acid called HCl. And the third category of cell says, I am going to produce an enzyme. So, the main purpose this enzyme is to digest the food, correct? But unfortunate thing is, this enzyme is always in an inactive state. Okay, so this inactive enzyme is known as, he is an inactive enzyme. So, he is always sleepy. He is always in a sleepy mode. His name is pepsinogen. He is always inactive. But what is his main work? He has to break the protein. But he is sleepy mode. So, who has to wake him up? By that time, we'll call this HCl. Hey, you HCl, just go and wake him up. Then it will go and it will just touch this pepsinogen. Hey, pepsinogen, just wake up. Look at here. Who is coming? Then it will open his eyes. Then we say, okay, you are right now become active. It becomes active pepsin. Though this active pepsin, it acts on protein. Now it digests the protein. Okay, how it is going to digest the protein? How it is going to digest the protein? As we all know, proteins are made up of amino acid like this. This is one amino acid. This is another amino acid. Correct? So like that, many amino acids are present. They are connected with the help of bond. Now this pepsin will come. It start breaking this bond between them. But once again, just like salivary amylase, it won't break complete protein chain. What it will say is, I am just going to make a small chain like this. If you are a 100 amino acid, then I am going to make 25 pieces. Okay? It means the small pieces of protein it will convert. So this piece we call it as peptone and peptide. It's a small protein chain. But still, our blood is unable to absorb. Do you think our blood is going to absorb? No. So blood will put condition already. If you want me to get absorbed, you have to become a simple substance. So here also in the stomach, only incomplete digestion occurs. Okay. But now the question is, what is the function of this mucus? This HCl acid is very acidic. So because of that, HCl inside the stomach, the pH becomes so acidic that it is 1.8. In such 1.8 pH, no enzymes will act. Only pepsinogen will show the daring to act on the food because it could be able to tolerate such acidic pH. But rest of the enzymes do not work on this pH. So that's why in the stomach, no carbohydrate digestion occur, no lipid digestion occur. Nothing will occur except this protein. That is also very little bit. Okay. And if I just allow this HCl to get spread throughout the stomach, it will damage the wall also, right? So that's why the mucus will be produced. So this mucus will protect the wall of the stomach. So when you eat some spicy food or some spicy related food or something like that, if it is heavy food, the HCl will become more and more. Okay. So then only the person says that I had this kind of food, I started feeling so acidity, gastritis. So why it is so? Because the HCl production will become so high. Sometimes what happens, this HCl start moving up also. That's why the person is going to feel some burning sensation in the esophagus. This is because of excess acidity. That we call it as acid reflux. It is going to be move up. Understood? So this is what the thing happens in the stomach. Now, where the food is moving right now? The food has to move into the intestine. Here in the stomach, the food can retain up to 4 to 5 hours. Hope you are writing everything. Food remains here. But one question has been asked in your textbook, what is other function of HCl? So they will ask, do you know? You say, we don't know, you just leave it. Suppose such question comes in your exam, what you will do? That's why you should know the function. So hope you have written all this point. Let me move on to the next slide. What are all the function of HCl? The first function is to activate pepsinogen into active pepsin. Already have told. And along with that, it is also considered as antibacterial. Just like salivary amylase, how it is going to kill the bacteria when it enters through food, like that even HCl is going to burn this bacteria. Suppose if the bacteria get escaped from the salivary amylase action, then it will directly come into the stomach. There it will get encountered by this HCl. Okay, that is another function. The third function is the food. What you are going to have, that it is iron rich food. It is very important to our body. That will be in the form of Fe3+. So if it is in Fe3+, our body do not absorb it. So it is near waste, simply it will be removed out through fecal matter. So that's why HCl, what it will do, it will convert this into absorbable form called Fe2+. So when it gets converted, then only our blood is going to absorb and suitable nourishment will be comes into our body. Okay. So these are all the three main functions you should know about HCl. Now, where the food is moving, it is moving into intestine. But here, again, we are going to see what? The fate of different food. What is happening? Okay. Let me talk about the sphincters and liver, pancreas. Wait, sorry students. Let me tell what are all the food components that we had initially and what is the fate of food right now after being entering into the stomach. Okay. So first, let us take the carbohydrate. So carbohydrate 100% you had. In that 100% what happened? Only 30% of the carbohydrate has converted into disaccharide form like this. Correct? So this is nothing but maltose. Who did this? With the help of salivary amylase. Salivary amylase had done. Earlier we had complete like this. complete glucose, but in that it has converted into disaccharide form. That also only 30%. But remaining 70%, it is as it is. No digestion has occurred. It remains as it is like a big chain. Correct. Now, this will also get digested and this disaccharide also get digested so that the blood can absorb it properly. Now, if you take the example of a protein, so this one happened where? It happened in the mouth. So this thing happened in the mouth. Now, when you take an example of a protein, so protein was a chain of amino acid. Now this big amino acid, of course, entire 100% has converted into what? It has been converted into small pieces like this. These are amino acid. I'm not mentioning it, that's it. But this small chain of protein are nothing but peptides and peptones. Correct? So where this thing has happened? It happened in the stomach. That means still the protein has to get digested, still the carbohydrate has to get digested. But when you look at the lipid, so lipid, what I said, it is a nothing but glycerol plus fatty acid. 3 fatty acids are attached to glycerol. So this is one molecule. Like that, there is another molecule. Like that, there is another molecule. So it remains 100% as it is. It is not yet get digested. So this is what the fate of our food right now after being entering into the stomach. Correct? So do you think our digestion process over? No. It has to enter where? Into the small intestine. So for the breakdown of all these components, complete breakdown of all these components into simple molecule, we need help of so many enzymes. Those enzymes are present in where? They are present in the pancreatic juice. They are present within the intestine itself. We call it as intestinal juice. Okay? And some of the action will be done with the help of bile juice, which is present in the liver. Let's see what happened there with the help of these enzymes. Okay? So this is also clear. Now, when you take the example, again, the food has entered into the esophagus. There is a muscular inward ingrowth that we call it as a sphincter. After being escaped from this, the food has entered into the stomach. Correct? Now from the stomach, it has entered into the small intestine. This is small intestine, duodenum part. Here also, there is an obstacle. These obstacles, we call it as a sphincter. So this is also sphincter. Sphincter means nothing but muscular outgrowth, that's it. This is also sphincter. Now food, when being entered into the stomach, the sphincter have allowed. Now the food, it becomes a semi-solid right now. Now it is passing through the sphincter into the small intestine. Okay. But the food which is entered here, this food is acidic. Why? Because within the stomach, the HCl is maintaining acidic pH, that is 1.8. Therefore, the food being get mixed with this HCl, it is also become acidic. So now the first thing is we need to neutralize this acidic pH. Why? Because no other enzymes will act on this acidic pH. Correct? So that's why we are taking the help of liver. So where the liver is present? Here it is. I told. Here is the liver. Or else I will draw here so that you can understand it. Imagine this is the small intestine. I will mention its part in this region. This is nothing but duodenum. So to bring this acidic pH into normal level, the liver is helping. Here is the liver. So liver says, don't worry my friend, I am always going to secrete a juice. That juice name is bile juice. But for time being, I am going to secrete here into this gallbladder. Every time I am going to produce it. But if there is no food in the stomach, why should I simply store it in my liver? I am going to store it in the gallbladder. Whenever you require, I will just send it. So now the time is coming. You have to release. So we are asking this liver. Liver says, okay, no problem, I will send. Now through this duct, the bile juice is entering into this small intestine. So here bile juice is coming. Now, one thing you have to remember, in the bile juice, no enzymes are present. No enzymes again. Then, how you say, ma'am, digestion occur with the help of bile juice? That is what I am telling. No digestion occur. Here, with the help of bile juice, emulsification occur. Emulsification means what? Imagine it's a big rock. It's a big rock. Now, if you take a hammer, if you just keep hitting on this big rock, what happens? You are going to get a small pieces, right? I won't say it is a soil. It is just a small piece. A big rock become a small piece, right? Like that, if it is a big fat molecule, it becomes small micelles. Small micelles or fat globules. That is the best word. So only this process can happen with the help of bile juice. So this process itself, we call it as what? Emulsification. Emulsification. And bile juice is. So many other enzymes are coming now. Okay, I have more water. I have some basic pH. So now I can neutralize. So this bile juice even helps in neutralizing acidic pH. So this is the two function of this bile juice related to digestion. So now bile juice is entering here. But still, for the digestion purpose, we want enzymes. Correct? So how the enzymes are producing? So here in this side itself, we have a pancreas. So pancreas says, I have enzymes. I will release. Don't worry. See, I have juice here. So I will send to juice. So in this juice, I am going to send enzymes. So here so many enzymes are there. See, all these are enzymes. They are very, very happy. So who are these? They are nothing but enzymes. Now all these enzymes are coming in order to break the food into the small intestine. And along with that, one more funny thing is, within the small intestine itself, some juice is producing. Okay? Small intestine itself, juice is going to produce, that we call it as intestinal juice. So in this intestinal juice also, we have enzymes. So finally, 3 juices are entering into this duodenum part. What are those? One is bile juice, which is coming from the liver. Another one is pancreatic juice, coming from pancreas. This is pancreatic juice, coming from pancreas. And another one is intestinal juice, coming from small intestine. But except bile juice, all these 2 juices are having enzymes to break these food molecules. What you have seen before? These food molecules, that is carbohydrate, small protein chain as well as the lipid, can be get broken completely with the help of these 2 juices. Okay. Now when these 2 juices along with bile juice enter into the small intestine, that is in the duodenum, what happened? Let's see. Okay. When these 2 juices, pancreatic juice as well as the intestinal juice, act on the carbohydrate earlier was like this. Correct? So this was around 70%. Now it has become completely free like this. Complete free glucose. Complete free glucose. And earlier 30% was like this disaccharide. Now it will also get broken down completely into small glucose. That means complete digestion of glucose occur. There are so many enzymes are there. I can list amylase, fructase, sucrase, maltase. So just now, time being, no need. But you just write down. Or else, if you want, I can write this side, no problem. The enzyme's name I will write here. Here, related to this, we have enzymes called amylase, maltase, fructase, sucrase. So like that, these are the enzymes. They completely break this carbohydrate. Okay, now it is free. Now when you come to the protein is something like this, no? Small chain. Now one enzyme comes. His name is trypsin. So trypsin says, he is a trypsin. He is having scissor again. All these enzymes are having scissor. Okay, I'm going to cut everybody. So all these amino acids become free. So these are all amino acid. This is one amino acid. This is another amino acid. Like that, all amino acids become free. Trypsin, chymotrypsin, so many enzymes are there. Now, when you come to lipid is nothing but how it is. Right now, lipid is like this, no? Glycerol plus fatty acid. Fatty acid, fatty acid. Now they act with the help of an enzyme called lipase. So first, what this lipase will do? First, it remove one fatty acid. Then it becomes glycerol plus 2 fatty acid, because one fatty acid has become removed. Now again lipid act on it. I mean lipase act on it. Lipase is also enzyme. This lipase is also enzyme. It is holding scissor to break. Now again it break. Glycerol plus 1 fatty acid. Again another fatty acid has become removed. Again lipid will get further breakdown into glycerol and separate fatty acid. Now everybody is free. Everybody is free. Now only thing is blood should get absorbed it. So blood should get absorbed fatty acid. Blood should get absorbed glycerol. Blood should get absorbed amino acids. So blood should get absorbed this glucose. So now everything will be get absorbed by the blood. But where this blood vessel is present, where absorption occur, that process occur in small intestine itself. So one thing you have to remember, in the small intestine, complete digestion as well as absorption process occur. Okay. Yes. So hope this part is very clear. Very, very clear. Now when you come to the small intestine, look at how the small intestine, actually how I have shown it. I showed something like this, no? It's like a tubular structure like this. When you look at this tubular structure, if I just enlarge this part, look at me carefully. If I just enlarge any one part, I am going to see something like this. Suppose if it is my small intestine, inside if you just imagine small needles I have placed, how it looks, like that only. Can you see? This is a round alimentary canal, that is small intestine. Here small tubular structure. This is enlarged portion. These are called villi. These are called villi. On the surface of the villi, there are small cells, again with a small projection. We call it as microvilli. Microvilli. And inside this villi, we are going to see the blood vessel. I'm just going to take only one portion of the villi and microvilli. I will tell you how absorption occurs. Absorption of food in small intestine. So let me consider this is villi. Okay. What I said, on the surface of this villi, we are going to see small structure again. These are cells only. So like this, it will be present throughout the surface of the villi. Now let's imagine here. Here is a blood vessel. Inside blood will be there. Blood will be there. Now imagine we have a glucose. So the glucose will be absorbed into the cell, that will be dumped into the blood. If you have amino acid, because it has been converted into small portion, no, that will be absorbed by this and again it will be entered here. So why it is showing many finger-like projection? To increase the absorption area. To increase the absorption area. So this small intestine length also varies from organism to organism. Okay. So like this, the absorption of complete food, whatever it has been broken into simple molecule, will be get absorbed into this blood. That we call it as the absorption. So if you take the small intestine length, in herbivores, it will be very lengthy. Why? Because cellulose is there. For the breaking process of cellulose, more enzymes, more bacteria required. Therefore, the length of herbivores is very long. More length or more lengthy. Where? In herbivores. Whereas carnivores, that is meat eating, meat get digested very easily. Therefore, it is very small portion. Got it? So this is what the intestine of herbivores and carnivores. Again, it is important. This is smaller. This is larger. Okay. So now We have understood what happens in small intestine. Now when it comes to large intestine, so this is large intestine. And here to this portion, the small intestine comes and connects. So after the complete digestion, complete absorption, the process of these two things has done, the rest of the material which have not been absorbed by the blood will be sent into this part, that is large intestine. Okay. So in the large intestine, what happened? Large intestine also very smart. It won't allow all the things to be get ejected out. So what it will do? In the food, the water droplets or water molecules, if it is present, that will be absorbed in the large intestine. So whenever the function asked about the large intestine, you should know here absorption of water occurs. Okay. So finally, the food, that is undigested food, that will be pushed up like this. So this entire region, what you are saying, we call it as a colon. This is colon. So the undigested food will be moved up into the colon, that will becomes here, and finally it will be collected in this region. This is called rectum. Rectum. Okay. So when it get collected in the rectum, then only the person is going to feel heavy to go for a washroom, that is to remove the feces or stools. So finally it will be ejected out through anus. So this process of ejection is nothing but ejection process. That is the last step of the digestive system. Okay. So here, if you observe carefully, here there is a worm-like structure is present, a finger-like structure. This is nothing but appendix. Okay. So this is what the overall function of the entire alimentary canal or digestive system. Hope I am very clear about this part. So heterotrophic nutrition also over. So the complete portion of nutrition, the topic itself has been completed. Okay. Now at the end, so hope you have written this part also in your copybook. Don't neglect anything. Now here in our textbook, a small thing has given so that some information has given as no more. Let us understand what they are given about dental caries. So dental is nothing but it refers to the tooth. Okay. So caries means decay. So here they are talking about the tooth decay. So before reading this, let us understand the structure of our tooth. Okay. Look at this diagram. When you look at this tooth, it is mainly having three structures. In fact, so many other structures are also there, but the main structure I have shown here. The white portion, what you are going to see when you just open your mouth and the teeth. So this part is nothing but enamel. It is the hardest part. It is even harder than the bones. Okay, it is a non-living part. And the inside, this brown color part, what you are seeing, it is a dentine. And below that, we can see the pulp. There is a pink color region. So here what happens? So whenever we talk about the tooth decay, so here the softening, because I said it is a very hard part. Enamel is a very hard part. But unfortunately, due to the action of bacteria, so this enamel start getting decay. That is nothing but tooth decay. It start getting softened. Okay. How it is going to happen? See, that is what they are telling here. Dental caries or tooth decay causes gradual softening of enamel and dentine. So here, the bacteria, when the bacteria sits, of course, whenever we are going to have the food, if you won't goggle your mouth properly, or if you won't brush your teeth properly, of course, the food particles that will be remain here only. Okay. So to feed on these food particles, imagine this is a bacteria. It start attacking here. So it start feeding on those food particles. Okay. So during this time, during this action, it is going to release the acid. Okay. So that acid start corroding, that acid start decaying or softening this part. Okay. So gradually it will be spread even to this dentine part also. So that's why whenever this part when touches to the nerves, which are so present inside the tooth, the person start feeling the pain. That's why we are going towards the doctor by having a toothache. Okay. So here, look at this. When bacteria acting on the sugar is nothing but the carbohydrates or the food particles which are so sweetened. That is nothing but sugar produce acid, that softens or demineralizes. Why demineralizes? Because it is also made up of calcium. So that will be start breaking down. That's why it is called demineralizes the enamel. Masses of bacterial cells together with food particles stick to the tooth to form dental plaque. Dental plaque means, look at this diagram. So here, can you see? So it start making a plaque, because it's not just by one bacteria. Group of bacteria will start attacking on this. Okay, they start releasing more and more acid. Obviously, it will corrode the upper layer part. That is nothing but plaque. The saliva cannot reach to the tooth surface because it will get blocked by these bacteria. The saliva cannot reach, and even saliva cannot even neutralize this acid, because we know that within saliva, the pH will be 6.8. Saliva pH is 6.8. So, when such pH enters near the acid which have been produced by bacteria, it could be able to neutralize. But unfortunately, saliva do not enter. It won't be get absorbed by this part. Okay. And plaque covers the teeth. So, plaque start covering the teeth. Saliva cannot enter there. Therefore, what the precaution we need to take? We need to brush our teeth properly soon after eating. Okay. So, that's why doctor advised to brush twice in a day. Early morning as well as even before going to the bed, in order to prevent this tooth decay problem. Okay. So, brushing the teeth after eating removes the plaque. That means the deposits which are so happen, that will be get removed before the bacteria produce the acid. If the plaque are there, then only bacteria attacks on it. So, if there is no plaque, if it has been removed, then bacteria do not attack and no acid production occur, no tooth decay. So, if untreated, microorganism may invade the pulp. First, it will enter the dentine part, it will decay, then it will enter the pulp and causing inflammation and infection. So, this is what the information they have given. Hope this part also very clear. Now, let's move on to the next topic called respiration. Okay, now let's talk about respiration. So, many students, they exactly don't know the difference between breathing as well as respiration. Correct? So, what they think, when you inhale oxygen, let me take example of human being only. When you inhale oxygen, right, and exhale carbon dioxide, that means we are giving out carbon dioxide. So, this is what they think, it's a respiration process. But actually it is not. What I did just now, inhalation as well as the exhalation, that is nothing but breathing. Then what is respiration, ma'am? In respiration also, we thought that oxygen will enter and carbon dioxide will exit. This is what happens. Yes, my dear, this is what happens even in respiration. But this respiration is a chemical process. And what I said just now, inhalation and exhalation, that is breathing, it is a physical process. So let me tell the difference between breathing and respiration, then you will understand clearly. Okay, look at here. Imagine he is a guy and it is the nostril of this person. Correct? So what he is doing? Now he is taking in oxygen. Correct? Now this oxygen will enter into its nasal cavity, then different parts of its respiratory system that I am going to talk later. Don't worry, but you just know about this. Now it will enter into its respiratory system and finally enter into the lungs. Correct? So now in the lungs, the oxygen is there. And in the lungs within, the carbon dioxide which is so produced in our body, of course everybody knows that carbon dioxide is going to be produced in our body, that will comes once again here itself. Okay, imagine the carbon dioxide is here. Now this carbon dioxide is again through this respiratory system comes outside. So here the exchange of gas is taking place. That is, oxygen is going inside and carbon dioxide is coming outside. Here also oxygen and carbon dioxide are playing, but the interaction is occurring between whom? It is with atmosphere as well as with the lungs. So between the atmosphere and the lungs, this interaction is happening. Correct? So that's why we say that it is a physical process, because here no chemical reaction is taking place. No breaking down of any component is happening. So therefore I can say this breathing is a physical process. Okay. Now let me come to the respiration part. So now let me talk about respiration. So what is respiration then? So imagine, okay, so this is a human being. Okay, so his body is made up of cells. Every cells need energy. That we just now understood by studying nutrition chapter. Correct. Now let me take any one cell. So imagine one cell I have taken. Okay. Now the cell is very upset. Why? It need energy. So for that energy, on which system we are depending? We were depending on digestive system. Correct. Suppose if I draw digestive system here, this is the stomach, intestine, everything we have understood. So here the glucose was there. The glucose was there. So this glucose has entered into the blood vessel. So now it is given to the cell. But still cell is not happy. Why it is not happy? Because to break this glucose, it requires what? It requires oxygen. So that's why the cell says, you have just given the glucose, but to get energy, I need oxygen, just do provide me. Then the blood asks to the lungs, just give me that oxygen. You have taken here, no, this oxygen, just give me. Then what you are going to do? Now with these lungs, what we are going to breathe in oxygen, so that oxygen we are going to take inside the blood vessel. So now this oxygen, once again, that will be delivered to this cell only. Correct? So now cell is very happy. Why it is happy? Because it has glucose, and to break this glucose, it got oxygen. Now with the help of these two components, it is going to get energy. So that energy, this energy will be in the form of ATP. ATP is nothing but it is a chemical component. It is a compound. It is a chemical compound. Okay. So in our body, energy will be in the form of a chemical. Okay. So during this process, it is going to produce even a waste gas called carbon dioxide. So this carbon dioxide cell do not want. Cell says, I do not want this carbon dioxide. It is a toxic to me. It is a waste to me. It is a poisonous to me. Just take it out. Then blood vessel says, okay, no problem. Just give me. Then this carbon dioxide again go to the blood vessel. And from the blood vessel again go to the lungs. And from the lungs again that will be comes out. Okay. So now, if you just leave this part and if you just focus only on this area, here the process which is happening inside the cell, here some chemical reaction is taking place. Correct? So that's why we say respiration is a chemical process. Here breaking down of glucose is taking place to get energy. Correct? So shall I write? So respiration is a chemical process. It's a chemical process. So now it's very clear we got the difference between breathing as well as the respiration. Now, when I just focus only on the respiration, here I have written an equation, right? A chemical reaction I have written. So I have told ATP. So let me tell you what exactly this ATP means. Okay. So hope you have written this part in your copybook. Let me move on to the next slide. So what is ATP? So before going ATP, let me write the equation of this glucose breaking process once again. So I have just written like this: glucose. When this glucose combined with oxygen, it got energy. And during this energy, it also released carbon dioxide. Only this much I have written, right? But actually, if I have to mention the same process in the form of equation, then how can I mention? The glucose is nothing but it is a 6-carbon compound. Okay, that is nothing but C6H12O6. It is the formula of glucose. Now this glucose, when combined with 6 molecule of oxygen, to break one glucose, how many oxygen molecule we require? 6 molecule of oxygen we require. Then in our body, 6 carbon dioxide molecules are going to produce. Along with that, even water molecules are also going to be formed. 6 water plus energy. So this energy is always in the form of a chemical. So that chemical name itself we call it as what? ATP. ATP. Now the question is, we have understood this equation, right? We have understood this equation. Now our main focus should be on this ATP. What is this ATP, ma'am? Very simple. ATP full form is, I will write here, adenosine triphosphate. Adenosine triphosphate. I will tell you what exactly happened, how ATP is going to form, in which way ATP is going to form, I will tell you. So hope this equation is clear about the breaking down of glucose. Look at here. Energy currency. ATP is called as an energy currency, just like how we are going to use currency for our expenditure. Correct? Suppose if I want to go to a shop, if I want to purchase something, I have to use currency, right? Right? So by using that currency, I can purchase anything, I can do shopping, I can go anywhere, because it is a power, it is an energy. Like that, my body in the sense, human body or animal body or organism body, if they have to use, if they have to do some work, so how they are going to do this? They should need energy, they need power, correct? So that energy is in the form of this ATP. I will tell you exactly how this ATP is going to form. Just focus here for 2 seconds, you will understand. Imagine this is our cell. Okay, this is our cell. Or else, I'll do one thing. Let me draw a big cell. Okay, imagine a big cell is there. So in this big cell, a chemical called adenine is roaming. This is adenine. Imagine, he is the guy, he is the adenine. So this fellow name is adenine. So this adenine is always roaming by holding 2 phosphate molecule along with him. Okay. So let me draw like this. This is one phosphate. This is another phosphate. So who is he? He is actually adenine. He is roaming in the cytoplasm by holding these 2 phosphate. Now what is the name I can give him? Not ADP. ADP means, sorry, not ATP. ATP means adenine triphosphate. But here this fellow adenine is holding 2 phosphate, right? Then I can call him as ADP. ADP full form is what? Adenosine. Adenine, adenosine both are same. Adenosine diphosphate. Now, what is the difference between adenosine and adenine that you are going to learn in your class 12th? So, when you take science, there is a particular branch called genetics. There you are going to learn in detail about all those things. But it is just a basic. You just know about this much. That is enough. Okay. Now, this ADP is simply roaming. Now, another fellow is here. Another fellow is here. So his name is inorganic phosphate. Who is he? His name is inorganic phosphate. So inorganic phosphate in a simple sentence or in a simple manner, I can write IP. IP means inorganic phosphate. Now somebody will come, and with the help of that somebody, what happened? These two will be combined by utilizing some energy. Look at here. Utilizing some heat energy or something. These two people were combined together. Now what happened? What happened? He is one phosphate, but he is an inorganic phosphate. Why inorganic phosphate? He is simply roaming in the form of a phosphate. That's why he is an inorganic. So this fellow is having one adenine molecule along with 2 phosphate. Therefore, is an organic molecule. But don't bother about these inorganic, organic. But you just know one phosphate is simply roaming and one adenine with 2 phosphate are roaming. Now, we are asking you guys to just come and combine. Okay. So, that is nothing but ATP. So, that reaction itself is nothing but ATP. ATP means, so this fellow is simply roaming, no? So, this fellow by having 2 phosphate, he is simply roaming. Now, we have joined this guy also. We have joined this inorganic phosphate also to this group. Now his name is ATP. ATP full form is adenosine. To this adenosine, 3 phosphate molecule we have added. That's why it is adenosine triphosphate. Triphosphate. So this is what the reaction which happens during energy production. So somewhere ADP is roaming, somewhere IP is roaming. So now this glucose, this oxygen, they are going to hold. Come here. You just become one molecule like ATP so that you need at the time of emergency. So now the ATP is ready in the cell. Okay. So why this ATP is ready in the cell? Because it will be waiting whenever a cell required, it will be utilized that. For example, I am going to have some currency at my home. Okay. So I am going to have some 1,000 rupees or 1 lakh or 1 crore. It is there in my bank account. So simply I am not going to use every time. Whenever I require, I am going to use. Suppose if I have to purchase something, if I go for a shopping, I will utilize that currency. So like that, In a cell, this ATP is there. It is an energy currency. Now, suppose, imagine this is our cell only, no? Imagine this is cell. He is full cell only. Now he is our cell. He is cell only. Now he thought that, oh, I need some work. I have to do some work. Then by that time, he utilizes this ATP. He says, hey ATP, I have made you, right? Now this is the time I need to utilize you. So by that time, it is going to just break this. So during this process, what happened? By utilizing energy, they have added it, right? Now, by using water, they are going to break this. Now ADP and IP, you get separate. So during this process, some amount of energy will be released here. The energy what you have stored, no? Now that energy will be released. So this energy, in fact, almost are equal to 30.5 kilojoules. That much value it is having when one inorganic phosphate will get breakdown from this ADP. Understood? So this is what the importance of ATP. Hope I'm very clear about this. So don't think too much. Don't bother too much about this topic. This much is enough for you. Right? Okay. Now, within respiration, we are going to see two types. So one kind of respiration, that is nothing but breakdown of glucose, which takes place in the presence of oxygen. Suppose if this glucose has to break, it asks the oxygen, you must come and you must present with me, then only I can get break. Okay, complete breakdown of glucose takes place. Then such respiration, which happens in the presence of oxygen, we call it as what? Aerobic respiration. Okay, for example, in all mammals, in all plants, in all animals, we are going to get complete energy so that end products are nothing but carbon dioxide and water. So this we call it as aerobic respiration. Okay, but in some organism, for example, yeast, and in some bacteria, and even in some lower plants, what happened? The breakdown of glucose takes place, but not complete breakdown of glucose. So that's why they are going to produce some compound which is not completely of water and carbon dioxide. In between, they are going to produce a kind of alcohol. They are going to produce lactic acid. But all these things are happening in absence of oxygen. So glucose is there. So this glucose is going to get break into small pieces. But in the absence of oxygen, therefore it is called anaerobic respiration. So if I am telling like this, I know you are going to get confused, but don't worry. Let me explain. First, let me take aerobic respiration by drawing simple diagram. I will explain. It will just get print in your mind, trust me. Okay? Done. Okay, let's talk about aerobic respiration. Let me take example of a human being only. Imagine he's a guy. So, of course, his body is made up of cells. Correct? Now, he had ingested the food has entered into his body. He had inhaled the air, that is oxygen, that oxygen also entered in his body. Now, only thing is, it has to get break. Correct? So, he says that I am going to inhale oxygen. Let entire glucose to get breakdown to get energy. So, now, the thing which is so happening, breaking down of glucose, must be taking place in a cell. It must be happening in every cell. Every cell is going to do that breaking process. So that's why let me take only one cell, let me enlarge that and let me show how this process is happening. So let's consider this is one big cell. Now whenever we talk about a cell, we know that cell organelles are present and cytoplasm is there. So this entire board is a cytoplasm, correct? Do you agree for that? Okay. Now within this cytoplasm, let me mark it. This is the cytoplasm. This is cytoplasm. So many organelles are present. For example, here our main focus is only on mitochondria. So let me draw this mitochondria first. Mitochondria is called what? It is a powerhouse of the cell. Hope you know this. Because this is the house, this is the area, the organelle where our ATP is going to form. Okay. Now imagine the cell is very happy because it knows that anyhow my body or my cell is going to generate ATP. So that's why it is asking glucose, you get in. Glucose you just get in. I know how to break you. So now the glucose which the person is going to ingest in the form of a food, now after getting breaking, after getting digested, that also further should get digested within the cell. So already in the nutrition chapter we have understood carbohydrate is made up of glucose. So that carbohydrate has broken down. Now the cell is got absorbed from the blood. Correct? So this is blood. Let me take it once again. So this is blood vessel. So now in the blood vessel the glucose is roaming. The oxygen is also roaming. Now the glucose is absorbed by our cell. Now let me write this as a glucose. So glucose, what I said, it is C6H12O6, right? C6H12O6. In fact, if I just elaborate and write only carbon molecule, it something looks like this. 1, 2, 3, 4, 5, 6. So total 6 carbon atom that glucose contains. And on either side, of course, it also contains hydrogen, oxygen, everything, but I am not writing. Now what happened? In the cytoplasm, this is cytoplasm I said, correct? In the cytoplasm, this glucose is going to get break, not with the help of oxygen. Listen to me carefully. This glucose is going to get break into two molecules. Two molecules in the sense, these three carbon component become one molecule and another 3 carbon become another molecule. So that we are going to get two molecule of 3 carbon atom. Okay, so there are 3 carbon atom molecule. That molecule we are going to call by a name as pyruvate. Pyruvate, I am not writing the definition of pyruvate or the full form of pyruvate, just I am writing this name. So pyruvate is a molecule which is formed because of breakdown of this glucose. So this process which is happening in the cytoplasm we called as glycolysis. So these things are basics. In your class 11, you are going to learn, there is a chapter called respiration. In that you are going to learn in detail. That also I have done in class 11 syllabus. It is very easy to understand. Once you understand these basic things, now you will understand everything. Okay. Now, this pyruvate now further will get breakdown by entering into this powerhouse called mitochondria. So this mitochondria is also waiting. I am just waiting here. You pyruvate just getting. Now the pyruvate has entered inside this mitochondria. Let me write here pyruvate. But for the breakdown of pyruvate, which is actually 3 carbon molecule, right? 3 carbon compound, we require what? Oxygen. That's why we say mitochondria is a powerhouse, because by utilizing oxygen, it is going to break this pyruvate. So when it gets broken down, it is going to release 3 carbon dioxide, because 3 carbon molecule was there, no? All these 3 carbon molecule will get converted into carbon dioxide, and during this process it produces water, and during this process it also produces energy. So that energy will be in the form of what? ATP. That is what we have learned. So this is complete breakdown of glucose. So here no glucose, no molecule left. Completely it has got converted into carbon dioxide, complete water as well as energy. Okay. So this type of reaction which happens in the presence of oxygen, we called as what? Aerobic respiration. Hope I am very clear about this. But in some organism, for example, as I have took just now, if you take yeast, or if you take bacteria, or if you take some other organism, they do not take this pyruvate for the further breakdown with the help of oxygen. Pyruvate will enter into the next step, but there the oxygen is absent. So in that absence of oxygen, we won't get carbon dioxide and water or complete breakdown of this pyruvate. We are going to get some other compound. So that is what we are going to learn in anaerobic respiration. I will tell you. Hope this part is clear and you have written in your copy also. Let me move on to this. So here this oxygen, the blood which is so absorbed it, no? So that also utilized by the cell, and that will be given to this mitochondria. Got it? Now let me talk about anaerobic respiration. Within anaerobic respiration, based on the end product, okay? So the pyruvate has entered. See here also, the glucose will enter into the glycolysis process and it will convert it into pyruvate. Because just now I said, for the production of pyruvate, we do not require oxygen. And here also in lactic acid fermentation, glucose will enter into the cell. Glucose will enter into the cell, that also get converted into pyruvate. Pyruvate without oxygen. But when this pyruvate entered into the next process, it will produce alcohol. That's why it is called alcoholic fermentation. When this pyruvate enters into the next process, it will produce lactic acid. Therefore, it is called lactic acid fermentation. Let us talk about this also. First, let me talk about alcoholic fermentation. Here what happens? In anaerobic respiration, let me take one cell. Imagine it is a yeast cell. It is a yeast cell. So, this is the cytoplasm of the yeast. Now, yeast says, I am going to take glucose. So, again glucose is 1, 2, 3, 4, 5, 6 carbon molecule. Okay. Now, this glucose is undergo the process of glycolysis. Because I said glycolysis do not require any oxygen. It will convert it into pyruvate. So, this is one molecule and this is another molecule. That means here also 2 pyruvate molecule we are going to get. Both are 3 carbon molecule. Both are 3 carbon molecule. Correct? So, this is same in both aerobic as well as the anaerobic respiration. But this pyruvate in aerobic respiration, what happens? Entered into mitochondria. But here, it won't enter into mitochondria. Directly, it will enter into the next step. So, those steps we call it as a fermentation process. So, in this fermentation process, what happens? It is going to produce ethanol. Ethanol is nothing but alcohol. Alcohol, and also it produces carbon dioxide. Carbon dioxide and alcohol. Of course, during this process, it also releases energy. So, ultimate goal is to get energy only. ATP. Of course, it is producing ATP, but here it is not completely producing water as well as carbon dioxide. There is an end product called ethanol. Therefore, it is called alcoholic fermentation. Okay. So, this is anaerobic. Here, they are not using oxygen. So, like that, if I take another example where oxygen is there, but not sufficient oxygen. That's why that also we have kept under anaerobic respiration, which actually happens in our muscle. Let me take one example. Imagine you are running. Every day you are not doing this activity. All of a sudden you got you start seeing a motivational speech in your mobile. You started running. So one day you started running, 2 days you started running, but third day or just one day after, your legs, your hands or your muscles are going to start paining, right? So you are going to get a muscle cramp, correct? Or if you just lift a heavy weight, then also we are going to get a muscle cramp. Why it is so? Because your body has not prepared. Your muscle cell has not prepared for such a vigorous exercise, such a heavy exercise. But all of a sudden, you did it, man. So by that time, what it has to do? It has to balance. It has to provide energy for you. By that time, you are running all of a sudden. Imagine some dog is chasing. Some mad dog is chasing at you. So if you think that I don't have energy, I don't have sufficient oxygen, let me get complete oxygen, then I will run. By the time the dog will come and bite. Correct? So that's why what your brain thinks, first you run. At any cost, I will supply some sort of energy, don't worry. So by the time what happened, the muscle cell, imagine this is our muscle cell. Our muscle cell is something like this only. It is like a cylinder shape. Okay. So in this muscle cell, as usual, here also the glucose will be supplied. You need energy for running. You just take this glucose. So this glucose once again will break down into pyruvate by a process called glycolysis. Because it is the cytoplasm. This is a muscle cell. Whenever I talk about a cell, of course, cytoplasm must be there in that. Correct. But for complete breakdown of this pyruvate, muscle cell is not having sufficient oxygen. So by that time, what happened? The muscle cell thinks, at any cost, you need energy, right? So if you just keep waiting for oxygen, of course, you are to be in a trouble situation. Therefore, you just keep run. I will do something. So what it will do? It will enter directly into the fermentation process. So in this fermentation process, what happened? It will produce a chemical called lactic acid along with the production of energy. So our ultimate aim is to get energy. So that energy we got. But we must get carbon dioxide and water, but that product is not producing here. Why? Because we are not getting sufficient oxygen. Therefore, because of insufficient oxygen, lack of oxygen, we are getting this lactic acid which has been accumulated within our muscle cell. So because of this lactic acid accumulation only, we are going to get cramp Oh, I am feeling pain in the hand, I am feeling pain in the leg. Why it is so? Lactic acid. This lactic acid gradually, when it gets sufficient oxygen, then converted into carbon dioxide, then all the pain, everything will get relaxed because it will enter into the blood. That process is different. Okay. So this is what about aerobic and anaerobic respiration. Hope I am very clear. So in our textbook, this schematic part has given. If you are unable to see this, you can open your textbook or you can watch here only in the side. I will explain. Just now what I have explained, no, the same thing has provided here. So glucose, it is a 6-carbon molecule. So in the cytoplasm, they have not mentioned glycolysis, but actually this process we call glycolysis. It produces pyruvate. Pyruvate is a 3-carbon molecule. Correct? So during this process also some quantity of energy is produced, but this is not enough. It is very little energy. Okay? Very 2 ATP. Do you know one thing? When one glucose has completely get burnt, our cell is going to produce 38 ATP. It is quite shocking, but in fact it is true. But in glycolysis, we are going to get only 2 ATP. Do you think is that enough? No, that's why cell thinks, anyhow, I have taken glucose. Why should I waste energy? Why should I just leave ATP? Let me produce more and more. That's why it will just push this pyruvate into mitochondria. Okay. But in anaerobic respiration, this thing won't happen. That's why there very less energy will be produced. Okay. You just forget that. Now you see. Suppose this pyruvate, we are focusing on this pyruvate, right? Suppose this pyruvate, if it is entered, if it is entered into the mitochondria in the presence of oxygen, what happens? It produces carbon dioxide plus water and energy. So complete energy it got. This is happen in the presence of oxygen. Suppose if there is no oxygen, then what happens? Example yeast. It will produce ethanol. It is a 2-carbon molecule. Of course, here also carbon dioxide, energy will be produced, but not that much energy. Like that, if there is insufficient oxygen, lack of oxygen in muscle cell, it produces 3-carbon molecule called lactic acid along with energy production. So this equation, this part is very, very important in your textbook. Okay, from respiration part. Hope I'm very clear. Let me move on to the next part. Okay. So we have understood how this process happens in animals, okay, or even in plants also. But if I just take exactly the plants, animals we can understand, but exactly in plants, many students get confused, ma'am. Plants actually take carbon dioxide, they are going to give oxygen, correct, ma'am? Yes, my dear, that is correct, but that happens during photosynthesis. Ma'am, even in respiration also, we think that they are going to take carbon dioxide and release oxygen. No, that is a wrong statement. In respiration, even plants also do the same process what we are going to do. They will also take oxygen and gives out carbon dioxide. It is quite confusing, correct? But let me clarify by drawing diagram here, okay? So imagine it is a plant. It will become a white plant. Okay, so this plant, just now I told no, this plant will do photosynthesis, this plant will do even respiration also like that, okay? Suppose if I just take only one leaf. In that leaf, small cells are there, no? Small cells will be present. We call these cells as mesophyll cells. Mesophyll cell. And this is one leaf. Suppose if I draw a big cell like this, I'm just going to mention only one mesophyll cell. Don't get confused. Let me draw the same diagram in the next slide. Okay? Okay, imagine this is our leaf. A big leaf. So whenever we talk about a leaf, we know that stomata will be there. Correct? For the exchange of gases. So imagine here the stomata are present. So already I have explained in photosynthesis part. But here I need to tell the difference between the photosynthesis as well as respiration. That's why I'm taking here. Okay. Now, so this is what the mesophyll cell. So whenever we talk about a cell, of course, cytoplasm must be there. So here it is the cytoplasm. Cell organelles must be there. Yes, here is the cell organelle. So this is the mitochondria, powerhouse of the cell. This is the chloroplast. So all these organelles are present within the cell, correct? Now, imagine the glucose has entered into the cytoplasm. How glucose has produced? By the chloroplast, by a process called photosynthesis, correct? Now imagine that glucose itself has entered into the cytoplasm. Now glucose is here. Glucose is here. Glucose means 6 carbon molecule. 1, 2, 3, 4, 5, 6. Same to same. Now this glucose, just like in plants, just like in animals, even in plant also undergo a process called glycolysis. Glycolysis. It means it also going to get break into 2 molecule of 3 carbon atom. That means which molecule it is? Yes, you are right. Pyruvate. So pyruvate is a 3 carbon compound. Correct? Correct. Now this pyruvate says, anyhow, I have mitochondria. Why should I waste producing ATP? Instead of producing ATP, why should I simply waste by lying here? Let me enter into mitochondria. So pyruvate entered into mitochondria. So now here is a pyruvate. This pyruvate will combine with oxygen and produce ATP. That is energy currency. During this process, it also produces carbon dioxide and water. Correct? This process is clear. That's why here also it is a powerhouse of the cell. Now, look at here carefully. Now the carbon dioxide which is so produced here during respiration process. So this process, what you are seeing, what is this process? This is respiration. Correct? Breaking down of glucose in presence of oxygen. Yes, it is happening. Now this carbon dioxide, why we require? For photosynthesis process, right? So cell says, chloroplast says, hey, you carbon dioxide, get inside. I need more carbon dioxide. Okay, so that's why the carbon dioxide will enter inside the chloroplast only. Correct? But ma'am, you said through stomata the carbon dioxide enter. Yes, my dear. The carbon dioxide which is so produced during the respiration is not sufficient. Is not that much enough. That's why plants will demand for more and more carbon dioxide. That's why whatever the carbon dioxide that we are going to breathe out, correct? Animals are going to breathe out carbon dioxide. Human beings are going to breathe out carbon dioxide. All that carbon dioxide will be captured by the plant through stomata. Okay? So that's why, look at here. Here also carbon dioxide is coming. Through this process also the carbon dioxide. Okay, let me take some other color. Yes. In this process also the carbon dioxide is coming. So this is carbon dioxide. So this also entered inside only. So this is also carbon dioxide, this is also carbon dioxide, but it required lot of carbon dioxide. It has entered inside. Now plants are going to absorb water, that everybody knows. By combining with water, it is going to produce glucose plus oxygen. Correct? Glucose plus oxygen. Now this oxygen, what the plants are produced during the photosynthesis process. This process is what? Photosynthesis only, no? Photosynthesis, this is respiration. So this oxygen will be utilized by plant itself for respiration process. But the oxygen which is so produced, it is more and more in number. So therefore plant says, do one thing, you take this oxygen, what I have produced here, you just take this oxygen in mitochondria, and rest of the oxygen what I have produced now, let me give it out. Let me give out. Okay, did you understand what I am trying to tell? Let me make this color indifferent. Okay. So this oxygen will excrete out through this stomata. Excretion in the sense, it will be goes out through stomata. Okay, and the carbon dioxide will enter. So this thing will happen during daytime. Correct? But during night time, what happens? During daytime, photosynthesis taking place. Therefore, it is going to utilize the carbon dioxide which is so produced within the mitochondria, and even it is going to take in carbon dioxide even producing in the atmosphere. Therefore, we says, okay, the plants are going to take carbon dioxide during the daytime and gives out oxygen. They gives out oxygen. Yes, we do agree for that. But when it comes to respiration, what they are doing? They are also utilizing oxygen. They are also giving out carbon dioxide. The same process they are doing just like that. Now imagine, let me make it clear. So during night time, So during night time, there is no sun, no sunlight. So no sunlight means no photosynthesis. So if there is no photosynthesis, what process should take place? Only respiration. So for that respiration, what they will do? They are not going to produce any oxygen here. Correct? So they have to depend on outside atmospheric oxygen only, no? So during night time, they are also going to take oxygen through stomata and they are going to release carbon dioxide. So this carbon dioxide which they are going to release, instead of utilizing here, because there is no photosynthesis, so they will just send it out. That's why elders are going to suggest that don't sit or don't spend more time under the trees during the night. Why? They were simply telling that ghosts will be roaming, something like that. No, all these things are false. Their main concern is to make you get alert, because under the trees, especially in the night, more and more carbon dioxide will be releasing. So it is quite suffocating. So that's why they were advising like that. Okay. So in the daytime, carbon dioxide will be taken, oxygen will be released. Why? Because for photosynthesis. During night time, they will do like us only. They are also going to take oxygen, release carbon dioxide. In fact, whenever we talk about respiration, one thing should be clear in your mind, that even plant also take oxygen and gives carbon dioxide. Hope I'm very clear about this part, about photosynthesis as well as the respiration difference in plant. Let me move on to the next part. Okay? Yes. Okay. So till now we have understood what happens or how respiration occurs in plants or in the animals when they get sufficient oxygen. If they are present on a terrestrial environment, yes, they do get sufficient oxygen. They are going to do all these things. Suppose if an animal, if it is present in a water, does it going to get the same quantity of oxygen? No. In water, in fact, in water, the oxygen will be in dissolved condition. So they are not going to get sufficient oxygen. Therefore, the aquatic animals are going to take more and more, or they breathe very fast. Why? Because the oxygen is very lacking. It is in a dissolved state. Therefore, as soon as possible, they are going to breathe and they are going to breathe out. So how it is going to happen? Let me draw a diagram of a fish and let me explain. Okay, imagine it's a fish. So let me put lipstick for this fish. Okay. So everybody, we know that since our childhood, we are studying that gills are the main respiratory structure in this organism, correct? So how the gills are present and where they are located? Like this. Gills are like a box. And here, so many slit-like structures, small openings will be there. In some fishes, this opening will be covered with a covering. We call it as a operculum. For example, look at here. Suppose if I draw a fish, if it is having a gills and gill slits, it will be covered by a structure like this. So this structure is called operculum. But if I just show operculum, you can't see how the gases are exchanging. Therefore, I am not showing operculum here. Okay. So this is nothing but gill and gill slits. Very simple. Very simple. What it is going to do, it is just going to take water first. So in the water, the oxygen is there in dissolved condition. Correct? Now when it takes the water, it will enter into the gills and that dissolved oxygen will be absorbed by the gills. Okay? So it will get absorbed. Here the blood vessels will be there. Here so many blood vessels will be there. They are going to absorb this. They absorb this oxygen. And finally, the water will come out. So this water, the water will be ejected out through these gill slits. This is what the process. In the gills, they are going to exchange oxygen and carbon dioxide. And the rest of the things that will be water, it has to move out, no? If it keep drinking water, it will become bulge and get blast. So that's why the water will be goes out. That will happen through this gill slits. So this is what the simple process happen in aquatic animals. So one thing we have to remember here, they breathe very fast. They breathe very fast. Why it is so? Because the oxygen is in dissolved condition. So that will happen with the help of gill and gill slits. That's it. Look at this image, you can understand. So oxygen is entering. In fact, oxygen means here, it is present inside the water. It is entering here through the mouth. And this is a gill arch, means these are the gills that will utilize this oxygen and carbon dioxide will be goes out. Can you see? Here blood vessels will be present within these gills that will absorb this oxygen. And from the body, the carbon dioxide which is so there, that will be gives out and that will be goes outside through water itself. Okay, done. Now, let us talk about in detail about human respiratory system. It is the important part of this topic of respiration. Okay? Yes. So look at here carefully, you will understand everything. So imagine a person, or else I'll do one thing. I need a bit more space. Let me go to the next slide. Okay. So we know that it is the nostril. That is nostril. We are going to inhale air through the nostril. Okay. So the air you inhale, it will go into this chamber called nasal cavity. And the food what we are going to take, that will enter into here. This is called buccal cavity. All those things I have told. And nasal cavity and buccal cavity will be get separated by a structure called uvula also. Okay, let me mark it. And I already told while explaining nutrition part itself, this is the common structure. This is the common structure through which both air as well as the food will enter. So what is this structure we call? Yes, pharynx. Okay. So now, here this pharynx is going to show bifurcation like this. It's going to show bifurcation. So imagine if there is a food, the food will get confused where I have to move, whether in this direction or in this direction. If the food moves in this direction, it is the right direction for the food. Why? Because this is the esophagus. Esophagus means what? It is a food pipe. Suppose if the food moves in this direction, it is a wrong direction. Why? Because it is a windpipe. So windpipe is nothing but technically we are going to call by a name called trachea. Trachea. Okay. So that's why to prevent this food from being entered into the windpipe, there is a structure. There is a structure which can prevent it. So this, in fact, we call it as a voice box. Voice box, or it is also called larynx. On this larynx, there is a, see, if it is a larynx, this opening, we call it as a glottis. On the glottis, above the glottis, there is a cap-like structure. It is called epiglottis. So this epiglottis is going to prevent the food. It says, hey food, don't get inside. If you get inside, the person will get choked. Okay. So you have to stay. It will close. That's why elders are advised us not to talk while eating. Okay. So now let us talk about the food, because our main focus is only on what? Respiratory system. Digestive system already we have understood. Okay. Now let us talk here. So this thing, this is actually opening like this. It will continue further. This food windpipe will continue further. This is trachea, I said, no? So this trachea, it is a pipe-like structure. It should not go, it should not get collapsed. If the air enter, of course, it will be stiff only. Stiff, it will be strong. Imagine you have a straw, you are just drinking some juice. So if you blow air, the straw will be normal and bulge. If you just pull the air, if you just do like this, then the straw become collapsed, no? But it won't happen like that in our case. When you just breathe out air, our trachea will not get collapsed. Why it is so? Because there is a protective ring present around this. Okay. So these protective ring look something like this. They are going to protect like this. So these protective rings are called cartilaginous ring or hyaline cartilaginous ring, because it is made up of a tissue called hyaline. That's why hyaline cartilage. Now this trachea further get branch like this. It further get branches So this branch is called primary bronchi. Shall I mention? So this primary bronchi further get branches like this. Secondary bronchi, then tertiary bronchi, then further branches bronchioles. So like that they are going to get so many branches like this. So all these branches we are going to see within the lungs itself. So all these branches are happening in the lungs. So the same thing we are going to see in another lung also, but I am not mentioning, that's it. It is same to same. That means what I said, these branches are primary, secondary bronchi, and these are bronchioles. I am not explaining in detail because you are going to learn everything about the detailed, minute structure, everything in breathing and exchange of gases. There is a chapter. There is a chapter called breathing and exchange of gases in class 11. So that also have done in class 11. Full syllabus already have told, no, it is there. When you come to class 11th now, you can understand each and everything. It is so easy and so interesting to understand. Okay? Okay. See, when you come at the end, here there are so many sac-like, balloon-like structure we are going to see. Like this. At the end. Let me show somewhere else here. Imagine this is the branchiole. This is branchiole. So this branchiole, like this, the branchioles are present at the end. So these branchioles are terminated by many sac-like structure. So these sacs are nothing but alveoli. Alveoli. In fact, to these alveoli, the blood vessels are surrounding like this. Blood vessels are surrounding. Understood? But for our understanding purpose, we can just show like this. If it is alveoli, this is our blood vessel. So that if any gases come, it can get exchanged with this. Understood? So this is what our human respiratory system. Hope this part is also very clear. Now, when you just breathe in, when you take oxygen, if you observe, if you just put your hand on the chest cavity, it seems that the chest cavity is expanding. Correct? Like that when you breathe out, we do relax. So, what happened exactly? Why this chest cavity get enlarged and why it is coming back to the normal position? This is nothing but mechanism of breathing. So, when you do like that, when chest cavity get expand, then only our lungs get expand and oxygen will enter. Okay, so when chest cavity comes to the normal position, the lungs also come to normal position, we will give out carbon dioxide. So who is going to expand then? It is by two types of muscle. Okay, so that I will tell you. Hope this part is clear. Please make a note of this. Okay, done. Let me talk about mechanism of breathing. Look at here carefully. If he is a person, if he is a human being, this is, we call it as a body cavity. This is the person's body cavity. Okay, I know these things are very new to you, but don't worry. You just know this is the body cavity. This body cavity can be bifurcated into two chambers. How? With the help of muscular structure. There is a muscular structure present like this. This muscular organ is known as diaphragm. Have you heard this? Diaphragm. So this diaphragm separate this entire body cavity into two chambers. So this upper chamber is called thoracic chamber and the lower chamber is called abdominal chamber. So in this thoracic chamber, our two lungs are situated. See, this is one lung, this is another lung. Okay. So in all the four corners, it has been covered completely so that it is an airtight chamber. Okay. Now, when this diaphragm make a movement, the lungs can come down. Or else, I'll do one thing. I will just take out only the thoracic chamber and let me show how it is going to happen. Imagine this is the thoracic chamber. Okay. So in this thoracic chamber, the downwards are nothing but diaphragm is present. I told, right? This is diaphragm and this is the lungs. So the lungs are crying. We don't have enough space. You are telling that you have to expand, you have to expand. How can we expand? I don't have space. By the time diaphragm says, don't worry, my dear, I am going to contract down. So contraction means what? So like this diaphragm which is so there, so like this, the diaphragm which is so there, no? Now the diaphragm says, I'll do one thing, I am going down. Don't worry, I will go down like this. I will go down. So when this diaphragm goes down in this direction, here it got space, correct? So lungs got space so that the lungs can expand up to here. Lungs can expand up to here so that the lungs become larger. Like that, even the lungs are also surrounded by ribs here. It will be having ribs on either side. Now, between the ribs, there are certain muscles present. Now, these muscles is, we are going to contract. So, we are going to go in this direction so that we can expand in this direction. So, when they move, imagine these ribs are moving. Look at here. They are making space for the lungs to get expanded in that direction also. So, automatically what happened? What happened? The lungs can move in this direction also. Correct? So, lungs are expanding here. So, earlier lungs were like this. Now, they are expanding in this direction on either side. Now, they are expanding in downward direction also. So, therefore, automatically the lungs become so wider, and because of this thing only, the oxygen will easily enter. When the lungs get expand, oxygen enter. So, when this diaphragm, okay, enough oxygen you got, now let me come back to the normal position. Again, it will go up. That we say relaxed position. So, by the time ribs also will come to normal position. So, by that time, the carbon dioxide that will be forced outside. So, this we call it as mechanism of breathing. So, here is a small clipping. You can understand how this mechanism is happening. Okay. Now, let's imagine how the gases are going to get exchanged when it entered into the alveoli. Because we have understood, let me come back. We have understood the oxygen what we are going to inhale. It has entered into the pharynx, entered into trachea, bronchi, primary bronchi, secondary bronchi, bronchiole. Finally, it will enter into alveoli. Okay. So, let me draw alveoli structure. So, imagine this is one alveoli. But this alveoli is surrounded by so many blood capillaries like this. But for our convenience, let us understand this same diagram in this way. This is our alveoli and this is our blood capillary. Blood capillary. Okay. So here what happened? The thickness of this alveoli as well as the blood vessel is very, very thin. So therefore, the oxygen which has so entered inside the alveoli can easily get diffused. What is diffusion? By a process called diffusion, they are going to exchange. You know about diffusion? Diffusion means movement of molecule from region of higher concentration to lower concentration. Let me take an example. Imagine I have a perfume bottle. So in this room, the fragrance is very less, or there is no fragrance in this room. So the fragrance or molecules are very heavy in perfume bottle. If I just spray it, what happened? Easily the molecules of those perfume will spread inside the room. That is nothing but diffusion. Region of higher concentration spreading to region of lower concentration. Correct. So here also oxygen molecule concentration are very high compared to blood. So therefore oxygen is moving or diffusing. Okay. Like that, the carbon dioxide which is so carried by different part of the body. Because when cell break this glucose, carbon dioxide is going to produce no. So that carbon dioxide will be carried out into the blood and again they are going to give it to alveoli. So it is by a process called diffusion. They are going to exchange the gases. Okay. So like this, the gases get exchanged, that will be breathed out. Now the question is, when they get exchanged, imagine the oxygen has entered inside the blood. So how they are going to take to different part of the body, because our body is very complex. So who is going to take? That is very simple. Very simple. Let me draw here again. So imagine oxygen is here. This is the blood. This is the blood. Now it has to transmit through the blood. The oxygen has to transport through the blood and it has to come near to the tissue. So imagine this blood is like a river. Okay. Now in this river, how we are going to travel? Tell me how we are going to travel in the river. So very 3% of oxygen. We call it as a solubility. So 3% oxygen says, we know swimming, we are just going to be dumped inside this blood. So they are just going to jump inside the blood. They just swim towards that means within the blood, they go towards the tissue. But remaining 97% are there, no? So what they will do? They are very panicked. They don't know swimming. These oxygen molecules. So that's why they says, hey blood, you have RBC. Correct? So we know that blood is having RBC. So these are the RBC. Let me take some other color. So these are the RBC which are roaming within the blood. So they utilize this RBC as their boat because within RBC we know that hemoglobin is present. Okay. Suppose if it is one RBC, within the RBC, imagine these are the hemoglobin. Now, to this one hemoglobin, I am just enlarging the structure. One hemoglobin, if I enlarge, it has 4 seats. That means it can combine 4 oxygen molecules. So hemoglobin says, you 4 oxygen, you come and sit on me. I can carry you. Then we call this structure as oxyhemoglobin. So like that, every oxygen will enter inside the RBC, they sit on hemoglobin, they will just move. So like that, they will roam and come towards the blood vessel, towards the tissue. It looks like a bone. One second, wait. So every RBCs are carrying oxygen. They are taking oxygen and give near the tissue. So they will deliver the tissue. So like this, they are going to transmit or transport throughout the body. So this is what the mechanism. Hope I'm very clear. Now if you read textbook, you can understand each and every word of your textbook and every line of your textbook. Okay. Yes. This hemoglobin is also called respiratory pigment. Why? Because they are carrying this respiratory gas. That's why. And compared to oxygen, carbon dioxide can dissolve very fast. Its soluble capacity is very more compared to this. Okay, let's move on to the next topic. So the topic of respiration is very clear. Let's talk about transportation. Okay, now let's talk about another life process, transportation. So what is transportation? Movement of molecules. That is from place to place. Just like how we are going to travel in a bus, that is nothing but transportation only. So like that, in our body also, there are so many material are going to be synthesized in a particular place, but they have to be transported to different organs. For example, we know that lungs are going to inhale the oxygen, correct? So this is our lungs. Through the nostril, we are going to inhale oxygen. It is there. But the oxygen should not be restricted in the lungs. It has to be transmitted, it has to be transported to all parts of our body, right? So it has to be needed for our leg, that is to our hands, leg, muscles, everywhere oxygen is needed. So like that, if I take an example of a digestive system. So here what happened? The food molecule, what we are going to eat, when it gets digested completely, it will become a simple molecule like glucose, amino acid, everything. So once again, it should not be retained here itself. It has to be transported to different organs, to different cells, so that there it has to provide energy. So who is going to transport? Okay, just like how I take the example of a bus. With the help of a bus, how we are going to travel to different places with the help of a train, aeroplane, anything. Like that, in our body, there is a transporting agent. So that transporting agent is nothing but the blood. Okay, so blood is a transporting medium here. So what it is going to do? As I told just now, if your digestive system consists of the food molecules in a simple way, like glucose, amino acid, so like that, if your lungs are going to have the oxygen, then with the help of the blood, imagine this is our blood vessel, it will be get absorbed within the blood. So blood is going to take this, so you oxygen get inside, so you glucose get inside. So now with this medium, that is nothing but with the help of the blood, it will be transmitted to different parts. For example, if it is required to a tissue, it will come near the tissue, it will deliver to the tissue. So oxygen you need, take this. You need glucose, take this. So this tissue is happy now because he got all the molecules. Correct? So this entire system, that is nothing but blood only, but blood alone cannot do the work. Correct? Blood alone cannot do the work. Because if you just look at this diagram, you can easily analyze. I have drawn a tube-like structure. Correct? So a tube is required, within that blood is there. So like that, if this tube is carrying the blood, somebody has to pump it. For example, in an over tank, if you have a water, that is a reserve of water, then only if you just put a motor switch, then only what happen? It will allow this water to travel or to move in a different pipe. So then only you could able to get the water in your kitchen, in your bathroom, in a toilet, everywhere. Correct? So like that, here, whenever we talk about any transporting system, particularly in a human being, we are going to give a name. What is that? That is nothing but circulatory system. So in a human circulatory system, mainly it consists of 3 components. What are those? One is nothing but the blood. As I have told, it's a liquid substance. But this blood cannot move, it cannot flow. It requires a tubular structure. So those tubular structure, it can be thick, it can be thin, it can be very, very delicate. But there are different types of tubes are there in our body. But altogether, we are going to call it as what? Blood vessel. So like that, as I have told just now, somebody should be there to pump it. Somebody should be there to make this blood to flow in a pipe to the different parts of our body. That is nothing but heart. It is a pumping organ. So now let's talk about all these parts one by one easily, and interesting part also we can understand. Don't worry. It's my responsibility to make you understand easily, but only your responsibility is to focus. Okay? Yes. Now let's talk about blood. What is blood? So in your lower classes, that is in your class 9th itself, we have studied about different types of tissues. Muscular tissue, connective tissue, all those things. So blood is also one of the fluid connective tissue. Okay. So whenever we talk about a blood, so the things comes into our mind is it is red in color. Correct? It is liquid in nature. But in fact, within the blood, there are so many cells are also present. So therefore, the blood, okay, let me write here only. We are going to say the blood is going to have 2 components. So how many components the blood is having? 2 components. So the first component is the liquid component or liquid part of the blood. It is called plasma. And another component is nothing but cellular component. So very simple. Imagine I have a test tube. So in this tube, let's assume I have filled blood. Okay. So let's assume here is the blood. So obviously in which color it is looking? It is looking in a red color, correct? But if I separate, as I have told just now, it is having the liquid part, it is having a cellular part. Like that, if I just separate it, I am going to get 2 things. 2 things means, see, let me draw here itself. So one thing is only liquid part. So that liquid part is somewhat yellowish in color. See, I will write here. In fact, there are so many cells are present. But we can't able to see these cells because the red color is so bright that other cellular part cannot be visible because they are colorless. So here, this white color cells are also present. And small cells are also present along with these red color cells. Okay. So, first let me write those cellular part. So those cellular parts are nothing but RBC, that is red blood cell. So the white color what you are seeing is nothing but a WBC, white blood cell, and another one is platelets. So these are the three cellular components. That means cells present within the cell. Now what I am going to do? I am just take out all those cells. Correct? So let me take out all those cells here. So these are the white color cells. So these are the red color cells. And let me consider these are the platelets. Okay, now I have separated everything. Only cellular part I have separated. Now I have left with only liquid. So that liquid, if I take it in another test tube, imagine I have taken another test tube. So in that I am just making this liquid part to get placed here. Then the liquid will be somewhat yellowish. So this is what the liquid present inside. So this liquid we call it as plasma. Got it? So let me write here. So this is the plasma. This is the cellular part. And these two components together form a structure called blood. Okay. So what we have learned? We have learned blood is a liquid in nature. In that the liquid quality comes because of the plasma, and rest of the things are nothing but cellular component. Those cellular components are RBC, WBC and platelets. Now the question is, what is the function of blood, ma'am? Why it has to present in our body? Just now I told. In our body, so many materials are going to be formed. So many material has to be transmitted from place to place. So who has to carry that? Of course, that should be carried by a liquid, right? So that liquid is nothing but blood. So here in the blood, especially in the plasma, all the glucose, all the waste material, everything is going to be present in a dissolved state. See, let me show here. Imagine this is our cell. Okay. So now this cell says, I need oxygen and I need glucose. So with the help of this, I can prepare energy. I can get energy, and here I am going to produce carbon dioxide. So this carbon dioxide has to be taken to the lungs. Correct? Who is going to take this? Of course, our blood. So this is our blood vessel. So the cell is going to dump this carbon dioxide into this blood, especially in the plasma. That means in the plasma, carbon dioxide is present. Correct? So it will be present in a dissolved state. So like that, here the glucose is there. Cell got glucose. Who has delivered it? Of course, blood only. So within our blood, the glucose was there. This glucose has been delivered to the cell. So like that, if this glucose has to deliver to another cell, this cell is also waiting. It will be delivered. So where this glucose is present? Of course, within the plasma. Okay. So glucose is present. It is not only just glucose. Your amino acid, your other materials like electrolytes, sodium, potassium, everything, they are present in a dissolved state. So therefore, I can write in the plasma, carbon dioxide is transmitting, food materials are transmitting, and even certain electrolytes, certain minerals, all these materials are transported by dissolving in the blood plasma. Okay, shall I write here? Dissolved state and transported. And what is about the oxygen, ma'am? How the oxygen is going to transport? So oxygen is not going to dissolve. Of course, only little bit is going to dissolve. As I have told just before this slide, only 3% of oxygen is going to be soluble in plasma. So rest of 97% who is going to carry? Just recall. Yes, our RBC. But RBC is also part of our blood, right? So RBC is going to carry oxygen. It carries oxygen. So like that, whenever any reaction occurs in a cell, say for example, material A combines with material B. I don't know which material it is, anything it could be. If A is combined with B, some useful product is going to form. Imagine some C is going to form. But along with this C, let's consider there is a material called D is also forming, but D is a waste material. Our cell do not require this D. Then what our cell will do? Cell says, anyhow, I do not require. But D has been produced because of the metabolic reaction. Because of the reaction what I have carried inside the cell. So that's why you blood, you take it. Take this and just dump it. Okay, just remove it along with the fecal matter or just remove it along with the urine. So as you know, urea is a waste material, ammonia is a waste material, uric acid is a waste material. So all these waste material are going to be dumped within this blood only. They will be transmitted to, that means that will be ejected out of the body. That means in the plasma, I can write even waste materials are also get transported. So urea and uric acid. Understood? So this is about the blood and its function. But our circulatory system not only consists of blood, it has to transmit or transport through a pipe-like structure, that is blood vessel. And who has to pump this? Of course, our heart. So before going to that pipe-like structure, blood vessel, let us talk about heart. So hope this part is clear for you. Let me move on to the next slide, that is heart. So what is heart? Heart is nothing but it's an organ. Where it is located? It is located between two lungs. Do you know that? I have told it, right? So if you consider this is our thoracic chamber, not thoracic chamber. This is our coelomic chamber. Diaphragm is going to separate, just now I told, in the respiration part. And these two are the lungs. So these two are the lungs. If I just stand like this, and if you consider these two are the lungs of my body, then obviously this would be the left side, correct? So this would be the left lung and this would be the right lung. So between these two lungs, the heart is going to be located. Especially it is tilted, slightly tilted towards the left side. So that's why we say that heart is present in the left side. But actually, in fact, the heart is not present in the left side, present between the lungs. Like this. Like this. Therefore, our left lung is quite smaller compared to right lung. Why? Because it has to provide the space for the heart. So here is our heart. So what is the size of our heart? If you just close your fist, okay, like this, and how much it looks, what is the size that you are going to see, that much size your heart is. Okay, so this entire heart is made up of muscle. Therefore, we say it is a muscular organ and it is going to pump the blood. Therefore, it is also called pumping organ. So, it is the size of our fist. Where is located? What is the location of the heart? Location is in thoracic cavity. Okay, now let's talk about heart. So, it is very important structure. Please understand it clearly because it is very, very important. Even in your higher class also you are going to study about this, but it is a basic. I have done, as I have told, have done a complete syllabus of class 11. In that class 11, there is a chapter called body fluid and circulation. So there you are going to learn about the blood. You are going to learn about the heart again. You are going to learn about double circulation, everything what I am going to do now in a circulatory system. So everything you are going to learn there. So you can see the video already have done. But if you are a neat point of view, if you are watching this video and if you are a neat aspirant, definitely these are nothing but like a basics. Please listen it carefully, but it is very easy, you can understand. Now, heart consists 4 chambers. Everybody know that. Upper 2 are called auricles, lower 2 called ventricles. Okay, so let me write. So this is one chamber. So like that, there is another chamber. Okay. So these 2 chambers are nothing but auricle. So this side is called left auricle. LA means left auricle. RA means right auricle. Ma'am, this is right auricle, this is left auricle. No, no. If you just turn the person like this, if you just look at my heart, I am standing in front of you. If you want to study my heart, how we are going to see? This would be the left side, right? And this would be the right side. So that is what I am drawing here. So this is the left auricle. This is the right auricle. Some people say it is atrium. No problem. Both are same. Atrium, auricle, both are same. auricle. So these two are the two chambers. Now below these two chambers, ventricles are present. So this is one ventricle. This is another ventricle. Okay. So now this is right ventricle, this is left ventricle. V stands for ventricle, A stands for auricle. Right? Now, imagine the two lungs are above. Right? Two lungs are above. Imagine these are the two lungs. Now from these two lungs, oxygenated blood is coming. Let me take it in, which color shall I take? Okay. Okay, I'll take yellow color. So now imagine the two pipes which are actually carrying oxygenated blood. So they are giving this to left auricle. So these two pipes, what I have drawn here, we call them as pulmonary vein. These two are pulmonary vein. So like that, here to the right auricle, from our different part of the body, that is from our lower part of the body as well as from the upper part of the body, a big veins are carrying deoxygenated blood. Whenever the blood combines with carbon dioxide, we call it as a deoxygenated blood. Here, the blood which is so coming, it is oxygenated blood. Okay, so this is oxygenated. So let me write, this side is oxygenated blood. So like that, from different part of the body, the blood which is so carrying carbon dioxide is called deoxygenated blood. See, from upper part of the body, this pipe is carrying deoxygenated blood. From lower part of the body, this pipe is carrying. So they are just dumping here. So here in the right auricle, deoxygenated blood is there. So which pipe these are all? Yes, these are nothing but vena cava. So this is superior vena cava. It's the largest vein. This is inferior vena cava. So what they are carrying? They are carrying deoxygenated blood. So this is clear. Now this oxygenated and deoxygenated blood will be pumped into this ventricle with the help of a valve. So here, if you observe carefully, we could able to see a kind of wall present between these two. Like this. So with the help of this wall, the blood will be moves down. Okay, here actually two walls are present. Here three walls. 1, 2 walls here. Here 1, 2, 3 walls. So with the help of this, the blood is coming down here. So we have given a name to these walls. So these are having two cup-like structure. Therefore, it is called bicuspid wall. And here three cup-like structures are there. Therefore, it is called tricuspid wall. So right now the blood is here. Imagine the oxygenated blood is here in the left ventricle. Deoxygenated blood is there in the right ventricle. Till we are not talking about the function. I am just showing the structure. That's it. But the blood which is over there, it has to transmit it to different part of the body. Correct? So who has to take? Again, there must be a pipe. So this pipe is arising from this structure. Imagine this is the pipe that is arising from this structure like this. Okay, so this pipe is actually carrying blood from this structure. So like that, there is another pipe arises from this. So another pipe arises like this. So this pipe is actually carrying what? Deoxygenated blood. So we have given a name to these pipe. What are the names we have given? So this pipe is nothing but it is the biggest artery called aorta. And this pipe which is actually carrying what? Deoxygenated blood, it is called pulmonary vein. This is pulmonary vein. Now, if you observe, there is a space. Now, this entire heart is actually connected by or protected by a muscle like this. It is going to be protected by muscle. And here in this region, there is a space, and here there is a region present in all these areas. So this region we call it as a septum. So very simple structure the heart is. So now we need to understand how the working mechanism takes place, how this heart is going to function. Okay, so hope this part is clear. Hope you don't have any doubts. Now let's move on to the function part. So for drawing the function, I will write very simple so that you will understand even still better than this. Okay. Okay, let's talk about the function of the heart. So I need more space. So let me move on to the next slide. So let me draw here. Okay, so what I have told. So this is actually the protective covering of the heart. So these are the 4 chambers, correct? So this is left auricle, right auricle, left ventricle, right ventricle. So this much it is clear. And this is the protective covering. It is called pericardium. Write down this word also. It's a covering of heart. So what I told, from here the aorta arises, from here the pulmonary artery arises. That's what I said. Let me draw that also. So imagine this is aorta. And this would be pulmonary artery. So here, between 2 auricle and between 2 ventricle, the region which is so present, we call it as a septum. In fact, if I have to show here very clearly, so this is auricles, and the region which is so present, this is the septum. So like that, this would be the ventricle, and the region which is so present here, that is also septum. So this entire heart is protected by the pericardium. So this is what the thing we have understood. And these 2 are the auricles and these 2 are the ventricles. Okay. Now, here, listen to me carefully, very simple. Imagine these 2 are the lungs. The lungs are waiting. So now lung says, I have oxygen. So do you need oxygen? You just come to me, I will give oxygen and you just give carbon dioxide to me. So blood vessel says, okay, we do agree for that, just give oxygen. So by thinking that these 2 are the blood vessels, they are coming from the lungs. See, they are coming from the lungs. So directly, which are so carrying oxygenated blood because the oxygen has delivered here. So now this oxygenated blood will be dumped into this right auricle. So till then that it was very in a closed structure. We says that it was in a relaxation state. Now in a relaxation state, all these blood is going to be filled in this chamber. Okay, now blood is filling in this chamber, that is left auricle. So which blood it is? Yes, it is oxygenated blood. Which is oxygenated blood coming from this. So this structure, what we have said, this structure is nothing but pulmonary vein. This is pulmonary vein. So like that, from different part of the body, carbon dioxide is coming. So that carbon dioxide has to transmit, right? So once again, here to the right auricle, all this carbon dioxide rich blood is going to be dumped. So imagine this is the carbon dioxide rich blood. It is coming. That is from the vena cava, it is dumping here. See, it is dumping. So now both the chambers are filled by the blood. Okay, both the chambers are filled by the blood. Now they have to send it to the ventricle. How they are going to send? Here the oxygenated blood is there. Here oxygenated blood. Here deoxygenated blood. How they should go down? That is what with the help of this wall. Here 3 walls are present. Here 2 walls are present. Correct. So now with the help of these 2 walls, tricuspid and bicuspid wall, the oxygenated and deoxygenated blood will be pumped down to the ventricles. How they are going to pump down? These 2 auricles are there, no? They are going to get contract. So when the auricles get contract, imagine the 2 balloons I have. So now if I just squeeze two balloons, the water, if you have filled in that balloon, how they are going to come down if you just make a hole in that balloon. So like that, here when these chambers, auricles, when they get contract like this, the blood will comes down and settle in the ventricle. So which blood actually? This is oxygenated blood and this is deoxygenated blood. Deoxygenated blood. Now it is filling here. Here deoxygenated blood filled. Here oxygenated blood got filled. Okay, very simple. So this is called atrial systole. So what the term I have given here? See, I have used the term. Listen to me. I have used the term called atrial systole. So atrial systole means atrial contraction. So now they got relaxed. Oh, we have done our work. Now it is responsibility of ventricle to get contract. Now they have hand over it to ventricles. Now ventricles are just seeing how much they are getting filled by the blood. So now entire ventricles have been filled. Now it is the turn of ventricles to get contract. Now ventricle says, okay, we are also going to get contract. Now they are going to get contract. So during their contraction, when they are going to get contract like this, the blood should not go up, right? It should not go back. So that's why here this place will be get closed. That's why we are going to say wall. Wall means what? Here the term wall is responsible for only movement of liquid in one-way direction, not backward. So once it has been come down, the blood cannot go back because they are going to shut their door. So that's why whenever these walls going to shut their door, it's going to give a sound called lub. Okay. So when the doctors are going to check your heartbeat, so you know stethoscope is an instrument. The doctors are going to put in their ears and there is a knob-like structure they are going to put on your chest. What is that? That entire instrument is stethoscope. With the help of that, they could be able to hear your heart sound. Correct? So that sound is nothing but lub-dub, lub-dub. How it is going to come? What do you think of it? It is because of the closure of this wall. Okay. Now ventricles are filled by the blood. Here oxygenated, here deoxygenated blood. Now ventricles are going to contract. So ventricles get contract. Again, the oxygenated blood is moving through this pipe. What is this pipe? Aorta. Correct? Now this aorta is moving to, aorta is coming, it is going, it is carrying the blood to different part of the body. It is carrying blood, that is oxygenated blood, to different part of the body. In fact, this aorta is the largest artery. Artery means what? The artery are nothing but the pipe. They are going to carry oxygenated blood. But this pipe is very largest in the body, therefore it is largest artery. But further, this aorta is going to branch into small artery. That is different. But the structure which is arising from here, no, from the ventricle, which is carrying oxygenated blood, this is called aorta. It is the largest artery, carry oxygenated blood. Okay, so now different organs, different tissues are going to get oxygen. But here carbon dioxide rich blood is there. It should also go outside. Correct. So who is going to carry this yellow color pipe? This is called pulmonary artery. Okay. See, from the pulmonary artery, the deoxygenated blood goes. If you just look at this pulmonary artery, it is going to be get branch like this. So here they are carrying this deoxygenated blood. Here is the carbon dioxide. It says you take carbon dioxide, and after that they will get exchanged with oxygen. Again, this pipe become this pulmonary vein. Okay, so this is nothing but pulmonary artery. So remember what I said just now. Whenever we talk about artery, always in that oxygenated blood goes. Whenever we talk about a vein, always deoxygenated blood or carbon dioxide rich blood goes. But here you are telling different, ma'am. You are telling reverse of this. Here pulmonary vein, you said this pulmonary vein is carrying oxygenated blood. This is carrying oxygenated blood. And this actually artery, but it is carrying the deoxygenated blood. What is this? Correct? Am I making confused? No confusion. Listen, in our entire body, whenever we talk about artery, always oxygenated blood. Whenever we talk about a vein, always deoxygenated blood. But only two exception. Only in the heart, there is an artery, instead of carrying oxygenated blood, carrying deoxygenated blood. This is pulmonary artery. And only one vein, instead of carrying deoxygenated blood, carrying oxygenated blood, that is pulmonary vein. So these are only two blood vessels. It's show exception. They are doing different work, opposite work. Okay. Now, when these ventricles get contract, once again, the blood will be moved up because it has to move in a higher force to the upward direction. So this contraction is called ventricle contraction. Okay. Ventricle contraction is also called ventricle systole. Ventricle systole. So after contracting, again they will get relaxed. So that relaxation condition is also called diastole. It's called diastole. So when they get relaxed, the blood should not come. So therefore, there are another walls are present here. So these walls, they will also shut their door. So that when they get relaxed, the blood should not go back. So that's why when they closes, it will make a sound called dub. So when these wall closes, it make a sound lub. When these aorta as well as pulmonary artery walls closes, it makes sound dub. So lub, dub, like that it makes a sound. So when auricles contract, it is called auricle contraction and get relaxed. That is called auricle relaxation. And ventricles get contract, it is called ventricle contraction. Ventricle get relaxed, that is ventricle relaxation. I repeat. When auricles contract, oxygenated and deoxygenated blood goes to ventricle. Then it will get relaxed. Then ventricles contract, the oxygenated blood goes to aorta, deoxygenated blood goes to pulmonary artery. Then they get relaxed. That relaxation is called diastole. So aorta carrying oxygenated blood to different part and pulmonary artery take this deoxygenated blood towards the lungs. So this is what the mechanism, very simple. So if you observe clearly, here there is no mixing of blood. Oxygenated blood moving separately into the left side of our heart. So here left auricle receive oxygenated blood from the lungs. Again it is jumping into left ventricle. Again it is taking to aorta. So only here the movement of oxygenated blood. So like that here only movement of deoxygenated blood in the right chamber or in the right side. So there is no mixing of blood. Why it is so? Because we people, that is human being or animals, we are complex. So in complex organism, we need more energy. So therefore, to supply more and more oxygen, the mixing of blood should not occur. So therefore, there is a separate chamber for oxygenated blood and deoxygenated blood. Got it? So this is what about mechanism of heart. Hope I am very clear about this also. Okay? Yes. Hope you are writing this in your copybook. Now let me move on to the next part. What is the next part? Types of circulatory pathway. So when you just consider all the organisms, that means all the vertebrates, that is from fish to man, we are going to see three types of circulation. What are those? One is double circulation. Another one is single circulation. And the next one is incomplete double circulation. So if I want to make a list of example, here in single circulation, fish comes under this category to show this. Fish are nothing but the Pisces. So in vertebrate, we can see 5 classes. Pisces, amphibians, reptiles, aves and mammals, right? So reptiles as well as amphibians. Amphibians and reptiles shows incomplete double circulation. Because in incomplete double circulation, these organisms shows 3-chambered heart. And in single circulation, the organism shows heart with 2-chambered, that is 2-chambered heart. But here, for the reptiles, there is an exception. What is that exception? Except crocodile. Because crocodile is having 4-chambered heart. And now, remaining 2 classes are nothing but aves and mammals. So, these aves and mammals, they show 4-chambered heart. And this mechanism is quite complex compared to these two. Let's learn one by one. Very simple, very easy. First, let's move on to double circulation. Just now we have seen, that is what double circulation is. Let me consider this is our heart. Correct? This is our heart. So, there is a separation. Or let me draw like this. So, this is our heart. Okay. Now, here what is happening? The heart which is so carrying the oxygenated blood. This is oxygenated blood carried by our heart. Imagine that will be supplied to tissue. So, this is our tissue. It has been supplied to our tissue. So, what the tissue will do? Tissue says, anyhow, I have carbon dioxide. So, I have carbon dioxide, you have given oxygen, right? So, when I give carbon dioxide, you have to take it. You have to give it to heart only. So, just take this. Now, these vessels become vein. They become vein and they will carry this deoxygenated blood, that is carbon dioxide rich blood, to the heart. Okay. So, this is one part of the circulation. This is one part. So, in this part, what is happening? The interaction is occurring between the heart and the tissue. Correct? So, this is heart and this is the tissue. So, this type of circulation is called systemic circulation. This is systemic circulation. But now, here the story do not end here itself. Again, it has to take this deoxygenated blood to the lungs, right? So, imagine here the lungs are there. Just now what we have studied, the same thing I am repeating here. So, here 2 lungs are present. The 2 lungs are present. Now, this vein is taking the blood towards the lungs. Which blood? That is nothing but deoxygenated blood. Now, it is given to the lungs. But what lungs is? After getting this deoxygenated blood, they give oxygen. Correct? That will be carried to the heart back. So, this blood is oxygenated blood that will be carried to the heart. Correct? So, lungs receive the blood from the heart and give oxygenated blood to the heart. Correct? So, here if you look at the interaction which is occurring between the lungs and the heart. Between the lungs and the heart. So, this type of interaction is called pulmonary circulation. So, this is pulmonary circulation. So, in pulmonary circulation, what is happening? The lungs are getting deoxygenated blood and giving oxygenated blood. And in systemic circulation, what happened? The tissue is getting oxygenated blood and giving deoxygenated blood. And what is the role of heart? Heart is getting 2 types of blood. So, heart is getting blood 2 times. So, heart is getting oxygenated blood also. And from the lungs, the deoxygenated blood also. Are you getting my point? So, as the heart is receiving blood for 2 times, therefore, it is called double circulation. So, this is 1, this is 2nd time. Understood? Double circulation. Pulmonary circulation between lungs and heart, systemic circulation between heart and tissue. Clear? Now, this happens where? In all aves as well as in the mammals and even in crocodile also, because it is showing 4-chamber heart. So, this is 1 chamber, 2 chamber, 1 chamber, 2 chamber, 3 chamber, 4 chamber. So, there is a separation of oxygenated, separation of deoxygenated blood. Now, when it comes to, so hope you are writing in your copybook. So, there is no such diagram. It is there in your textbook, but if you just look at the diagram, you can't understand very clearly. And especially for incomplete double circulation and single circulation, no diagrams are given. So, that's why it is better to write in your copybook. Okay, whatever the things that I am writing there on the board, no, please write down in your copybook. Right now you may not understand. Right now you may not know the importance of the diagram. But at the time of examination, you will recall me. Trust me, you will thank me at that time. Okay? Yes. Now let's move on to the single circulation. What happens here? So the single circulation usually happens in fish. Imagine this is our fish. So in the fish, here is the gill. Here is the gill. And this is the heart of the fish. This is the heart of the fish. Okay. So look at here. The heart is having only two chambers. One is auricle, another one is ventricle. Now blood is coming and giving to the blood which is so going here and given to the gills. So this is our fish gill. This is the gill of the fish. And here the deoxygenated blood is going. Therefore it is called venous heart. This is venous heart which is actually pumping deoxygenated blood. Deoxygenated blood. Now in the gills, as it ingests the water, no, so it will get exchanged with the oxygen. Okay, and now the oxygenated blood will goes from the gills. Now oxygenated blood will goes from the gills to the different part. For example, see here the tissues are there. It will be provided to the tissue, this oxygenated blood, and from the tissue it will take deoxygenated blood and given back to the heart. Just given to the heart. That means heart is receiving blood only one time. I will show you separately here if you are not understanding this. Imagine this is the heart of the fish. This is the heart. What I said, venous heart. Venous heart means which can take only carbon dioxide. Okay. Now, this is what the gill. Now the blood is going from the heart to the gill. Did you understand? Here it will get exchange. Here it says, I am just giving carbon dioxide. Anyhow, you have oxygen. You have oxygen. Now your carbon dioxide give out and oxygen you just get inside. So gill is having oxygen. Now from the gills, the blood vessel will be there, of course, in the gill also. So now these blood vessels which are carrying what? Oxygenated blood. Here oxygenated blood is coming. See, here it is coming. With whom it gave? It gave to tissue. Imagine here is the tissue. So now it has provided this to the tissue. Tissue, you take this oxygenated blood. Now tissue says, okay, anyhow you have given me oxygen, I will give you carbon dioxide, you take it. Now that carbon dioxide rich blood is coming from the tissue and entering into the heart. Okay, so now they are pouring into the heart. So how many times heart is getting blood? Only one time, right? Only one time. So the same carbon dioxide is pumping back to gills. So therefore it is called single circulation. Got it? Now, another type is incomplete double circulation that we are going to see in amphibians and reptiles. So here what happens? These organisms are having 3-chambered heart. Okay? See, look at here. This is right auricle. This is left auricle. And this would be ventricle. So only one ventricle. Let me write here. Only ventricle. So here what happens? See. Right auricle is going to get deoxygenated blood. And this left auricle is going to get oxygenated blood. That means heart is getting blood how many times? 2 times. Therefore double circulation. But it is incomplete. Why incomplete? Because look at here. This is deoxygenated blood which is entering into this ventricle. And this is oxygenated blood that also entering into this ventricle. Let me make this ventricle in different colors so that you can understand. Okay. From this ventricle, there is a structure arises like this. So this structure is actually carrying mixed blood to the tissue. Okay. So imagine here it is carrying towards the tissue. Here is a tissue. So this tissue is carrying mixed blood. Ma'am, does it Won't harm to the tissue? No. There is a special mechanism. It will receive only oxygen, not carbon dioxide. So that carbon dioxide, even though it provides, it won't bother. You just give it back. I will send it to the heart only, so that the heart will pump into the lungs. So it will just give deoxygenated blood to the heart back. So this deoxygenated blood, it is going from here. Again, it will be branched like this, and it will give back to the lungs. It will give to the lungs. Of course, here also interaction occur between the lungs and heart, between the heart and the tissue. Just like in human case, what we have learned in double circulation, but here it is incomplete, somewhat mixed. There we have a separate chamber for oxygenated and deoxygenated, but here everything has been mixed, no? Therefore, it is called incomplete double circulation, because we are seeing the mixed blood in ventricle. So the same mixed blood is running towards different part, towards the tissue also, towards the lungs. Lungs, it will give carbon dioxide. Clear? So hope I am very clear about the circulatory pathway. Now let's move on to another topic. Before going to that, look at this. This is your textbook diagram. This is important for your examination point of view. All this structure already I have explained. Pulmonary artery, which is carrying pulmonary artery is nothing but what? Carrying deoxygenated blood taking towards the lungs. And this is pulmonary vein, left atrium, left ventricle. This is right ventricle. And this is the region, that is septum. Vena cava from lower part of the body, vena cava from upper part of the body. And this is the right atrium and left ventricle. And this is the aorta. Every structure I have explained. And in this diagram, you can see the double circulation they have shown. See, lungs and capillaries. This is heart. This is lungs. So, exchange taking place between heart and lungs. There is pulmonary circulation. They are not mentioned, but it is actually pulmonary circulation. And heart and the tissue, this is systemic circulation. And aorta is actually carrying two different parts of the body. So, aorta is further branched into small artery like this. Okay, and again that will go into vein, and from the body again it goes to vena cava and heart, that is to the lungs. So everything is very clear, I have explained. So please go through your textbook, you will understand. Yes, now let's talk about the last part of human circulatory system, that is nothing but blood vessel. So what are blood vessels? Let me explain. In fact, blood vessels are of three types. Okay, done. I will write here only. Pipes only, but their thickness based on their function, we have given a name. So the one blood vessel which is actually carrying oxygenated blood, we call it as artery. And the blood vessel which carry only deoxygenated blood is called vein. And very, very small pipe which actually exchange the material is called capillary. I know you are getting confused. Let me explain through diagram. Imagine this is our tissue. Tissue means what? It will be having cells, correct? So imagine these are the cells which are present within the tissue. Why these cells are waiting? They are waiting for glucose, they are waiting for oxygen, everything. Now the artery is coming. This is the artery. It is very thick. Artery is made up of 3 layers. Look at this diagram. Look at this. This is the outermost layer. This outermost layer is called tunica adventitia. And next to this tunica adventitia, there is one more layer. Can you see up to here? This much thick. Very, very thick. Very, very thick. This is made up of muscle. This layer is called tunica media. And inside this tunica media, there is another structure. This is inner layer. It is called tunica intima. So total 3 layers are there. Okay, that much thick pipe it is. From here, the blood cells or blood will come out. Okay, so that is what I am drawing here. So this is artery. It is very, very thick. So from such a thick pipe, the material cannot exchange. Of course, here blood is coming. Blood is coming. So in that blood, what are the things we have? We have glucose, we have oxygen, we have so many things we have. But all this material cannot diffuse from this thick pipe because it is made up of 3 layers called artery. This is actually carrying oxygen, no? Therefore, we say oxygenated blood is carried by artery. But we cannot make these cells to get starved like this. We need to provide things to these cells to survive. That's why what artery did, artery made a plan. What it said, I'll do one thing, I will branch into small layer. Small layer means, let me remove this outer layer. Let me remove this middle layer so that I can just show only this inner layer. Are you getting my point? This is outer layer and this is middle layer. And this is inner layer. Correct? Correct. So the blood is moving through this inner layer in artery case. But what capillary did? It has just shut down both outer layer as well as inner layer. Sorry, outer layer as well as middle layer. It is having only this inner layer. Can you see? Only inner layer it is having. So obviously the pipe thickness become very small. Correct? Like this, the arteries are branching into a small tube. Understood? So in this small tube, the oxygenated blood is going. So these small tubes are nothing but what? Our capillaries. Our capillaries. So capillaries are very thin that material can easily diffuse. You said you want oxygen, take oxygen. You want glucose, take glucose. Anything you can give. And now you are giving carbon dioxide. Just give, I will take back. So it will take carbon dioxide. You are also giving carbon dioxide, give. So now all this carbon dioxide will join to these capillary. Now blood become deoxygenated blood. Now small capillaries join like this and become a pipe. Again a big pipe. Again a big pipe. So this pipe where the blood is going along with carbon dioxide is also going. Therefore, this is called vein. Vein. So vein from different part of the body join to become a very big vein. Then it will actually reach us. It will actually reach us to the heart as vena cava. Okay. But here the blood should not go back, right? The carbon dioxide should not go back. Therefore, in between there is actually walls present. So therefore, the speciality of the vein is wall. And here, the blood which is so comes from the heart. Imagine this is our heart. And the blood is coming. When it is pumped by the heart, the blood will comes from the aorta and then enter into the artery with a very high force. Therefore, to withstand such a high force of blood, the arteries are very thick. And capillaries are required for exchange of materials. Therefore, they are very thick. But here, the blood is not coming to the vein. In fact, they are going back to the heart. Arteries are taking the blood from the heart. In fact, veins are taking blood to the heart. So, therefore, here the force will be very less. Force will be very less. But only thing is, it should not come down. No? Therefore, walls are there in between to prevent the backward flow of blood. Because the blood has to move in this direction. It should not go back. That's why. Therefore, here, to withstand for such a high pressure, it is very thick compared to this. How much thickness it is, ma'am? 3 layers will be same to same. Look at this diagram. This diagram, artery over. Look at this. So here one side is artery, one side is vein. Let me write here. This is artery, this is vein. So every structure is same to same. So this is outer layer. This is also outer layer. Outer layer is same thickness. And look at the middle layer. Middle layer is very thick here. And here the middle layer is bit thin compared to artery. Okay, so vein middle layer is, muscle layer is very thin. Because it need more space in fact, but not that much thickness. And of course the inner layer, it is also made up of single layer, single celled layer. This is also single celled layer. So lumen is very broad. Lumen means the space. Here the space is very less. Got the difference between artery and vein? Okay. So now if you look at this diagram, you can understand this is vein. This is artery. See, when they branch together, they will become a very thin pipe called capillaries. Here is the difference. Artery which carry blood away from the heart. Here, capillaries are exchange material between cells and the blood. Just now I have explained. Veins collect the blood from different organs and bring back to the heart. And if you look at the structure, arteries are very thick. It is actually elastic in nature, elastic wall. Here it is very, very thin, only one cell thickness. This is only one cell. One cell means like this, single layer cell. And veins are not as thick as artery, but it has wall. Just now I told. This is what the difference between all these structure. Now let's move on to next topic. Here some information is given about the blood pressure. I will read and explain. Very simple. Yes. The force that blood exerts against the wall of a vessel is called blood pressure. So you might have heard, when your parents go to a doctor, they are going to check their blood pressure, correct? With the help of an instrument, they are going to wrap something around their hand, especially on the upper hand. Then there will be an instrument, something like this, right? So here in this area, the reading will be shown. So mercury will be moving up and down. So that here the digits are showing where the mercury is moving. And here this wrapping, this band will be wrapped so tightly, correct? So this instrument is nothing but, it is known as sphygmomanometer. Okay, I will write here separately. Sphygmomanometer. It's an instrument used to measure the blood pressure. BP. Full form of BP is blood pressure. So you may ask, ma'am, we are healthy, we don't have any blood pressure. Everybody must be having the blood pressure only. If the blood is not having a pressure, how it is going to run? That means how it is going to flow in a vessel, right? Suppose, as we have learned just now, this could be the artery, this could be the vein, this could be the capillary, anything it would be, correct? So blood has to move here, right? Blood has to move. Then only the target materials like glucose, oxygen, everything will be supplied to the cells. So these are the cells. If they are supplying these material to the cells, means they have to flow. So if something is flowing, means they must be having, it must be having pressure, correct? As here the blood is moving, we say that, oh, blood is having pressure. This is nothing but blood pressure. So this blood pressure, it is going to show the variation depending on the type in the sense when the heart is going to pump, when it is going to relax. By the time the pressure get varies. For example, listen here carefully. Before reading, first let me explain. Just now we have came to know that heart is having 4 chambers, correct? Yes. These two are the nothing but left side, left auricle, left ventricle. So like that here, right auricle, right ventricle. So this is our whole heart, right? So when heart get contract, listen to me carefully. This is very easy and very simple, and it is a basic for you guys for your future. So these two chambers are nothing but auricles. Either it is a left auricle, right auricle, whatever it is. But the auricle has to get contract. Then only blood, that is the deoxygenated blood will come here. The oxygenated blood comes down here to the ventricles. When the auricles get contract, no? So that contraction, the term contraction is also spelled or pronounced by a term called systole. Systole means nothing but contraction. If the auricles contract, we say that auricle systole. Like that, when the blood fill in this chamber, then they will also get contract. These two are nothing but the ventricles. When ventricle contracts, we say that the ventricle systole. Okay, so here auricular systole, like that ventricular systole. Now tell me, when the blood goes to the vessels in the sense these pulmonary artery as well as the veins. So this is nothing but pulmonary vein. This is nothing but aorta. Aorta is also artery, I said. When the blood is going to move up, when the ventricles get contract. So when ventricles get contract with the pressure, the blood will rush up into these vessels with a high force. Okay, so that we call it as systolic pressure. As the blood is moving, Because of the contraction, we call it as systolic pressure. Now tell me, whether the systolic pressure is having high or low rate. Obviously high rate. When you push the water, okay, so obviously it moves in a high speed. Suppose if you are not pushing anything, then obviously it will go in a normal speed. Correct? So like that, after contraction, it must have to get relaxed. The heart has to get relaxed. When it get relaxed, okay, the term relax is also going to be called as diastole. Remember, these terms are very important. Systole means contraction, diastole means relaxation. When it get relaxed, so nobody is there to push the blood, so blood is moving in a normal speed. Then it will show the different reading. So when somebody is pushing the blood, who is pushing? This ventricle is pushing, that is systole occur, the blood moves in a high speed. I am standing here, I am a blood. If somebody is pushing from my backside, then obviously I will run fast. Correct? My pressure will be more. If nobody is pushing, means I will move in a normal speed. Correct? So that normal pressure is diastolic pressure. When somebody pushes, no, that is systolic, that is called systolic pressure. Okay? Now let me read this point. Blood pressure you have understood. This pressure is much greater in arteries than veins. Why it is greater in arteries? Because the artery is the one, it has to supply blood to all parts. Correct? If it is not moving in a high pressure, then how come throughout our body the oxygen glucose will be supplied? Correct? So it has to move in a high pressure. Therefore, it is always higher in arteries than in veins. Because vein is having nothing. It has to collect the things like carbon dioxide and waste material. It has to just move. That's it. The pressure of the blood inside the artery during the ventricular systole, that is ventricular contraction, just now I told no, is called a systolic pressure, and the pressure in artery during the ventricular systole, diastole, that is nothing but relaxation, is called diastolic pressure. Okay, so we are just measuring the ventricle part, because whenever the blood is moving to the artery with the pressure or without pressure, the contribution is by whom? By the ventricles only. Because even auricles are going to put a systole. That means even though they are contracting, the blood is not moving to a different part of the body. When auricles get contract, it will just move down to the ventricle. That's why we won't consider these two for measuring blood pressure. Only ventricle part. Remember. The normal systolic pressure is 120. Now if you measure my blood pressure, okay, when my heart pushes the blood, the reading will be shown here, that is at 120. Okay, so it is 120 millimeter Hg. Millimeter Hg is mercuric value. Okay, so when the heart is in a relaxed state, ventricles are in a relaxed state, my blood is moving in a normal pressure, that pressure must be 80. Okay, that's why we say 120 by 80 is nothing but normal blood pressure of a person, healthy person. Okay, it is systolic by diastolic pressure they are measuring. The blood pressure is measured with the help of instruments, sphygmomanometer. I said high blood pressure is also called hypertension. Suppose if a person, if his body condition is not like this, so he's having lot of tension, so much of problem, so his heart will be beating very fast, the contraction force will be very high. So that, suppose imagine if the systolic pressure is 130 or 140, then diastolic pressure is 90 or something, then we say that this person is having high BP. BP will be there in everybody. In my case also BP is there, your case also BP is there, everybody case the BP, blood pressure must be there. But the blood is moving in a low pressure or in a high pressure. For that we are going to measure the BP. Okay, so if he's having more systolic and diastolic pressure, we call it as high BP. High BP is also called hypertension. Okay. Why it is caused? It is caused by the constriction of arterioles, because arterioles become so narrow in such cases. When it becomes so narrow, arterioles means they are talking about capillaries. So blood has to move in a very high speed. So they have to struggle a lot in order to move, such a narrow range. Okay, imagine if the pipe is very wider, the blood can easily move, right? Suppose if the pipe becomes so narrow like this. Here, look at the space between here, two line and here. So by the time the blood, if it is rush in this place, it has to move in a high speed, right? That is what they are telling. When the arterioles get constrict, arterioles means capillary, which results in increased resistance to the blood flow. So blood will face lot of resistance. and it can lead to rupture of artery. So here internal bleeding occur, artery may get ruptured. So that leads to this hypertension and so many side effects will be there. So that's why the doctors are going to give a medicine to such people so that their arterioles should get dilate. Dilate means it should get wide enough so that everything should go normal. Okay. So here the main thing what you have to focus is systolic pressure, diastolic pressure. There is ventricular systole, ventricular diastole, and their value 120 as well as 80. And if the value increases, we call it as hypertension. And sphygmomanometer, it's an instrument that will be used to measure the BP. So such question may appear in your MCQ. Please make a note of all those things. Right? Done. Now, let's move on to the next topic, platelets. What I said, when you just take a blood, the blood is having two parts. Though it is a liquid, but pure liquid part is what? Plasma. And within this liquid, I said there are cellular part. So these cells are nothing but RBC, WBC, as well as the platelets. Correct? Everybody know about this. What is the function of RBC? If I ask the question, now you can able to answer. Ma'am, it is very simple, ma'am. RBC are like a boat. So that whatever the oxygen that we are going to inhale, that will be present on this hemoglobin. So RBC, just like a boat, it will run in the blood, it will reach towards the tissue. So the main function of the RBC is to carry oxygen, right? And now, what is the function of WBC? So just imagine if somebody sneezes in front of you, or okay, let's forget that example. Let's take something else. As I'm standing here in this room, don't you think the bacteria are here? Of course there. And in my hand, the bacteria are there or not? Of course it is there. And sometimes if anybody brings some sweets towards me, and if I just take the sweet without washing my hand, all of a sudden or within 1 or 2 days, am I going to fall ill? No. So in our daily situation, so many things we are going to do knowingly or unknowingly. Correct? In all these situations, the bacteria are going to enter in our body. The germs are going to get entered in our body. Who is going to protect from all those pathogens? Pathogens means these virus, bacteria, these are foreign agents, no, which do not require to our body. We consider them as pathogen because they are going to cause disease. But every time you won't fall ill. In very severe cases, unfortunately, our body unable to withstand for such a bacteria or a virus, then only we are going to go to doctor, we are going to take medicines. Correct? But rest of the time, we are healthy. How it is possible? Though we are facing a lot of pathogen in our daily life, but still we are healthy means somebody is there in our body, somebody is there inside our body to protect us, just like a soldier, how they are going to stand in a border to protect our country. Correct? So those soldiers are nothing but WBC. So WBC's main function is to give immune. It is for immune system. So you might have heard the term immunity. Immunity means it is nothing but resistance power against the disease. That comes because of this WBC. Then what is the function of platelets now? Platelets' main function is for blood clotting. It is responsible for clotting of blood. So why blood has to clot? Blood is moving so freely. Let it get moved. Why it has to get clot? If it get clot, it is a problem, no? Yes, if it is get clot within the arteries, it is a problem. But accidentally, if this artery or a vessel get break or it get ruptured, then the problem will become serious, right? Imagine I am just going on a road in a barefoot. I am just walking, okay? And unknowingly, I have just put my step on a glass piece. Then what happened? The foot, the vessel which is so present on my foot, that will get ruptured, right? Imagine this is a blood vessel of my foot. So here it is made up of cells like this. Consider this is one blood vessel. It could be artery, it could be vein, it could be anything, but it is a blood vessel. Okay. So this blood vessel consists of cells like this. Now inside blood is moving. So let's assume these are RBC. And here these are platelets. Here and there they are moving. And these are white colored WBC. So everybody is moving in this blood. Now all of a sudden, Because of the activity, what I did unknowingly, because of that, my blood vessel got ruptured like this. It got damaged. Correct? Now what happened? Tell me. So when it gets ruptured, obviously we just made a way for these blood cells to move out, right? So they start flowing out like this. That's why the bleeding occurs. When we get hurt, when we get injured, the bleeding occurs because the vessels will get damaged. But after some time, this process will not continue throughout the day, or this process will not continue for 2-3 hours. After some time, after a few minutes, it will just block, correct? Some kind of brownish color substance will be formed in the area where we got injured. What is that brownish color substance? That is nothing but blood clotting, okay? So here what happened during this process, immediately these platelets are going to get a signal. So what type of signal? Okay, our artery, veins or anything, blood vessel has got ruptured, so we need to block our blood. So immediately it will rush towards the damaged area and start clumping like this. But still the blood keep moving. So they are unable to stop. So by that time, so many reactions occur inside our blood so that the fibers will be present like this. Remember, there are certain fibers. So these are the fibers. We call it as fibrinogen. So they are present in an inactive state. Because of the reaction, so many reactions, the instruction will come to the fibrinogen so that it have to become active form. So that active form we call it as a fibrin. So they will get converted into active form of fibrin. And immediately they will come towards the ruptured area. Ruptured means the damaged area. They block like this. Just like a mesh. Just like a mesh. And on this mesh, these platelets will also be just get blocked in this way. So totally everything has been blocked by these fibrinogen as well as by the platelets. Now there is no chance of leaking the blood. Correct? The leakage do not occur, the bleeding do not occur. So the blood is going to get clot there itself. And gradually it is going to get healed and everything will be normal after a few days. But for that moment, the blood has to get clot. It's a main duty of our cells. So who is going to do that? That is by platelets. So that is what the mechanism of the platelets. Understood? So this much information is enough for you. Now let's move on to the last topic called lymph. It is quite confusing for many students. If you read your textbook, you start feeling, ayo, what is this? What I have read? I'm not understanding anything. Because in a sentence form or in a text form, what you are going to read in your textbook, no, even though every information is given in our textbook, but we are unable to imagine the things, how it is exactly happening in our body. So the information what I am giving now, right now, this will be retained in your memory throughout your life. But only thing is, please do focus. Okay? Done. Imagine I have a cell here. Imagine I have a tissue. This is our tissue. Just now we have studied everything. So if I say tissue, obviously the tissue must be having what? Cells. So these are the cells present. Tissue are made up of cells, right? Okay. So now the cells are waiting for what? Glucose and oxygen. Who is going to bring it? That is by artery. Imagine this is the artery. So I have shown everything just now. This is artery. It is bringing everything. But artery cannot supply because it is very thick. That's why it is going to get branch like this. What is this branch we call? Capillary. So in this capillary also the blood is moving. The blood is moving. Blood is having what? Plasma, cellular material along with this dissolved material like glucose is there, oxygen is there, minerals are there, proteins are there, WBC is there, RBC is there, everything is there. Everything is there. Now, the material which is so present inside the blood, except RBC. RBC says, I am very bigger in size. Imagine this is the blood vessel. That means I am talking about the capillary. Imagine this is the capillary. So let's assume this capillary is having small pores like this. Small opening, small pores. So through these pores, the material which is so flowing in the blood, they start getting diffused. Are you getting? They started moving like this. So that is what the thing happening here. They started come and collecting here around the cells. So here, the things which is so collecting, that is in the form of a liquid, we call it as tissue fluid. Tissue fluid. Okay. As it is present outside the cell, right? Not inside the cell, outside the cell it is present. These are cells. So these are cells. And the fluid is present outside the cell. Therefore, we also call it as extracellular fluid. In our textbook, they have mentioned this term extracellular fluid. Now, in this extracellular fluid, what are the materials are coming and collecting? Your glucose is there. I am writing here for our convenience. Our glucose has been dissolved. Oxygen has been dissolved. Many minerals have been dissolved. And even some of the WBC came by escaping. But not our protein. But not our RBC. RBC retained within the blood itself. Some of the protein which are bigger in size retained within the blood itself. Because they says, oh, we are bigger in size, we can't diffuse. Imagine there is a small gap. Very small gap is there in the wall. If I have to move, if my size is very big, if I am very broader, then how can I pass? The person who is very lean, who could able to pass, but not me. Let's assume, if a small kid could able to cross that area, that a small place where the width is very small, this is because the girl or a boy, the kid is very small. But it is not for an elder like me. Correct. So like that here also RBC proteins are very bigger in size. They cannot cross this small area. Okay. But everything can be dissolved here except those RBC and WBC. So they will be as such how they are present in the blood plasma, like that only they are present within the tissue fluid. Correct. So that we call it as an extracellular fluid. Now what they are doing, so the glucose is coming down, the oxygen is coming down, they are going to deliver the things to the cells. Because the main function is to make these cells to get survive. Correct. And after that, they are going to collect the carbon dioxide, the waste material, which have been produced from the cell itself. So finally, they will add up into the blood only and they become vein, I told. So they only become vein. So these vein, they started joining together and they move towards the heart. So this much we have understood, correct? This much we have understood. Now, if the tissue fluid, which is so collecting here within the tissue, if it start accumulating more and more, then the organ or the tissue start bulging. That should not happen in our body. Therefore, this is colorless, right? That's why I have mentioned in a white color. Now, that's why in order to collect the extracellular fluid, there are certain vessels. Vessels in the sense, pipe-like structure, like this. They have been attached to the tissue. Can you understand what I am trying to tell? So these are the pipe. So in this pipe, this extracellular fluid only going. That is only going. So the same tissue fluid as it is moving through this pipe, we call it as lymph. Did you understand what I am telling? So this fluid itself, we call it as a lymph. Why? Because now these tubes, I can give a name called lymph capillaries. Capillaries. Okay, and these small lymph capillaries, they join like this. They join like this. They join to become a big vessel. So that in that vessel also the same lymph is moving. So this is called lymph vessel. This is called lymph vessel. And this lymph vessel is also carrying the same lymph fluid. Now again going to be, see, look at here the funny things. What they are doing, they are going to dump the same thing into the vein itself. Now the question is, ma'am, why they have to do such a drama? Instead of this, they would have sent here back into the veins itself, no? Why they are going here? This is because they have a strategy. What type of strategy? Here, imagine there is something is there. Just like a checkpoint. So this checkpoint, we are going to call by a name called lymph node. So in the lymph node, so many police are standing. Imagine he's a police. So he's an investigating officer. What is investigating? So let me take an example. Imagine you are in a place or in a village. You are a lab technician. You want to check the quality of the water of that village. So you are going to bring some water as a sample. Correct? Now with the help of microscope, you are going to check whether the water is good for drinking or not. Everything you are going to analyze. Okay, and after that, what you are going to do? If it is good, you are going to pour back that water into the river only, because after checking, no use. Okay, you said to the villagers, okay, this water is very good for drinking, you can use it. Suppose if that water is having any bacteria, pathogen, then what you are going to do? Immediately you are going to give signal to the villagers, don't drink that water, it is not fit for drinking. So we need to treat that water. So you start taking action, you are going to treat that water so that you will make that water to be fit for drinking. Correct? So just like that, it is also like a lab. Okay, so in that lab, there are certain cells are present. Why these cells are present? By chance, in the blood, if any pathogen has entered. So to avoid confusion, I am rubbing this glucose under. Now you have understood this concept. Okay. Now imagine if some culprit has entered. Let me take which color, man? Okay, this color I am going to take. Done. Now this fellow is a bacteria. Now he has entered into this blood and through the capillaries along with the diffusion. So this fellow also got diffused because the glucose is coming. So he want to have glucose. So he is also running behind that glucose. Now the tissue fluid, when they are collecting in the lymph capillaries, this bacteria has also entered here. Now this bacteria is also moving in this lymph. And now when it comes near the lymph node, now they are going to encounter by these cells. These cells are nothing but WBC only. These are WBC. Now these WBC came to know that, okay, some bad man has been entered into our family, that is in the blood, we need to take action. So if 1 or 2 is coming means, let's imagine how many friends are there in the blood. Half is bacteria. Okay, so all are actually spoiling our blood, we need to kill them. So immediately they start producing a kind of substance called antibodies. So hope you know that antibodies. So these antibodies are again enter into our blood. They start killing, they start attacking these bacteria. So these 1 or 2 bacteria will be killed by these cells only. But there are plenty of bacteria will be there in the blood. No, they are simply roaming by escaping. They will be get killed by these antibodies. So that is what the purpose. These are antibodies. That is one thing. And the second thing is, if fluid is present, imagine if there is 100 ml, but only 70 ml of fluid is enough for the tissue. Then rest 30 ml will be taken back. It should not be retained in the tissue. It will be sent again back into the blood. If it is retained within the tissue will get bulged due to excess of water. So like that, so many functions will be done by our lymph. So hope I am very clear. Hope you are writing all these things in your copybook without any confusion. Please don't get confused. If you want to rewatch it again, just like a revision, as I have mentioned in the beginning itself, you can use the timestamp method. Okay, just go to that particular topic, just click on the time. Directly you will be taken to that particular topic, you can watch it. Okay, so it is the best method of doing a quick revision. Done. Yes, here I have mentioned the points of lymph. One is it drains excess of fluid from extracellular part and back into the space, that is into the blood. Okay, and another thing is fat transport. The fat which is unable to dissolve in the blood, that will be transmitted through the lymph only, and of course for the immune system. Okay, now the last part of the transportation, that is transporting plants. It is very simple, very easy to understand. Within 5 minutes we will understand. Transportation in plants, it is not so complex. It is not that much lot of cells, lot of functions. It is not having that much. Why not such a complexity? Because lot of energy is not produced in a plant body. How it is going to be happen in human case or in animal case? Why? Because in a plant body, majority of the tissues are dead tissues. If I just consider this plant, imagine this is a plant and these are the roots of the plant and this is the leaf. And if I talk about the bark are dead part. So like that, within the cell also there are so many dead tissues are present, and within the root also so many tissues are dead. Only few are living, majority are dead tissues. So for those dead tissues, nutrition is not required, energy is not required. So why it has to spend a lot of energy for transportation for these dead cells? So therefore, the plant's main target is to supply energy and nutrition only to the living parts. Okay, so that's why the system is not that much complex. Now, let me come back to the. So now imagine, we have studied in the beginning itself, the leaves are like a kitchen. These are the leaves. Leaves are like a kitchen of the plant. Why kitchen? Because it is going to utilize the raw materials like carbon dioxide and water, right? And with the help of photosynthesis, they are going to produce glucose and oxygen. So this is nutrition part we have learned. And this glucose combine with oxygen and produce energy. So that energy will be in the form of ATP. During this process, carbon dioxide is released. So this is respiration. So this thing is going to happen in the leaves. So leaves are having energy. Indirectly I can say, so leaves can have energy because the photosynthesis taking place. Correct? But I said, they use this raw material, carbon dioxide and water. Carbon dioxide will be supplied through stomata. Then what about the water? And even if they want to repair their body parts, they need so many other material like nitrogen they require, because in a protein or in amino acid, nitrogen must be there. And so many other molecules, minerals are required. Where they are present? Of course, they are present in the soil. If you want a nitrogen, nitrogen is there. If you want the minerals, minerals are there here. So many. Water will be there in the root. So it has to get absorbed from the soil itself, no? So root is the part, they are going to supply these things. Correct? Now the question is, the energy, though it is produced within the leaf, will not be restricted to this part. It has to transmit it to different part of the plant body. Because every plant body need energy. Right? So in our case also the same thing has happened. Though the blood, that is digestive system, is going to break the food, the energy is not restricted there only. The blood was the medium which is taking to every organ, that is to every corner of our body. So like that here must be, somebody must be there to carry this energy from leaf to different part. So like that, somebody must be there to carry these minerals to different part up to the leaves. So if the plant is very simple, then this process could happen with a step called diffusion. Diffusion already we have learned in class 9th. Movement of molecules, higher concentration to lower concentration. But here the plant is very, very huge. Diffusion cannot compensate all these things. So for that purpose, we need somebody to conduct the materials. So such a tissue we call it as what? Conducting tissue. We call it as conducting tissue. So conducting tissues are of two types in plant cases. What are those? One is xylem and another one is phloem. So we have learnt in class 9th itself, xylem and phloem. Just assume that we are just doing a revision of this. Okay. So xylem is called water conducting tissue. It is a complex tissue. Both are complex. Why complex? Whenever we talk about a tissue, it is made up of only one cell. But here, though it is one tissue, it is made up of 4 different cells. That's why we say it is a complex tissue. Even phloem is also complex tissue because it is also made up of 4 different cells. But here this phloem structure is carrying or transport the food. Therefore, it is called food conducting tissue. This much is clear. So if I just go towards the xylem. So what are all the different cells present here? 4 cells I told. The 4 cells are called xylem vessels, xylem tracheids, or let me just mention it as a tracheit. And another one is xylem parenchyma. And last one is xylem fiber. Look at here. Out of 4, only parenchyma, xylem parenchyma is a living tissue. And the rest of the things, this tracheit, vessels, these are all dead tissue. It's dead. This is also dead. This is also dead. Then you may ask, why it has to present, ma'am, if it is a dead? Imagine you have a full water in your over tank. So it has to supply to our kitchen, it has to supply to our bathroom, it has to supply to our utility everywhere. So you are going to put a pipe connection so that whenever you put a switch, the water will come through a pipe. Then the pipe is a living? No. Why pipe you have kept? Even though it is a non-living, it is made up of plastic. This is because it's a channel. Correct. Through that tube, the water is coming. It is just giving us support, ma'am. That's why we have kept. Here also, my dears, these tubes, they will be in the form of a tube. They are just giving support. Okay. So, if you just look at the xylem vessel, it is very broader like this. And this portion, this broader portion is the one through which the water enter. And here also small pores will be there. But this xylem is very broader. But when you just look at the trachea is somewhat narrow like this. It will be narrow. Here it will be not having any lumen, but side by side it will be having pores. We call it as a pits. If the water has to enter, it will enter through these pits. But here the water enter directly through this. And these parenchyma, they are just a living. If it is having any food material, it will be stored. And the xylem fiber, it is like a fiber. They are just giving a support to this. That's it. Okay. Now these vessel, tracheids, everything, they will be present together. They are not present separately. I will show this image. You can understand. Look at this. Can you see? If you just cut a plant in a cross section, how we are going to see the structure? The same structure I have brought. Okay. So this is the vessel. Below this, a big pipe is there. Look at here, a big pipe. The cross section of this pipe I have taken, this is a vessel. And here small tube-like structures are there. No, these are vessels. Can you see this? Sorry, tracheids, and this is the vessel. And side by side only, they have not actually mentioned, the fibers will be there to give a additional support. And here certain cells will be there. Let me show it in a red color. Cells will be there to store the molecules. It is parenchyma. So, they are present something like this, one after other. If this is one xylem vessel, below this only another xylem vessel will be there. Below this another xylem vessel will be there. So like that they are going to present one below the other. On either side they will be having tracheids like this. Here also. And this is parenchymatous cells. These are living. How you came to know it is living? Because it is having cytoplasm, nucleus, everything. But these two do not have. And on this side itself, the fiber will be present. So like that, they are present together. And whenever the water is moving means, it will be moving upward like this. Always it move upward because it has to move. It has to absorb the water from the root itself. No, always it move upward towards the plant. Okay. So this is what the xylem. So when it comes to the function of the xylem, let me move to the xylem function. Its main function is to transport water as well as the material which are so dissolved in the water that will be transmitted. How they are going to transmit? Listen to me. Imagine this is a plant and this is the leaf. So I need to supply the water to the leaf. Okay. So here is the root. Here is the root. Imagine these vessels are present in this way. So it is understood that side tracheids are there, parenchyma, fiber, everything is there, but I am just showing only the vessels part. So now it will be present throughout the leaf, throughout the part. Correct? So in the leaf, what happened? We all know that the stomata will be present in the leaf. So through this stomata, the water will be get lost, right? The water will be lost. That process we call it as what? Transpiration. So when the water keep getting lost from here, this portion will start getting dry. Correct? The concentration gradient created. So it become lower concentration and water in the bottom part become higher concentration. So now the water molecules start moving from higher, this is higher concentration, to lower concentration. Lower concentration means upward. But it is against the gravity. So we know that if any molecules are moving, that should go below, that means downward only, because it has to follow the gravitation. But this is against the gravity. So how this process is going to carry? Let's take a simple example. Imagine you have a juice. So to take the juice, we have provided a straw. Okay. So with the help of a straw, you are just sucking the juice. You are drinking this. So what you are going to do? You are going to put a pressure, right? You are going to suck this liquid. That force we call it as a suction force. So here also the same suction force is applying. So because of this force, the water molecules are start pulling each other. So when they start pulling each other, obviously they start moving like this. So as the water is transmitting here in the form of transpiration, So water molecules are coming up in order to replace that lost water. So like this, the water will be conducted throughout the plant body. That is from root to different part. Understood? So xylem vessels and tracheids of root, stem, leaves are interconnected. Okay, so water loss through stomata, that we call it as a transpiration, and it creates a suction, not free, suction force, and pulls the water. That's it. So water conducting tissue we have understood. Then what is food conducting? So food conduction occurs with the help of phloem as well. So here also phloem is having 4 components, just like xylem. What is that? One is sieve tube. So this sieve tube is a dead tissue. Another one is companion cell. Then again phloem parenchyma and phloem fibers. One thing, it's not a dead tissue, it is also living. Sieve tube is living, companion cell is living, parenchyma is living. All these 3 are living. So only fibers are dead. But there in the vessels, xylem, 3 are dead, only 1 was living. Therefore, xylem itself is a dead tissue. Phloem is a living tissue. So if I say sieve tube, how it will be present? Like this. Look at this diagram. I will write separately here. Look at this. Here they have mentioned sieve tube, right? So if I just draw one sieve tube, it somewhat look like this. It's a tube-like structure only. Here, we have seen your parents, your mom is going to sieve the material with the help of a sieve plate, right? So like that here also small pores will be there. It looks like a plate only. But this entire tube, it will be having channels like this. The pores will be opened here down. Understood? So as it is in the form of a tube, we call it as sieve tube. And these plates are called sieve plate. So to this sieve tube and sieve plate, the food which have been manufactured in the leaf, that will be transmitted. So these tubes will be interconnected. Again, this tube will be interconnected like this with another tube. But it says I am a living. But if you want to say some cells are living, it must have nucleus. But this cell is not having nucleus. So how you can say living? Therefore, one of his close friend will come. Therefore, we call it as a companion. So companion. Oh, he is my companion. She is my companion. We are going to say like that, no? So like that, his close buddy is the companion cell. So this companion cell is present along with this. So he is going to say, we will share our nucleus together. So this nucleus is for me and for you also. So like that, generous it is. Look at here, the companion cell is present on either side. The same diagram I have written, companion cell. So companion cell is to give a support to this sieve tube. And obviously here, the parenchyma will be there. And these are the fibers. So like that, it will be present on other side also. Okay. So now imagine this entire plant. This is the entire plant. Okay, now we have understood. Now imagine here is the leaf. So the leaf has prepared the food. So the food will be stored in the form of what? Starch. But this starch says, this phloem says, if you are in the form of a starch, I can't able to transmit. I can't able to transport. So I will give some energy, or you can utilize some energy. That energy is nothing but ATP. So with the help of ATP, you just convert yourself into sucrose form. So now when it get converted into sucrose form, imagine this is the sucrose. So when it get converted like that, so this sucrose will be transmitted through this sieve tube, and that will be supplied to all parts. Imagine these starch or the sucrose will be present in the vegetable, or it is present in the where that is in the leaf, or it may be present in the fruit, anything. Now it will be get converted and it will be supplied to growing parts. So leaf is growing, so it will be supplied to leaf. So some bud is growing, it will be supplied to bud. So like that, it will be supplied to all growing parts. So this overall process of food transportation, we call it as translocation. Okay, what is the term? Translocation. Translocation is nothing but transportation transport of food. But here it is not single direction. If it has to move from here to here, it can go in this direction also, downward direction. Suppose if the food are there in the root, imagine carrot, beetroot, like that, the food is there. So it can go up also. So therefore, the movement of food through phloem, it is bidirectional, remember. But movement of water is always unidirectional. It is always from root to the leaf, not from leaf to root. We won't say, oh, water is coming towards the root. No, water is actually absorbed from the root towards the different part. Therefore, it is always unidirectional. This one is bidirectional. Clear? Yes, transport of water over transport of food. Transport of soluble products of photosynthesis. Photosynthesis means that glucose, starch, everything is called translocation. It occurs through phloem. The substance delivered from storage organ like roots, fruits, seeds to growing part. Substance move in both upward and downward direction. Therefore, it is called bidirectional. Okay, clear. So, hope I am very clear about the transportation in plants also. Now, let's move on to the last topic of this chapter. Okay, it is very small topic. We will finish it faster. That is excretion. Okay, now let's talk about the last topic of this chapter, that is nothing but excretion. So what is excretion? We all know that removal of waste material from the body, correct? So whenever we consider the waste material, what are all the waste material generated in our body? Yes, the digested waste. So digested waste is nothing but fecal matter or feces. It is ejected out through anus, correct? So this is undigested waste. After digestion, what are all the things which are considered as undigested that will be ejected? So, in general, we are going to say that digested waste, or let me mention it as undigested. So, like that, respiratory waste. So, during respiration, the carbon dioxide will be considered as the waste molecule. It's a waste gas that will be released out through lungs. Correct? So, these are the waste material. So, like that, in our cell, imagine this is our cell. So many metabolic reactions are taking place. Let me write it as a cell. So metabolic reaction means, as I have explained earlier itself, it can be anabolic or it can be catabolic. Imagine a material called A combines with B so that it is going to produce a chemical called C and D. So out of these C and D, let's consider the C is useful material whereas D is not. Then of course the D is the waste material, right? It's a waste. So this waste is considered as nitrogenous waste. Why nitrogenous waste? Because in this waste, the nitrogen is present, that's why. So the nitrogenous waste can be classified into three categories. One is ammonia. The second one is urea and the third one is uric acid. So in human being case or in mammals case, if ammonia is produced, but immediately it will convert it into what? It will get converted into urea. Because for excretion of ammonia, more and more water is required. But we are not going to drink such a 10-20 liters of water, right? In order to excrete this ammonia, hardly we are going to drink around 2 to 3 liters. So that's why such a ammonia or such a waste are going to be produced in animals where they are going to live in water itself. That is in aquatic animals. Whereas in normal human being, that means in the mammals, human beings, as well as in the reptiles, urea and uric acid are the nitrogenous waste. Okay. Now, whenever we are talking about the excretion, our main focus is only to excrete this nitrogenous waste. Understood? So, different organisms are going to excrete such waste in different manner. Say for example, if I take aquatic animal like fish, they are going to excrete through a process called diffusion. Or if I take an organism like amoeba, so whatever be the excretory product, how it is going to excrete? It is through body surface, once again by diffusion. So like that, all organisms have their own excretory organs or excretory structure. Right. So here in this chapter, we are not discussing about other animals. We are mainly focusing on human being. Okay, the excretion in human being. Let us talk about that. So hope this part is clear for you. Now let's talk about the excretory system in human being. You are studying about excretory system since your lower classes. That is since your 7th and 8th standard itself, I think. So need not to explain much. But there are certain things, there are certain terms which are new to you. Okay, so for such thing you have to focus. Understood? And like that, from the beginning, I have given information related to all these topics like nutrition, as well as the transportation, everything, respiration. So many information I have given. Hope you have written all those information in your copybook. Don't neglect anything, because it is very useful. It is very helpful for your future also. If you are really liking the video, if you are really feeling interested in the chapter, then what you are waiting for? Just hit a like button and do share it with your friends. Okay? Yes, let's talk about the excretory system of human being. So what does the excretory system consist of? So we all know that pair of kidneys are the main excretory structures. So kidneys are actually reddish in color. They are bean shaped and reddish in color. Okay. Then from this kidney, a tube-like structure arises like this. This is one tube, this is another tube. So these two tubes are called ureter. And finally, they will come and join to a bag-like structure. So this bag is called urinary bladder, and finally this is called urethra. So this is what the excretory system. Okay, let me mark it. You have to be very conscious. Here I have mentioned pair, correct? So pair of. Whereas here only one urinary bladder, only one urethra. Whereas two kidneys and two ureter. So this is the part of our excretory system. But when you just look at your textbook, the diagram, in this diagram, they have mentioned these two things also, right? Something like this. And something like this. Correct? So they have mentioned it as renal artery and renal vein. So this one, let's assume this is the renal vein and this is the renal artery. Now the question for many students that, ma'am, these two structures are also a part of excretory system? No, my dears. Just look at the term. Here they have mentioned vein. Here they have mentioned the term artery. So whenever we are going to see the terms like vein or artery, that is a part of what? It's a part of circulatory system. So how come it could be a part of excretory system? Then why they have mentioned here? Why they have showing this part in this diagram? That is what the question. I will explain, don't worry. But hope this much is clear for you, right? Okay, let me explain that part. So before going to this, first, let us see where exactly our kidneys are located. See here, the person is turning back. That is, it is the back side of this person. This is the vertebral column, correct? And look at here. On either side of the vertebra, the kidneys are located. Here they have mentioned 12th rib. Why 12th rib? You are going to study in your higher classes. From the vertebra itself, the ribs are going to be arise like this. So there are around 12 pairs of ribs are present. The 11th pair as well as the 12th pair are going to protect our kidneys, which are actually present in the back side. The ribs, which are actually present in the front portion, okay? But they arise from the back. They arise from the vertebral column and they are coming towards the ventral side. This is the ventral portion of a person. This is the dorsal side, okay? So they arise from the dorsal and coming to the ventral, facing towards the ventral, okay? So here the 11th and 12th also, they are just present like this. They are present like this, and inside our kidneys are located. Okay. So even if you are not understanding, don't bother. You just know the kidneys are present on either side of the backbone. So rest of the things you will learn in your higher classes. Okay. Now, here I said that I will tell you about this renal vein and renal artery. I will tell you what is this exactly. So now this structure is very clear. This is our excretory system. Now let's imagine this is the heart. This is the heart. From the heart, aorta is going to arise. That everybody knows. Correct? So this aorta is branched into many arteries. Correct? Because it has to supply the oxygenated blood to all organs of our body. So now let's assume one of the artery is coming down like this. So this is what the artery which is coming down, which is carrying what? Oxygenated blood. So from this artery, the branches arises and they are going to be joined to the kidney. So what they are supplying actually? They are supplying the oxygen to the kidney. Correct? So this is what the thing they have mentioned in the diagram. So the upper portion they have not shown. They have just shown only this part, that is renal artery. Okay? But actually they are arises from the aorta branching itself. Okay? They are supplying the oxygenated blood. Okay? Now, after supplying oxygenated blood, let me mark this first. This is renal artery. Okay? So the branches are supplying oxygen to the kidneys because kidney should also respire. Correct? And after supplying oxygen to the kidney, what kidneys are going to give in return? Yes, carbon dioxide. So this carbon dioxide must be carried by veins only, right? So imagine here the veins are arising. Here one vein is arising, here also vein is arising. So these veins decided, okay, let us join together like this. So they are joining. They are joining here. And finally, small veins are going to be joined to the vena cava, and finally the deoxygenated blood will be given to the heart. Okay? But this part which is so present above the kidney, which have been connected to the heart, those things have not mentioned. But it is understood that if I am talking about the renal vein, it is actually a part of circulatory system. If I am talking about the renal artery, it is a part of circulatory system. So that's why I have mentioned this part. Okay. So these things are also very clear. Now, if I just take out a kidney, if I just consider a kidney is an organ. Correct? So kidney is our organ. It's a filtrating organ. What it has to filter? It has to filter our blood. Okay. But within kidney, there are so many cells are present. Small cells are present. Those cells are considered as structural and functional unit of kidney. They are nothing but a nephron. Okay. Each kidney consists structural and functional unit called nephron. So around millions of nephrons are present in each kidney. Now let us talk about how this nephron looks. Okay, now let's talk about the structure of nephron. If you are liking the video, please do subscribe to our channel also. Okay, so let our channel to get spread to many people, let it get reached to many students, so that everybody can utilize the knowledge of this class. Right? Okay, let us talk about the nephron structure. See, first in the nephron, we are going to see a cup-like structure like this. So this cup-like structure is called Bowman's capsule. And after that, it is going to show many foldings like this. Again, there is a loop-like structure. Again, there is a folding. So this overall part we are going to call tubular part. Tubular part. But we have given a separate name for these tubules. So this part particularly called PCT. You are going to learn about this chapter in your class 11th because there is a special chapter called Excretion Excretory, waste and their elimination. So I have done that video also in class 11th. It's a beautiful chapter as well as interesting chapter too. Okay. So this is once again basic for that chapter. If you have understood the concept of these nephrons and all, then there it becomes so easy for you. Okay. So PCT means what? Proximal convoluted tubule. In fact, in biology, there is a meaning for every term. For example, see, I am telling proximal convoluted tubule. What is the meaning of this? Proximal means very near. So Bowman's capsule says, you tubule means tube. You are very near to me, right? So proximity means near. Proximal convolutions means many foldings. So it is present very near and it is showing many foldings. So what is this? That is nothing but tubule. Therefore, it is called proximal convoluted tubule. And you are very distant. Okay, from me, you are in a very distant place. So therefore, you are distal. Same, you are also showing convolutions. Convoluted. It's a tube part. Therefore, it is also called tubule. So in a short form, we can call DCT. And this one is Henle's loop. Henley's loop might be the name of the scientist Henley. He might have identified this location or the structure of the nephron. Therefore, it is termed as Henley's loop. It is a loop-like structure. Like that, many nephrons are going to be joined to a common duct like this. There is a common duct which is going to get joined by many nephrons. So this duct is called collecting duct. So this is what the structure of nephron. That's it. Now I said the nephron is a filtrating unit, or it is a structural and functional unit of the kidney. Why it is a structural and functional? Structural because all these things are present within the kidney given a shape. And why functional? Kidney is going to do a function called filtration. When it is going to filter the blood, then only urine is going to form. So we know that urine formation occurs through kidney only. But exactly in the kidney, which structure is doing this function? This nephron. Okay. So let me explain that part also. Look at this. It is your textbook image. This is they have mentioned here, Bowman's capsule we have understood. Tubular part of the nephron we have understood. Collecting duct we have understood. But here they have mentioned about the branch of renal vein, capillaries, as well as the glomerulus, renal artery. What are these terms, ma'am? You have not explained this. Let me explain, don't worry. Look at here. Now you tell me, what is this? Bowman's capsule. This is proximal convoluted tubule. This is Henley's loop. And this one is the distal convoluted tubule. And finally, they join to become a collecting duct. Correct? Now, if you just recall, or let me show the image only. So here, the renal artery is joining like this towards the kidney, right? So this structure, what you are seeing? This structure, what you are seeing, no? It is called arteriole. Why arteriole? Because from the artery this branch is arising. Therefore, it is called arteriole. Now, this arteriole is entered into Bowman's capsule. See, this arteriole is coming. It is entering into this Bowman's capsule and it is going to become a very small pipe further. See, further it becomes small pipes. So, these pipes itself we are going to call as what? Capillaries. The capillaries itself considered as glomerulus. Because it's a part of Bowman's capsule right now. That means attested with the Bowman's capsule. And this one is afferent. Afferent means they are going towards the Bowman's capsule. Therefore, it is called afferent arteriole. Okay. And finally, this capillaries, that is the glomerulus, they join to become, further, not away. Further, this capillaries only. They become artery only. But they forgot to give blood. They have forgot to give oxygen to rest of the part. So, therefore, they decided, okay, let us branch like this. And let me go by taking one of the branch around this Bowman's capsule, around these structures, and let me supply blood to all these things. So, they are supplying glucose, oxygen, everything to the nephron part also. So, whenever we talk about the artery, of course, the cells, they must give back carbon dioxide, right? So, that carbon dioxide will be collected from these structures. They become a vein. So, now, see, they become a vein after collecting the carbon dioxide. So, they are going to join like this. They are again going towards the what? Renal vein and join to the heart. Okay. So, this is what the renal vein, this is what the capillary. So, these were the blood vessels. But it is not a part of our excretory system. It is a part of circulatory system. Why they are present? Because they have to give oxygen. They have to filter the blood. Therefore, they are occupying like this. Okay. Now, the question, how our kidney is going to filter the blood? How the filtration process occur? How urine formation process occur? Let us understand. If you just read your textbook, you will get confused. But if you understand this concept and then if you read the textbook, you will understand everything. In fact, urine formation in our body occurs by 3 steps. One is by filtration. There is a specific term called glomerular filtration. So these are the three main steps. So from this, the urine formation occur. One is glomerular filtration. Second one is tubular reabsorption. Third one is tubular secretion. I will tell you. Look at here. Okay, so this is our Bowman's capsule, correct? So now, what I have told, this is afferent arteriole, and they are branched to become glomerulus. And they further join to become a capillary. That is what I said, right? So this is also nothing but blood capillary only. Now, here what happened? The blood which is so taking all the material. Say for example, the glucose is there in our blood. The things what we have eaten. So the glucose have also been filtered here. Actually, which material has to get filtered? In our body, urea is the nitrogenous waste. Urea has to get excreted out. Okay. But of course, urea will come. But along with urea, water will also come out. Glucose will also come out. Many amino acid will come out. Many minerals will come out. In fact, all these things are very essential to our body. But still, all these things have been filtered here in this part. That is in the Bowman's capsule. Through this glomerulus. So this is the first step in the urine formation. Okay. So this is called glomerular filtration. But our body thinks, you are doing all these things because for the glucose only, man. But if you just remove all this glucose in the form of urine, what is the purpose of having the food? What is the purpose of doing all these things? So you have to retain glucose in the body itself. You have to retain 90% of water you are excreting out. You should not do like this. So that's why here the tubular part is there, no? Tubular part means PCT, Henle's loop, DCT, all these things, they will decide. What they are going to decide? Whatever the material that they are present in this filtrate, we are going to just absorb it. We will just dump back into the blood. Okay. That means here the cells which are so there, they are going to absorb this glucose. They will dump into the blood. They will absorb the minerals. They will dump into the blood. They absorb the water, dump into the blood. So like that, glucose, water, many amino acids, so many molecules that will be get reabsorbed from the filtrate. That is the second step in the urine formation. Okay, that is called tubular reabsorption. But funny thing is, while absorbing everything, or while absorbing the necessary material, our cells of the tubule, they are going to absorb many ions also. For example, hydrogen ion, potassium ion, they also started absorbed by the cell. But such ions, if it is present excess in our body, then it is a problem. Okay, so that's why blood thought that it should not be there. For example, imagine here H plus ion is there, potassium ion is there. All these things should not be present excess in the blood. So therefore, blood says, you man, why you have absorbed everything? So you said, I am going to reabsorb only necessary material, but you have absorbed everything. No, I will just give it back. Okay, that is called secretion. I am going to secrete it back. So therefore, many things further secreted within the tubules. That is the third step called tubular secretion. So these three processes will be happen. Okay, first process will happen only in the glomerulus part. And after that, this tubular reabsorption and secretion that will take place throughout the nephron. That is in PCT, DCT as well as Henle's loop. For example, suppose if the filtrate comes, see, by running through Henle's loop as well as it came near the DCT. Imagine now the filtrate in the DCT. Now the cells of the DCT will also think, okay, whether this person has drunk enough water or not. Whether in the body of this person enough water is there or not. If we have had enough water, then it will be removed out in the form of a urine. That means the filtrate will be comes out in the form of urine. That is through collecting duct. Suppose if you have not had enough water, then body thinks, that means this DCT thinks, she has not drunk enough water today. Okay, let me do one thing. The water which is so present, let me just reabsorb it back into this blood. So again, it will dump the water into the blood. So like that, by looking at the body condition, it is going to absorb the materials as well as the water and dump back into the blood. And finally, the waste material which our body thinks, okay, this is the nitrogenous waste, this is excess water, this is excess mineral, then that will be removed out here in the form of urine. This is what the three steps of urine formation. So this much information is enough for you. As I have told, no, when you come to class 11th, you are going to study in detail how these processes are going to happen in all this tubular part. Okay, so hope I'm very clear about this. Now, the urine which is so coming out through collecting duct, how it is ejected out through urethra. Let us understand that. That process we call it as micturition. Okay, so let me draw here. Okay, imagine here the collecting duct, they are actually releasing urine. So from each kidney, from the collecting duct, the urine is coming. Now this urine will move through this ureter. So every minute it will be keep coming and collecting in this urinary bladder. This is urinary bladder, correct? Earlier, the urinary bladder is not in a bulge shape. It is somewhat flaccid like this, flaccid. As the urine start getting collecting like this, then it start getting expand. Okay. So imagine the urinary bladder has been completely filled by urine. Now it got expand like this. But how we are going to release it through urethra? It is not a voluntary signal. I mean, it should be comes from our brain. Imagine this is our brain. So here, there are so many receptors are present. There are certain cells are present. These receptor cell, they have connection with the neuron. So these neuron will be connected to our brain. So now imagine the urine droplets start accumulating here in the urinary bladder and finally it touches this receptor. That means expanding receptor. So it will be taken the signal and given to the brain. So brain thought, okay, so this bladder has been completely filled. Now it start giving permission to this urethra. That means the signal will come from the brain to the urethra. Now you can open and you can release the urine. So by that time we'll just search for the washroom, will feel urge to urinate. Okay. So this process of release of urine through urethra is called micturition. Understood? So this is what the overall process of excretion in human being. Now when it comes to plants, okay, let me explain this topic also because it is actually given in a box as no more, but it is important. Okay, what is this? Imagine the function, we know that the function of two kidneys are nothing but filtration. Suppose if any one of the kidney has got failed, then another kidney is enough to filter the blood of a person. That is the body. Suppose if two kidneys get failed, then what happened? Then how come the blood is going to filtrate? The person will get die, correct? So therefore, there is an alternate method for such people. That is in the case of kidney failure. So that alternate method is nothing but artificial kidney or hemodialysis. So here what happened? The person will be taken to the hospital. There a machine will be present and that machine work like a kidney. That's why we call it as an artificial kidney. I will tell you, it is very easy, very simple to understand. Imagine he's a guy. Now he's crying because his both kidney has got failed. Now this is the hand of this person. In this hand, the arteries are there. This is the artery. So in this artery, the oxygenated blood is coming. Correct? Now what happened? Here is a machine. This machine is called dialyzing unit or dialyzing machine. So in this dialyzing unit, what happened? They are going to put a liquid. Okay, imagine here is the liquid. So this entire liquid is called dialyzing fluid. Dialyzing fluid. This dialyzing fluid is just like our blood plasma, but it do not have any nitrogenous waste. That's it. So in our blood plasma, minerals are there. Here in dialyzing fluid also minerals are there. Electrolytes like potassium, chlorine, everything will be there. But urea, nitrogenous waste will not be there in this dialyzing unit. Okay. Now here what happened? The person, his kidney has got failed, right? So his kidney has got failed. So that's why here the blood will be taken through the syringe. Here we have injected a needle. So through this needle, we started taking the blood of this guy and that blood will be passed to there is a nitrocellulose paper. So here is a tube, this tubular structure. This is a porous tube in fact. Okay, it is a porous tube and in this tube the blood is moving. This is made up of cellophane. Okay. So blood is moving. Blood is circulating. So now the blood, which is consists actually nitrogenous waste, correct? Because both the kidney have got filled. It is not filtrating anything now. So that's why the blood is having nitrogen in it. I mean nitrogenous waste in it, urea in it. So that's why all this urea that will be ejected out, all the nitrogenous waste will be ejected out, that will be removed out. So that will be keep collecting. So imagine here, through this structure, they are keep collecting the nitrogenous waste. And every time, through another way, they are going to send this dialyzing fluid. Because the dialyzing fluid is also become contaminated, no? That's why every time they will be keep sending here the fresh dialyzing fluid. And the contaminated, that is urea, everything has been mixed, that will be collected here in the another bottle. Okay. So like that, the person's blood will be purified through the cellophane porous tube. And finally, the blood should not be ejected out, or else he will get die. So blood will be dumped back into his body after being filtrated. So how they are going to dump? See here in the vein. This is the vein of the person. Now this same blood will be again connected here after being filtered, and that will be dumped into this vein. Okay, so this entire dialyzing unit is considered as artificial kidney, as here the blood of a person is filtered by taking it out is called hemodialysis. Okay, so hope this much is enough, and this much information you have understood, that is more than enough for you. Now let's move on to the plant case. So in plant's case, okay, one second. Okay, it's not there. Let me write here. So in plant's case, it is very simple. Plant says, we are not like you guys. In our body, so many cells are dead tissues only. So why should I bother? Majority are dead tissue. If anything, if I consider it as a waste, I will just make it to accumulate within the leaf, and I will make the leaf to get fall. Okay, so the fallen leaf consists the waste material. So like that, as we know that the gum we are going to obtain from the plant body, the resins are going to be obtained from the plant body, but it is actually useful for us. But for the plants, these are the waste material. So they will be just accumulate here, they will be just release it from the body as a waste material. Okay. So like that, if you just take out a cell, imagine here cell is present. If I just take out a cell of a plant, nucleus will be there, but there is a big structure, we call it as a vacuole. So in this vacuole also, they are going to secrete the waste product or a waste material. So like that, they are going to excrete their waste from their body itself. So this much information is enough for you. Nothing to worry. Okay. Now, if you read your NCERT, that is your textbook, you will understand each and every line, every concept. Okay. So this is what the overall chapter. Hope you enjoyed all the concept. Thanks for watching.